Okay, I think this should be just about right. If everyone is ready, we can begin.
Perfect.
Fantastic. I hope there was time for that coffee. Job well done. So I would like to thank everybody for participating, for agreeing. I understand there's a lot of factors at play here, different time zones. Everyone has other stuff to do and ongoing projects. So we really appreciate the help, the assistance, and the participation. Prior to commencing with Bryan Fry, Professor Theodore Abatzopoulos, who is the person responsible for allowing this to happen within our university, he will take the floor to give the opening speech, and we will continue with Bryan Fry right after.
Okay, thank you very much. Good morning to all. I would like to ask for your forbearance to say a few words in Greek for the Greek attendees. The rest of the webinar will be held in English. Θέλω να καλωσορίσω το ελληνικό ακροατήριο, φίλους και φοιτητές που παρακολουθούν ή θα παρακολουθήσουν αυτήν την εκδήλωση. Όταν ο Θωμάς Δάφτσιος, που είναι δευτεροετής φοιτητής του Τμήματος Βιολογίας του Αριστοτέλειου Πανεπιστήμιου Θεσσαλονίκης, ζήτησε την αρρωγή μου για να οργανώσει το σημερινό webinar, ευνηδιάστηκα ευχάριστα για τρεις λόγους. Πρώτον, γιατί μια τέτοια σημαντική προσπάθεια ξεκίνησε από έναν φοιτητή. Δεύτερον, γιατί η σημερινή ημερίδα θα ρίξει φως σε ένα πολύ παρεξηγημένο θέμα. Και τρίτον, γιατί το πάνελ των ομιλητών απαρτίζεται από εξέχοντες παγκοσμίως γνωστούς ομιλητές. Σας εύχομαι λοιπόν μια ενδιαφέρουσα παραγωγική παρακολούθηση. And now in English. I'm really happy to welcome both guest speakers and attendees on today's webinar. This event is one of the very rare occasions held in our country and now is released. I am referring to the very interesting part of biological sciences nearly never targeted properly in Greece, which dealt with toxin and venoms from aposematic animals and venomous snakes. It has been a popular demand for such an event, and thanks to some motivated students with the catalytic role of Thomas, has been successfully organized with the title Venom and its biological roles. We are delighted of hosting some of the most distinguished scholars and researchers for covering a broad range of themes which include, among others, microbiology, genetics, toxinology, toxicology, herpetology, venomics, clinical care of snake bites, and zoology. All these thematic areas secure the interdisciplinary approach of this event. I would like to welcome our guest speakers one by one, starting with Sir David Warrell, Professor Emeritus from Oxford University, UK, José María Gutiérrez, Professor Emeritus from University of Costa Rica, Bryan G.Fry, Associate Professor from University of Queensland, Australia, Juan José Calvete, Research Professor from Institute of Biomedicina of Valencia, Spain, Wolfgang Wüster, Reader of Zoology from Bangor University, UK, Timothy P. Jenkins, Assistant Professor from Danish Technical University, Denmark, Carl Barden, Director of Med-Toxin Laboratories in Florida, USA, Vasiliki Moulasioti, Doctorate of University of Juan, and last but not least, Ilias Strachinis, Doctorate in Aristotle University of Thessaloniki. I would like to wish you all a very fruitful and interesting attendance. Thank you very much.
Thank you. Thank you, Professor Abatzopoulos. Like I said before, we don't really know a lot about this specific segment in our country. It's kind of vague, not a lot of experience, even though we do have venomous species, 5 of them when it comes to snakes. And if it wasn't for Professor Abatzopoulos, essentially this wouldn't have been possible. So again, thank you very much for this and for making this happen. So continuing, we will take the chance and carry on with Professor Bryan Fry. If that's okay with you, you can take the floor.
Sure. Okay, well, thank you very much for having me. I really appreciate it. and I shall see if my hate-hate relationship with Zoom continues. Start sharing the screen, although you think we'd all be masters at this by now. All right, so just checking that you can see the first slide.
Absolutely.
All good? All right, woohoo, step one. Okay, so we all know venomous snakes cause a myriad of issues, and that the evolutionary processes that make them so absolutely fascinating from a theoretical perspective also underpin the absolute clinical nightmare that they can be, where just even a couple 100-kilometer change in, and you can have, you know, a completely radically different venom phenotype. So how do we make sense of this, where we have so many venoms in so little time and so little resources, particularly with the current funding crunch that's happening around the world? Now, a lot of the historical methods for ascertaining bioactivity are excellent methods, like the chick-by-venter assay, for example. Superb, gives you an insane amount of information all at one go, where you can discriminate between presynaptic neurotoxic effects, postsynaptic neurotoxic effects, and myotoxic effects all in the one assay. So you get a lot of information, very reproducible, but it's very low throughput. You can only run a few at a time. Lots of manual steps from the dissections through to the actual setting up of the organ baths, establishing the tension on the wires, etc. And naturally, these can lead to errors where, you know, the dissection itself is fairly simple for the chick-by-venter side of it. Mouse phrenic nerve preparation, the mammalian equivalent, nowhere near as straightforward and is actually quite error prone. So you can get a lot of issues. Similarly, if you're looking at other types of tissue preparation, such as the aortic ring, doing endothelium denuded assays, really easy because the endothelium is denuded and damaged as you make it, as you set up the assay. So therefore, doing endothelium intact assays is really tricky, requires very experienced pairs of hands, and therefore is low throughput, manually intensive, and error prone. So what's the solution? Well, anyone who knows me knows that I'm a geek and I like toys. So we've been setting up a lot of high throughput robotics-based platforms for neurotoxicity and coagulation testing, and we're now setting up for cytotoxicity. Now, these have the advantage of you're getting rid of the meat puppets, you're getting rid of your humans, you know, all right, rise of the machines. This means you can pump through an incredible number of samples. For example, with our Octet HTX, we can run up to 96 samples simultaneously every 15 minutes. It's fully automated, where it's fed by the robot. We have a 2-meter Hamilton Vantage robot that feeds the plates into the Octet. So you can set up literally days worth of experiments and go do something interesting like look for snakes outside. And it's reliable when we plot the data for the Octet or for our equivalent machine, the Stego StarMax, on the blood side of things that we call Dracula, that we actually get error bars when we try to plot the data in Prism, because it says that the error bar, we get an error message because it says that the error bars themselves are smaller than the line icon and therefore can't be visualized. That's probably the best problem to have. All right, so we'll start off by looking at the coagulation side of things. And toxins can do one of two things. You can have an overdose scenario where the clotting is turned on. This is your procoagulant. And this is where you ultimately have the generation of endogenous thrombin, of prothrombin being converted to thrombin, and then the endogenous, the body's own thrombin, then in turn converts fibrinogen into very stable, long-lasting fibrin clots, which are the lattice that form the very backbone of a blood clot. And this, in a prey item, will cause them to stroke out very, very quickly. And as anyone who's ever seen anyone having a stroke, you know that this is a very rapidly incapacitating event. So long before a prey is dead, if you induce stroke in it, it can't fight back or flee or jump or run away. The opposite scenario is anticoagulant, and this can be done in 3 different ways. And we often see all 3 happening simultaneously to different degrees by a different venom. So direct inhibition of the clotting enzyme, so blocking your factor 10A, blocking your thrombin, or further upstream enzymes. That's an excellent way to be anticoagulant. You can also be anticoagulant by directly depleting the amount of fibrinogen, the ultimate building block, through direct destructive non-clotting cleavage of fibrinogen. So the fibrin Fibrinogen goes from fibrinogen to degradation products with no clot in between. But then you have a third type that we call pseudoprocoagulant, where the venom itself acts directly on fibrinogen in a thrombin-like way, but thrombin-like only so much in that it forms a clot, but it forms an unnatural clot. The fibrin clots are disordered, and therefore they're very weak and they break down extremely quickly. And this contributes to a net effect of depletion of fibrinogen. So it's an anticoagulant effect. But in a lot of the literature, you see a lot of errors in the literature because they talk about anything that forms any kind of clot as being procoagulant. Well, a venom acting directly on fibrinogen to form a weak transient clot that depletes fibrinogen levels and completing to an anticoagulant action is not procoagulant. True procoagulant is generation of endogenous, the body's own thrombin, where the thrombin itself, the natural thrombin, acts on fibrinogen. So you need a way of discriminating between the two types of clots. You need a way of measuring clot strength. And we do that through thromboelastography in our lab, which is an excellent way of telling you what kind of clot that you have. Because if you're doing something on a plate and you're just looking for changes in the ocular density, just turbidity, that tells you nothing about what kind of clot it is. And whenever we suspect that we have a true procoagulant venom, we always measure in specific assays exactly what factor is it turning on. We'll cover some of that sort of work. Bit of background that there is no guided evolution of any sort. There's no hungover methamphetamine intelligent design fairy saying, poof, have another toxin. No, it doesn't work that way, of course. The target selects the toxin, where some things like, for example, down there on the bottom at 15, where you have thrombin inhibition. Well, this is an excellent way if you're a blood meal feeder like a mosquito, or if you're some of the viparrid snakes that are best described as Ebola with fangs and that kill by hemorrhagic shock. Taking out thrombin is an excellent way to promote either of those two very different kinds of functions. So therefore, if you have a toxin that binds to thrombin, like any other trait, if it confers a benefit, however small or incremental, that trait becomes fixed and amplified. So therefore, so you want to think of it as, that since the target selects the toxin, we should see a lot of convergence. And that's exactly what we see, that there's a number of different sites that are much more amenable. Because, for example, giving your prey animal cancer will ultimately kill it, but it's not going to help you very much on the short term. And venom, of course, is all about immediacy of action. So as mentioned, that our way of looking at the coagulation side of things, our first port of call is always Dracula, because it can run 8 samples simultaneously and just keep pumping those out. And each run, if it's a procoagulant venom, you get your result in as quick as 6 seconds if it's a brown snake. So very fast acting assays, and it can fully automated. We can set up racks of trays and just let it rip. Extremely reliable, and has a number of different types of assays in there, where we can make anywhere from using plasma inside the cuvette through to we'll actually make up our own artificial pathways to work out exactly what is acting where. So we can do procoagulant, true procoagulant, pseudoprocoagulant, anticoagulant, anything that we want by changing what goes into that cuvette. The machine itself has a refrigerated rack, and it can hold 78 different reagents in that refrigerated rack and take whatever volume we want out of each one and any possible combination that we want. So we have infinite flexibility of setting up assays. So it can be running one cuvette on one assay and another cuvette on a completely different assay simultaneously. Good example of the utility of this is that in the Australian elapids, at the base of the Australian trees subsequent to the radiation by Micropicus, we have the evolution of a true procoagulant trait. And this is, in this case, it's the recruitment of one of the natural blood enzymes, factor 10A, for use as a weapon. And this therefore combines with the body's endogenous factor 5A to bind to prothrombin, to activate it to thrombin with that body's own thrombin, then in turn acting directly on fibrinogen to form strong, stable clots, resulting in catastrophic strokes. But the key here is that even though it was an early evolving trait, it didn't become amplified until much later in a number of convergent lineages. What we have here is the organismal tree. The warm colors, your red, are where that trait has been amplified. And you can see it's been amplified to a number of varying degrees. For some of them, such as our whip snakes, it's nowhere near as potent as what we see, such as like in the brown snake, where we have an insane clotting time of 5.4 seconds. So we see, a number of different lineages, some of which were quite surprising, where some of them are very rare and little or no bites on record. So for example, about a third of the way down, Pseudopunctata, that came out as potent as a tiger snake. And even more interestingly is we see an ontogenetic shift where we have a juvenile that we estimate was about a yearling. And we want to try to get some actual true neonates. We reckon that they're going to be almost not acting at all in the blood and probably much more potently neurotoxic, which is something we've seen in Pseudonia, where baby Pseudonia are neurotoxic nocturnal lizard specialists, while adult Pseudonia, which are the Australian brown snakes, are daytime active, blood acting mammal specialists. So radical changes ontogenetically is with precedent in Australian elapids. But with Pseudopunctata, that's not only the first documentation of potent procoagulant activity within that entire genus, but also the first documentation of ontogenetic shift. And also down there at the bottom in Hemiaspis, about 20% up the way from the bottom, diminutive snakes, very rarely encountered, only know of one bite on record, and it did produce a blood disturbance. And as you look at the potency, that's getting up there in potency to tiger snake level, and it's certainly more potent than some of the lethal snakes out there. So it goes to show that one of those snakes, if it's large enough, could produce a very significant bite. We've done the antivenom testing, quite variable antivenom responses, so which fits, of course, to the fact that it goes back 25 million years for this particular trait. We can also see very extreme variations within a species. So this is looking at different Bothrops, but particularly the Bothrops atrox populations. So the red line is venom only, blue line is venom and antivenom. We're looking for a nice shift. So for example, the San Bento population, a big shift, really potent neutralization of the venom. But if you look at the Santorin population, virtually no action by the venom. What's interesting is that Santorin are your floodplain snakes where they're feeding a lot on frogs, animals with a very high prey escape potential, where it's obviously you can't scent trail a frog. If it jumps in the water, it might as well go through a wormhole. It's gone. That snake is never going to see that frog. So there's an extreme selection pressure for a fast-acting venom because of the prey escape potential, which is, so it's not just what you eat, but what are the chances of that prey being able to escape, or in the case of the taipans, where they're feeding on very dangerous prey, the chance of prey retaliation. So in the case of taipans, people ask, why do they have so much of such potent venom? Well, they're feeding on animals that could very easily injure or even kill a snake. They're a specialist for dangerous prey. In the case of the Bothrops, different selection pressure, and that's prey escape. Now, what's interesting is that the overall activity of the venoms, if you look at the potency, they're quite similar in potency, but the Santorin one is more potent. But the key is that the Bothrops venoms are procoagulant through two mechanisms simultaneously. One is activating factor 10 into factor 10A, which then in turn converts the body's prothrombin to thrombin. The other activity is acting directly on prothrombin to convert it to thrombin. So simultaneously, you have the clotting cascade being turned on. Just through historical contingency reasons, we worked out that the antivenom is being made using populations of Bothrops that happen to be rich in prothrombin activating toxins and comparably poor in factor 10 activating toxins. And as the case turns out in the Bothrops atrox Santorin population, and also independently Bothrops nuidi, both of those are very rich in factor 10 activating metalloproteases. So even though it's the same toxin class, snake venom metalloproteases, the ones that are activating factor 10 are geographically and biochemically just slightly differently enough to the ones that are activating prothrombin that the antivenom cross-reactivity is very poor. So it's a case of not just making sure when you're having antivenom manufacturing that you have the same toxin class represented, but the functional groups within a toxin class are just as important, because here we have the exact same toxin class, enzymes that are biochemically and structurally very similar to each other, but just enough difference that there's not much cross-reactivity. So it goes to show that, like, so that dynamic diversification that makes them so interesting makes them so frustrating and so challenging from the treatment option side of things. We also do a lot of work looking at venom resistance. So anecdotally, in the Australians, that the blue-tongued skink is, has been anecdotally thought to be resistant to snake venom. And just after we published this study, a birder, it's always a birder. It's never a reptile person who sees such cool things, but it's always someone out looking for plants or birds. Sent me these photos on the left there. Thank you, Victoria Austin. You're awesome, but So she sat there and watched this red-bellied black snake try to feed on a blue tongue. Bit it savagely over and over again. She could see blood. Clearly it was envenomated, but it was completely unaffected. And eventually the snake just gave up and crawled away. And I love the belligerent attitude of the bluey. No fear. It's like, yeah, come have a piece if you think you're hard enough. Yeah, that's it. Keep slithering. Just love that attitude. And it fits perfectly with the results that we have, that the blue tongue is very resistant to Pseudechis porphyriacus, the red-bellied black snake, very resistant to that venom. The related skinks that are, in the same genus aren't, like Reticulus rugosus, aren't resistant to the same, anywhere near the same level. So it's something very specific to the blueies. They live alongside the red bellies. The blue-tongued skinks are, of course, slow-moving, quite vulnerable prey animals as a result. So it makes perfect evolutionary sense that they would be resistant. But what really caught us off guard is that if you look at figure A in the top there, Cryptophis nigrescens and Demantia semophis, as I mentioned in the previous work, have independently amplified that factor 10A trait. And what's interesting, and Pseudonia textilis also contains factor 10A as a major enzymatic component of their blood-acting venom. The blue-tongued skink is not resistant to their venom. So their resistance is not a general resistance to factor 10A, but it's super selective for the type of factor 10A toxins that are in the red-bellied black snake venom, that through simple evolutionary drift is up to maybe 80–85% identical to the toxins found in Cryptophis or Demantia. So highly similar, and same biochemical action, but different enough that there's no cross-reactivity, which is absolutely fascinating to see that level of selectivity. So we certainly didn't expect that. We expected to see resistance, based on the anecdotes, but we didn't expect to see something that cool of that level of selectivity to the resistance. That was very unexpected. Now, moving on, this is what I consider our most medically important and relevant work. And if anyone ever asks what the study of snakes has to do with human medicine, you point to, of course, the antivenom side of things. So David Warrell published a report describing, as he puts it, the deplorable medical practices where Indian antivenom manufacturers make antivenom that's quite affordable, but it doesn't necessarily work outside of India, but they're selling it in Africa saying, hey, look, you have saw-scale vipers, we have saw-scale vipers. Our antivenom is much cheaper than the South African antivenom, buy it from us. So David reported a 20-fold death rate increase in hospitals that switched antivenoms. Now, if you're selling a life-saving product, it is manslaughter at the very least if you sell it without doing any testing to show that it worked. They did none. So we tested it, and we tested a number of different antivenoms. So here's the results for VINs. So looking at the bottom there, that's the Indian antivenom. Works very well against one type of saw-scaled viper found in India. That's just the results are happen to be within the Pakistani populations with the same subspecies. But you can see that it fails against another type of saw-scaled viper, in that particular case from North India, fails completely. So Indian antivenoms have problems even within India. And most of the Indian antivenom is made using venom collected around the Chennai region by Ram Whitaker and the Arula snake tribe. They have a collective there. So you have a very narrow geographic region that's being represented. But you can see there where this is the organismal tree. Warm colors are where the antivenom works. Blue and black is where the antivenom fails. You can see an absolute total failure across everything in Africa. Now, in comparison, we have the ICP antivenom on the top, which is made against West African saw-scaled vipers, works brilliantly against West African saw-scaled vipers. Unsurprisingly, fails against East African saw-scaled vipers. In contrast, the South African antivenom is made using a 2-to-1 ratio of East to West African venoms. And as you can see, it works twice as well against the East African, but still picks up the West African. Neither of them work against Indian or anywhere in that region venoms. We sent them our results prior to publication, got no response, and they're continuing to sell their antivenoms in Africa. Rob Harrison and Nick Casewell did a follow-up study looking at additional snake types in addition to looking at saw-scales again with a broader range of antivenoms that they bought in Africa that are being sold, Indian antivenoms. And of course, they corroborated our findings showing the total and absolute failure. To me, this is murder. If you know it doesn't work, it's murder. And unfortunately, this is symptomatic of the despicable and predatory practices that plague the developing world, such as you can go back to things of medicines being tested without proper consent, through to things like the AIDS scandal of the US pharmaceutical companies that worked out ways of treating their plasma to kill the virus, and then dumped all their previous plasma stocks on Asia for pennies on the dollar, causing a massive bloom of HIV infections across Asia, leading to $1 billion lawsuit and 6 of their executives going to jail. But of course, the ramifications of that are still being felt today, which is what sparked off the massive AIDS crisis in Thailand, Vietnam, and Hong Kong. And of course, that's continuing to burn today. So unfortunately, the antivenom side is just symptomatic of the much more fundamental problem, that basically people are scum. All right, so moving on to, it's like that classic study that showed that, what it takes to become a head of a major corporation is, you fit all the check marks for being a sociopath. And, so it's unsurprising we end up with things like this. So looking at the nerve side of things, just like with the blood, we see the same kind of convergence of targeting, that certain sites are more amenable for being used as are more vulnerable to actions like toxins, like hitting calcium channels, for example, is an excellent way. Postsynaptic nicotinic acetylcholine receptors have been hit a number of times independently just by the snakes. So our way of looking at these is using the octet, where we can make artificial nerves effectively. And what that allows us to do is, first off, study animals that it would be ethically or legally impossible to do, such as a study we just published this week on primates. Good luck, for giving our, we'd like to, kill 40 gibbons, thank you. And of course, then we'd like to make a gibbon-human hybrid of the receptors. Yeah, good luck with that. Even if you had unlimited funding and an island located on a tropical island, or a lab located on an island on tropical coastline somewhere of a country with no extradition laws, you'd still run into logistical issues of good luck, making some of those kinds of assays. But using artificial nerves, we can study all sorts of prey selectivity and also the evolution of resistance in a number of different lineages. So when we did our first validation study, we compared a generalist, the death adder, with a fish specialist, the olive sea snake, and a snake specialist, the king cobra, Ophiophagus, as the Latin name tells us, snake eating. And unsurprisingly, as you can see, the death adder, that there's no real selectivity, that you can see that, from the lines above, that, they're very close in potency across. Mapping that over the organismal tree of the different prey types, you can see there's very little difference in the relative potency. In contrast, unsurprisingly, our fish specialist was more potent on fish, and our snake specialist was unsurprisingly more potent on snakes, therefore giving us the patterns that we would expect and validating the method. So we've looked at a number of different lineages. For example, like with a lot of the cobras, they've been sort of commented about that, even though we see three-finger toxins in their venom, including, classic alpha neurotoxins, we don't see much neurotoxicity even in our rat assays, let alone human clinical effects. We'll get to the human side of it later on, but even on the rodent assays, we don't see much in the way of neurotoxicity side of things. Nowhere near, you might see some, but nowhere near the level. Well, cobras love frogs. Like when I do a lot of field work for cobras up in Hong Kong, and the first way that we find a cobra is we find the frogs. We find where the frog soaks are that have a lot of frogs, and wherever there's a lot of frogs, we have a good chance of finding cobras. And unsurprisingly, based on what we know in their natural history, most of them are extremely potent on frogs and more potent on frogs than other taxon. Some variation, of course, but you can say as a general rule, they're much less potent on rodents, so you wouldn't see very much effect for a lot of these venoms on a rodent-based assay. Now, honey badger, of course, is well known to everybody as not caring. The infamous video, honey badger don't care. Dreybeck did a study a number of years ago where she sequenced the alpha-1 of honey badgers, postulated a couple sites where it had mutations relative to other mammals as being potentially conferring the resistance. But of course, she couldn't do any assays, whether with native honey badger tissue, the honey badger organ assay was not going to get ethics approval, of course, even if you could manage to catch enough honey badgers, let alone being able to do mutant studies of putting it in, changing residues. Good luck putting the honey badger alpha-1 into the oocyte and expressing that. That could take you about 10 years just to get one variation to work. You change one amino acid, you reset the assay, and you got to redesign the parameters all over again. So her work remained theoretical. So we tested that where she had postulated two residues in particular, a tryptophan at position 187 or a phenylalanine at position, an arginine at position 187, which is in place of the tryptophan that you normally have there, or a leucine at position 189 that normally has a phenylalanine. So we tested the native, the cobra venom didn't bind to the native. We tested a mutant where we put it and swapped the arginine for a tryptophan, and bang, massive amounts of binding. However, the position 189, where we swapped the leucine for a phenylalanine, we didn't see a change in the binding. So position 187, that single amino acid change is what confers the resistance. Now, in the same study, we did a look where Burmese pythons are slow-moving terrestrial snakes living alongside of 2 to 2.5 meter true cobras or up to 5 meter king cobras, snake-eating snakes. Of course, once they hit adulthood at 6 meters, they're not going to be vulnerable to predation, but they, of course, start out as small snakes, small, slow-moving snakes. So young to medium-sized Burmese pythons and presumably other type, terrestrial pythons are exquisitely sensitive to, or exquisitely vulnerable to predation. And we showed that they're actually extremely resistant to snake venom alpha neurotoxins. And we documented that it's due to a fascinating mutation in the receptor where at position 191 and position 195 there on the top, you can see that there's a lysine. Lysines are positively charged. And so, but in the ancestral state, there's a D or an E, there's an acidic residue in place of it. So you have a negatively charged state in the typical ancestral state that we see in different pythons. And so what this means is that, and that negative charge is something that's common across most animal postsynaptic nicotinic acetylcholine receptors, is to have a negatively charged orthosteric site, the binding pocket where acetylcholine binds. So if you have a negatively charged site, that puts strong selection pressure for the evolution of toxins that are positively charged, because that helps guide the binding. There's, of course, other interactions that are going on, but that opposite charge attraction is one of the major drivers for the initial binding. In this case, the replacing a negative charge with a positive charge creates a same charge repulsive effect. So it's like trying to bring the same side of two magnets together. It's not going to happen. So in this case, we have an electrostatic repulsion occurring of the neurotoxins. And we don't see that in the snakes in Australia, the black-headed pythons and the carpet pythons that aren't being predated upon by snakes rich in alpha neurotoxins, or the Macklitz python of New Guinea, or the reticulated python, which as babies are highly arboreal. By the time they come down from the trees, they're big snakes. And in that case, they are slammed into by the neurotoxins. So we found a number of different cases. We found 8 different independent evolutions of this charge reversal to create an electrostatic repulsion. Another example, obviously don't have time to go through all of them, but another example is the mole snake of South Africa, slow-moving terrestrial snake, living alongside of a lot of snake-eating alpha neurotoxin-rich cobras. As you can see in that middle picture of a Cape cobra trying to predate on one, the middle and the right. But in that particular case, it bit it, seemingly without effect, and the mole snake eventually slithered off after the cobra gave up. Same sort of thing, where it has a change in position 191. In this case, only one of the residues is changed to a negative or to a positive charge, but that single introduction of a positive charge is enough to repel the neurotoxins, giving them very, very strong resistance to the alpha neurotoxins. And sometimes even just getting rid of the positive charges is enough, where savanna monitors, slow-moving, vulnerable prey items, their larger relatives, the black throat monitors, are also vulnerable, but they in turn also eat cobras when they get large. That particular case, that's a cobra having a go at a black throat. Person sat there and watched it, and the monitor eventually got bored and walked off. So in this case, they don't have the repulsive effect, but they've mutated so they no longer have the attractive effect. And as you can see, we've compared them to the Komodo dragon and the parenti, which have an identical nicotinic acetylcholine receptor, or the Mertens monitor, or a mutant where we put negative charge on both sites. That as long as you have a negative charge and no positive charge correspondingly, one negative charge is enough to attract the neurotoxin. So that's why, like in the mutant there, if you look at you can see some increase in attractiveness with a mutant for some of the venoms, but nothing significant. And in some cases, it's not even any difference. But in contrast, getting rid of a negative charge at either site that we see in the savanna and the bigularis dramatically reduces the affinity of the neurotoxins, and therefore conferring some level of resistance. They're not as resistant as a Burmese python or a mole snake, but they're much less sensitive to neurotoxins than any of the other monitors that we've tested. Now, here's a paper that we just had come out this week on neurotoxin resistance in primates. So the African and Asian primates are living alongside of a large number of big neurotoxic daytime active snakes. Not that they're trying to predate on the snakes aren't trying to predate on the primates, but there's a lot of accidental encountering. There's a paper that came out a year ago showing that the rise of humans shaped the evolution of cobras, of driving the evolution of spitting in 3 occasions. Well, here we've shown the reciprocal, that cobras have shaped the evolution of primates. And in particular, if you look at the organismal tree where the cold colors are the most resistant, if you look at the last common ancestor of humans, chimps, and gorillas, it evolved a much greater degree of resistance than the already resistant African and Asian primates. Now, what's really striking is that for this theory to hold true, we need to see an absence of resistance where there aren't these kinds of evolutionary selection pressures. And sure enough, that's what we see in our South American clade, where the neurotoxic snakes there, the alpha neurotoxic snakes, are nocturnal, small, burrowing coral snakes. And you can see by the bright red colors, extremely sensitive to alpha neurotoxins. Similarly, at the very bottom there are Madagascar primates, where there are no venomous snakes. I mean, there's some poultry rear fang snakes, but they really don't count. Yet again, extremely sensitive to alpha neurotoxins. So those kinds of results are just as important for our hypothesis as the fact that, with the snake venom detection theory, shaping primates, and such as eyesight, the changes behaviorally and morphologically in the last common ancestor of chimps, gorillas, and humans, coming down from the tree, spending a lot of time foraging, including moving objects, including quite substantial objects to forage, inquisitive, attacking snakes. There have been well-documented cases of chimps using branches to attack cobras, so aggressively encountering snakes, that it makes perfect evolutionary sense for there to be a shaping pressure for the evolution of resistance. It's needless to say, this paper is giving primatologists absolute aneurysms. Just looking at another look here, where what we did was, as I mentioned, we're using artificial nerves, so we can look at the structure-function relationship. So we made all sorts of mutant versions where we swapped the human version with that of sensitive primates to work out which are the key residues conferring sensitivity. And it's position 187 and 189, where we have a serine in place of the ancestral tryptophan, and we have a threonine in place of the ancestral phenylalanine. So just like the honey badger, position 187, but also 189, where we have a very strong biochemical change between the threonine and the phenylalanine. So yet again, these were the kind of structure function studies that would have been impossible using any other method. So kind of looking at a little bit more, going further afield, looking at, so we've published quite a bit on blood-acting toxins from monitor lizards and other anguiomorph lizards, where they share the same kind of toxins that we see, like from the Gila monster, such as the ability to block platelets, the epinephrine-induced platelet aggregation pathway through to destruction of fibrinogen using the exact same type of kallikrein enzymes. Well, we just published on the first neurotoxins, where it had been known that Heloderma were neurotoxic, but the mechanism of action had remained uninvestigated or undetermined. And we report that it's acting on the domain 4 of the NAV 1.4 type of sodium channels, and that monitor lizards also act at that exact same site. And in fact, the arboreal Varana salvadori, the crocodile monitor, where it has a very high chance of prey escape, is actually about twice as neurologically potent as the Gila monster, or and even more potent than, again, for the beaded lizards. While the lace monitor is about as potent as the Gila monster at that same site. We also showed that they hit a lot of these ion channel toxins are very promiscuous. So a lot of times you see crossover between sodium and calcium, because sodium and calcium channels are each other's most recent molecular ancestors. They evolved from a last common ancestral ion channel. So we see a crossover, and yet again, the salvadori is about twice as potent as the Gila monster, while the lace monitor is about as potent as the Gila monster. So this was a hugely fun study to do, and we're now working out exactly what toxin types are doing that. And so for our last one, stonefish also had been known to cause clinical neurological symptoms, but this hadn't been recoverable in the lab, and we worked out why. And that's because if you freeze dry the venom, that's where the LY venom is there, you lose the activity for the binding to alpha-1. Versus if we use fresh venom from our stonefish in the top there, our reef stonefish that we've named Space Cake. We like to have names for our animals. That the fresh venom is extremely potent on alpha-1, but the freeze-dried venom lacks that activity. In contrast, though, there's another type of neurological activity where they're not acting on sodium channels, but they are acting on calcium channels. And in this particular case, the type of toxins involved are not sensitive to freeze drying. So a completely different toxin class is involved. So thank you very much for your time and attention. And I shall stop screen share.
Thank you, Brian. Thank you very much. There is a question written in the Q&A. I don't know if you can see it, or I can read it out loud.
No, I just have the chat. Oh no, there's Q&A over there. Okay, so considering venom variability, when it comes to the treatment options and antivenom efficacy, are current antivenoms in Australia effectively treating snake bites? And that's a yes, no, maybe kind of question, where Australia hasn't changed their antivenom formulation since about the 1950s, and nor are they, because despite Australia using venomous animals basically as tourist icon, more people die of weird sex in Australia every month than die every year from snakebite. On a really bad year in Australia, we have 2 people die. Average is only about 1. We only have a few hundred bites. For the usual suspects, the antivenoms work brilliantly, to the point that our monovalents are boutique products. We really don't need them. Our polyvalent covers a narrower spectrum of snakes than polyvalents on other parts of the world. If you look, for example, at the African polyvalent and the approximately 60 million years of evolution covered by that polyvalent, the Australian one, is just incredibly, like I said, it's a boutique to have a death adder antivenom but nevertheless, Australia can afford it. Having a monovalent does allow you to use less antivenom, but on the other hand, the Australian antivenoms are so incredibly almost over purified that they have a fraction of the adverse reaction rates that we see for overseas ones. Like in Australia, the adverse reaction rates are in the lower single digits, while some of the cheaper and nastier overseas products, so for example, the Thai Red Cross one is well known as being extremely allergenic and having an incredibly high, like up to 80% adverse reaction rate. Very well documented, have anaphylactoid through to true anaphylactic reactions, and serum sickness all the way to catastrophic crashes. Australia, it's very, very low, which combined with pre-medication makes it even lower. So it's incredibly well purified. As far as the efficacy, by and large, yeah, it's, I would say that, for the snakes that are doing the actual biting, we get very good coverage, and so, even for the ones where it's not working as well, you just keep pumping a bit more in and it'll eventually cover it. Like with the brown snakes, for example, brown snakes from Queensland, you can mop those up with a couple of vials. Brown snakes from the Northwest, like, going north of Broome, I've heard of cases of it requiring up to 20 vials. It eventually reverses, but it's unlike situations such as like that Sean Bush had published with the Southern Pacific rattlesnakes, and the work he's doing at Loma Linda, where he's pumping 60 to 80 ampules of antivenom into people and then still dying. So in Australia, it's a matter of the antivenoms by and large will work if you pump enough in, and what we consider a lot is still trivial compared to what happens overseas. So we've had some cases of some very rare snakes, like with some of the work we're doing with Suta, the curl snakes, where the antivenom worked against the neurotoxicity, but didn't touch the myotoxicity. But you're not going to die from their myotoxicity. You're just going to be really hating life for a few months as your muscles repair themselves.
Understood. Well, I also got a couple of emails, people asking about your work with the Komodo dragons, the Arctic octopus, and someone mentioned 57 bites. Is that correct? Snake bites.
No, there's been a few along the way, but just like Lewis Hamilton has wrecked a few cars along the way.
Okay.
We try not to dwell upon that, but shit does happen, and the key is being prepared. Like with anything else, that the snake bites are sometimes the least of your problems. Where I've broken almost as many bones in the field as I've had snake bites in the field, including breaking my back in 3 places on one occasion, and requiring 4 months in the hospital and massive surgery, and which is why I have a spine that looks like Wolverine now on X-rays. So that's why, like, when we go really remote, like up to Ashmore Reef to do sea snake work, we bring a doctor and antivenom with us. And Wolfgang and I lost a friend of ours in Burma who was in the middle of nowhere, and they didn't have a doctor and they didn't have antivenom, and a bite happened. Things do occur. So you need to be prepared for those kinds of extractions. And if extraction isn't feasible, where in this case extraction was delayed because of inclement weather, that you need to be able to treat something in isolation, whether a snake bite or something more mundane like a compound fracture in a bone sticking out of someone's leg.
Understood. Okay, well, maybe we can elaborate on the Komodo dragon and the Arctic octopus bits in another instance, if that's okay.
Yeah, so start with the octopuses. So I always wanted to go to Antarctica, and I needed to come up with an excuse, and obviously it wasn't going to be reptile driven. And so basically my entire, these grant applications are ridiculously long, but my entire application could be summed up on on a post-it with a very large crown, which is nobody's looked at the venom. So we went down there. We're part of a 2-month expedition, part of the Census of Marine Life. Anything with venom was ours, and we were doing benthic trolling all the way down to 4K below the surface. So we got anemones, we got bristleworms, we got 254 octopuses. Now, we had done work showing that all octopuses are venomous, not just the blue-ringed occhi, but all occhies. Like if you dive anywhere in the world, go dive in the Mediterranean, you'll see clamshells that are empty, and there'll be a hole in the side. Well, that's where an occhi has drilled through the side and injected their venom. Go down to your local fish shop, buy an octopus, cut it open. Just above the eyes, when you cut into the mantle and open it up, you'll see a pair of really big discs. Those are the venom glands, and that's a shared trait across all occhies. So the blue ring gland just happens to be one of the few that's medically important. But we know that some of the are like people, aquarium keepers, public aquariums here in Australia know that if you get bit by a Maori octopus, your arm is going to swell up like a balloon. It's well known among the keepers that you're going to get a big puffy arm that's going to be like that for a few days. So we're really interested in particular in the occhies. Of that, we DNA barcoded everything because I didn't know anything about any octopus. And in the process, we discovered 4 new species, one of which went into an entirely new genus. I did no work on Antarctic occhies before we went down there, knew nothing about the taxonomy. So much to my great surprise, it turns out that Antarctica has giant octopuses. We got 3 of those, including one that had a mantle like a beach ball. And we have a picture of me holding it up like this, and the arms are down on the ground, and there's still about 1 meter of arms on the ground. And I'm a big guy, and I'm holding it up, and even so, there's still a lot of arms on the ground. It was awesome. The venom glands on that were probably about 5 inches across and about a ½ inch deep. They were amazing. So what we wanted to do was see what was in there, but also, was there a temperature dependent action? So that's been a long-standing question, even within the snake field, of will a venom enzyme act differently if it's injected into a frog that has a body temperature of 20, or a mammal at mid-30s, or a bird at 40? These kinds of questions haven't been tested. We're actually, with our robotic equipment, we can actually adjust the temperature of the plasma. So we're now actually doing those kinds of tests with amphibian, bird, and mammalian plasmas at different temperatures. So we did that with the Antarctic occhies. Was we measured the speed of action of their toxins, and unsurprisingly, considering that they're living in water that's down to -2 or colder, because salt water, of course, has a freezing temperature much lower than fresh water, which is why you put salt on the road to ruin paint jobs on cars and to get rid of the ice. So when you're down at 4K below in the polar areas, you can have it can be -10 down there. So unsurprisingly, the toxins were more active at 0 degrees, and that if we took blue-ring octopus similar toxins and put them at 0, we've killed the activity. They just kind of stopped moving. But conversely, if we tested them at sort of tropical water temperatures of the mid-20s, the blue-ring octopus toxins were at their happy sweet spot, while the Antarctic occhi toxins denatured. They fell apart at only in mid-20s. So we had antifreeze venom. Now moving on to the Komodos, this comes to a case
for this one, maybe
we
should keep it for the next bit, the next time we see, so that it is the main theme of the presentation.
Oh, right.
If that's okay.
Right. Sure. Of course, no problems.
Because we're a
nice
bit
cliffhanger.
backtracked a little bit.
Leave them wanting more.
Yeah, absolutely. Well,
everything you've heard on Discovery Channel is a lie. Of course, you already knew that.
Well, that's the plan.
Basically, Komodo dragons have been the reptile equivalent of Shark Week.
Okay, well, thank you very much for this. Bryan Fry has also shared with us a couple of documents, essentially 2 books. I will be sharing those with everyone.
And also, if you go to my webpage, Screw a Sylvia, I put all of my papers up on my webpage. I'm sure Wolfgang does the same. So all the papers can be downloaded up there. Still need to put a few. It hasn't been updated for a couple of months because of a baby, and oh no, we're publishing so much. We're about 6 papers behind, but I'll get them up eventually. But it's science to the people. I mean, we're paying to do the work. People are reviewing it for free, and then we pay to publish, and then they charge people to access it. The mafia wish they had a rocket like this.
Well said.
All right, well, thank you very much. And sorry that I won't be able to attend anybody's talks. I've been up since 4. Who knew that babies get up early? And I'll probably be up again early tomorrow. So I'm gonna go get some sleep.
Hopefully there's gonna be a second chance for the Komodo dragon.
Oh, glad to.
So people looking forward to that.
All right. Take care. Thank you again.
Thank you very much, Bryan. Okay, well, first off, I would like to apologize for the backtracking in time. Wolfgang Wüster is the next speaker upcoming. So Wolfgang, you can take the floor and we can backtrack again a little bit by 5 and 10 minutes moving on forward, if that's okay with everybody.
Okay, well, thanks very much indeed for the invitation and asking me to be there. It's a great pleasure and a privilege. I'm now going to try and share the screen. It worked earlier, which probably means it's not going to work now. Let's see. Can you see that?
Yes, absolutely.
Okay. So I'm going to talk basically about how snakes use their venom, which is kind of what I'm most interested in, and some of it will overlap with things that Bryan was talking about earlier. So how snakes use venom primarily in defense and offense. So first things first, snakes are just another bunch of animals living in the wild. They interact with other species, either as prey or as predator or in other ways, and that means that all aspects of their biology are subject to natural selection, largely based on these interactions. They're subject to selection pressures just like any other animal. The venom of venomous snakes is just part of the interface between the snake and its environment, and therefore it's also subject to natural selection. So if we want to understand venom and venom evolution, what we need to understand is a bit about snake biology and what the snakes actually do with their venoms. So first things first, what's snake venom? I'm assuming most of us here are well aware of the fact that venoms aren't single substances, single chemicals. They're complex cocktails with a very large number of bioactive proteins. And that extreme complexity of venoms is, of course, what leaves room for variation in venom composition. And that variation in venom compositions occurs at all levels. Bryan already talked about ontogenetic variation. It also occurs between conspecific populations of one species and also between very closely related species. And that variation is, of course, both interesting from an academic evolutionary point of view, but it's also the scourge of anyone trying to treat snakebite and deal with antivenoms. And Bryan referred to Echis. It is the classic example. If you're a West African farmer and you get bitten by Echis ocellatus and you go to the hospital and you get an antivenom raised against Echis ocellatus, your chances of dying are perhaps of the order of 2 to 3%. And that's mostly those cases who simply get to hospital so late that it's too late to actually help them. On the other hand, if your local hospital got sold venom by an Indian manufacturer who raised the venom against Echis carinatus from Asia, then your chances of dying are more of the order of 15%, which is pretty much the same as if nothing was done whatsoever. That venom simply doesn't work. That antivenom simply doesn't work against Echis ocellatus. You might as well be injecting dishwater. So venom variation matters both in academic terms for evolution and so on, but it also matters in terms of medicine and snakebite treatment. And it's important that we try to understand what happens and why it happens. So why is venom so variable? What selective pressures drive venom evolution is really what we're asking. Most of this stuff is going to be driven by natural selection. And to understand that, we need to think what snakes actually do with their venoms. And really, it boils down to 2 major functions of venom. One is diet, foraging. Snakes use venom to overpower their prey. And the other is defense. If we talk about diet first, snakes are in a relationship with their prey. They exercise selective pressures on each other. A lot of venomous snakes specialize in eating relatively large, well-defended prey, like this gaboon viper here eating a cat. Clearly, if you want to eat a cat, that's a well-defended prey item. That cat could do a lot of damage to the viper. So therefore, there is selective pressure on the snakes to optimize their venom for their prey, to overpower it effectively before the prey can either escape or fight back and injure the snake. So that leads to a prediction, which is that venom should be optimized to the prey that the snakes eat. And you would expect, for instance, either very specific activities to be higher towards diet taxa than towards others, or higher lethality. And Bryan showed some examples in the lab. So what we're showing here, some examples of it, of testing it on real live prey. The example again are saw-scaled vipers, echis. One of the delightful things with echis is that many of the species eat a lot of invertebrates, particularly well-defended invertebrates like scorpions or centipedes. I don't know if you've ever tried to eat a live scorpion or a live centipede, but I reckon you want that dead pretty quickly before you start swallowing it. And among the echis species, there are 4 major species groups, and they differ quite radically in their diet. So Echis carinatus in Asia and Echis pyramidum in Northern Africa, those 2 groups have a diet dominated by arthropods, mostly things like centipedes and scorpions. Echis ocellatus is a bit more vertebrate heavy in its diet, and Echis coloratus very rarely eats any arthropods. So we've got very, very different diets in these 4 species. And we then explored whether there was any difference in the lethality of the venom of these 4 species groups towards arthropods, particularly scorpions. And what we found is a quite remarkable level of agreement between diet and arthropod-specific lethality. So the 2 heavily arthropod-eating species, Echis pyramidum and Echis carinatus, had venoms that were extremely lethal to Scorpiomaurus. It took very little venom to kill them. Echis ocellatus, with its more kind of 50-50, more mixed diet, had venom that was less lethal to the arthropods. And then Echis coloratus, and for comparison, Bitis arietans, which are very much primarily vertebrate eaters, their venom was extremely weakly lethal to scorpions. So that was one of the early examples where we were able to show that these venoms are, these snakes very much a venom that's tailored to the diet that they are eating. Clearly, carinatus and pyramidum and ocellatus have some toxins in there which very specifically kill arthropods. Interestingly, the mouse LD50 for these species doesn't actually differ very much at all. And in the meantime, there have been a large number of examples of species and species groups where, again, we found evidence of adaptation of venom to diet. So some of these were shown here, and there's more of them accumulating all the time. So in the meantime, the idea that diet is a major driver of venom evolution, of the evolution of different venom compositions, has actually become the sort of basic paradigm. If you talk to anybody about, oh, these snakes have different venoms, then many people will start to say, okay, well, what do they eat? So that's become very widely accepted. So venom is essential for foraging by snakes. So therefore there's selection for prey-specific venom, plenty of examples on there and accumulating all the time. And that also then leads to prey resistance against venom. So the snakes are under selection to optimize their diet. The prey, in turn, are under selection to escape the venom of the snakes, to resist it. And what you end up with is predator-prey arms races, where both the prey and the predator play a kind of biochemical cat and mouse game, trying to overcome resistance in the case of the snake and trying to escape venom action in the case of the prey. So natural selection for foraging is likely to be a very common and widespread driver of venom variation and venom evolution in snakes. Not for everything, but widespread. So what about defense? So snakes are part of ecological food webs, just like any other animals. They have predators, and clearly predation exercises natural selection. And snakes are under natural selection to avoid predation, just like any other animal. The question is, to what extent does this actually drive the evolution of venom? So how do snakes use their venom in defense? Well, we actually know surprisingly little about that. It's very well documented from humans, of course. That's how we know what snakes do with their venom in defense. We know they use it because that's how people get bitten all the time when they step on snakes. This was a film producer in Costa Rica who stepped on a Bothrops asper and felt sorry for himself for quite a long period of time. But we only really know very much about it in the case of snake-human conflict. We know remarkably little about the use of venom in snakes against natural predators. And we don't know very much at all about the extent to which selection for a defensive function has been important in driving the evolution of venom or the venom apparatus. So we need to think a little bit. If you're trying to use venom for defense, what do you actually want to achieve? And to understand that better, we can look at other animals with defensive venoms. And the answer is what you need in your venom if you want to use it for defense is you need a venom that causes instant severe pain. And I'm sure most of us here will have been stung by a bee or a wasp, which have totally defensive weapons, especially honeybees. And if you get stung by a honeybee, it hurts straight away. You don't get stung by a honeybee and an hour later you think, oh, I say, it starts to hurt a bit. No, it hurts straight away. Lionfish, well, if you want to know what a lionfish sting is like, go to YouTube and search for lionfish sting. And there's some beautiful videos of divers getting stung at 40 meters and screaming into their regulators all the way from 40 meters down to the surface through all the decompression stops and all the rest. It must be phenomenally painful and it's instant. That's what a defensive venom is predicted to do. The function, of course, is to repel any predator before the venomous animal is damaged so badly that it's going to die in turn. There's no point in deterring a predator an hour or two after you're dead. That doesn't help you. So what's the story there in snakes? We predict defensive venoms. Venoms that have been selected for defense should cause instant severe pain. There are certainly well-known exceptions to that. So this is Bungarus ceruleus, the Indian crate, and crates are notorious for getting into people's houses and biting sleeping people at night. And people either don't wake up from it at all, or they wake up, but there's no pain, there's no swelling, nothing's happening. So they just go back to sleep and think nothing's happened. And then they wake up in the morning and they're close to death from severe neurotoxicity. But is there a widespread pattern? Is there something, can we generalize across snakes whether most snake venoms were selected for defense or not? So to try and find out more about that, well, we've got, Bryan talked about all these assays. There's another available assay, which is that lots of people do stupid things with snakes and end up getting bitten, and we can simply ask them what actually happens in terms of pain. And this was done by my honors project student, Harry Ward-Smith, who ran an internet survey on pain after snakebite. If anybody in the audience filled that in, thank you very much. And the basic idea was just to test the speed of development of pain after snake bites. Now, if a venom evolves primarily with a foraging function, there's no real specific prediction of pain. The snake doesn't really care whether its prey item dies rolling around in agony or whether it peacefully falls asleep. And one might predict that in a foraging venom, pain might increase fairly slowly as a byproduct of other venom activities. It may well get very painful later on as other venom activities develop, but you wouldn't particularly expect it to be painful early on. And you might also expect it to be quite variable because there's no strong selection for or against pain in a foraging venom. On the other hand, in a defensive venom, we would expect to see what we see in a bee sting or a lionfish. We would expect to see very rapid increases in early pain reaching its maximum within what we could think of as an ecologically relevant time span, which is where the snake would have a chance of getting away from the predator before it's too badly injured. What was found, basically, more or less, the general picture was more consistent with the idea of the foraging venoms in all the major snake groups. We're finding that the early pain is relatively minor compared to the maximum pain level experienced later on during the course of envenoming. So this shows, again, the pain in the first minute, after 5 minutes, and the maximum pain experienced after the bite. And you can see that that maximum pain later on is much more severe than the early pain in all these snake families. We can also look at some individual pain trajectories. So these are individual pain trajectories for Bitis erytans, the puff adder, and Crotalus atrox, the western diamondback. Now, both of those are snakes that you really don't want to get bitten by. They're well known for causing bad tissue destruction and things like that. The colored lines are individual cases, individual pain trajectories, and the dotted lines are the mean for both of these species. And you can see in both of them, the puff adder and Crotalus atrox, the early pain is really quite minimal, and it stays relatively low within the first 5 minutes, and it only gets severe much later on. So not at all what we would expect from a defensive venom. And the other really remarkable feature, we asked how long it took for people to suffer pain so severe that they couldn't carry on doing what they were doing. The only thing they could think about was the pain. And remarkably, that within the first 5 minutes, that only happened in about 15% of bites. Okay, in about 35% of bites, it happened after more than 5 minutes. So the kind of time where it really wouldn't benefit the snake because it would already be dead. And in 55% of bites, it never actually happened at all. So in 55% of bites, people never suffered pain so severe that they couldn't sort of focus on their normal activities anymore. So really that militates quite strongly against the idea that most snake venoms are heavily selected for defense and for causing pain. So upshot of that, the dominant pattern is that snakebite pain is a relatively slow onset. No strong evidence for generalized selection for defensive function. Now, obviously, a questionnaire survey like that is extremely noisy. It's quite subjective. People's memories may be faulty. So yes, it's a pretty, I'm not so much painting with a broad brush as throwing a pot of paint around the room. So they're likely to be exceptions. So where might we go to find exceptions? Well, the first port of call is probably these guys here, cobras spitting in cobras. Now, spitting in cobras is the one adaptation of the snake venom apparatus that is purely defensive. Spitting cobras bite their prey just like any other snake. Spitting is reserved for situations where they're defending themselves against predators. The other thing that's really nice here is that spitting evolved 3 times independently. It evolved in a group of African Naya, it evolved in a group of Asian Naya, and it evolved in the genus Hemacartus from Southern Africa. And that's great because it means we have 3 replicate experiments and we can test ideas, what we would expect to happen, and have 3 different examples, have 3 independent experiments to test our predictions. So spitting cobra venom in the eyes is extremely painful. On the skin, it normally has no effects, but if you get it in the eyes, then it's excruciatingly painful by all accounts. I've never had it happen, but by all accounts, it can be excruciatingly painful, causes massive inflammation of the eyes, and can lead to permanent eye damage. So it's a very effective long-distance defense for these snakes. I think we're fairly familiar nowadays with the morphological adaptations, particularly the fangs, where in spitting cobras, the fang directs the venom forwards when it's squirted out under pressure. In non-spitting cobras, it goes downwards. Spitting cobras have quite sophisticated behavioral adaptations in terms of, not just aiming at the face, but making sure that they cover the face of the opponent and adjusting the amount of head wobble according to the distance and even anticipating movement. So they're actually pretty sophisticated in that. What we had no idea until now was to what extent venom composition is adapted to spitting. Do spitting cobras actually have venoms that are more effective when applied in this long distance defense way than non-spitting cobras? Now, previous work suggested that while most cobra venoms have cytotoxins, and cytotoxins cause tissue destruction, and it was suggested that the pain in from cobra venoms would come from cytotoxins and presumably also the ocular pain in spitting cobras. Now, to try and look at that in a bit more depth, we were part of a multi-center collaboration that was led by Nick Casewell and Talene Kazanjian from Liverpool School of Tropical Medicine, and basically evolved a huge range of different analyses of venom composition, and also a whole series of functional assays, primarily where we tested to what extent different venoms and different venom components activated sensory neurons, which would be what causes the perception of pain. And the first thing that we found was that, so this is again the phylogeny, the evolutionary tree of cobras. And the color of the branches indicates how potent the venom is in causing nociceptor activation, i.e. pain, and the warmer the color, the more red the color, the more effective, the less venom it takes to actually activate pain sensations. And you can see that the three spitting groups, Hemacartus, the African spitting Naya, and the Asian spitting Naya, are particularly potent in causing pain, in causing nociceptor activation, whereas non-spitting cobras and the outgroups generally are fairly weak in causing nociceptor activation. And interestingly, that wasn't related to the presence of cytotoxin. Most cobras have cytotoxin in varying amounts, and the pain had nothing to do with, or very little to do with, the presence or otherwise of cytotoxins. So looking at the venom composition of these different groups, so these pie charts here indicate in light blue cytotoxic three-finger toxins, then other, mostly neurotoxic three-finger toxins, PLA2s, metalloproteinases, and other toxins. And we can see that all of the cobras have a variety of cytotoxins, and it doesn't associate with spitting or with the amount of pain the bites cause. On the other hand, the thing that is quite striking is that the presence of large amounts of PLA2s, phospholipases A2, was largely restricted to the spitting lineages, the African spitting cobras and the Asian spitting cobras and the ringcults, Hemacartus, with a little bit of noise in there in the case of Naya subfulva, which nonetheless doesn't cause much pain. But basically, PLA2s were strongly associated with spitting. So can the PLA2s explain the pain caused by cobra venom? Could that be an adaptation to make venom in the eyes by spitting cobras more painful? We then basically dissected the venom and tried the different components of the venom to try and understand the mechanism by which these venoms cause pain and activate nociceptors, thereby causing pain. So we had representatives of the three different spitting lineages, Hemacartus, Naya siamensis for the Asian spitters, and Naya nigricollis for African spitting Naya. And okay, PLA2s, all the spitters have a lot of PLA2s, they're more painful. Does the PLA2 cause that? No, it doesn't. The PLA2s do nothing in terms of nociceptor activation. The PLA2s do not cause pain. Okay, what about the cytotoxins? Nope, the cytotoxins don't really explain it in full either. They do cause nociceptor activation, which you would expect for a tissue-destroying toxin, but they really didn't explain the full level of nociceptor activation caused by the whole venom. On the other hand, if you mix the PLA2s and the cytotoxins, then you get high levels of nociceptor activation and you start to recapitulate the pain caused by the whole venom of these snakes. So the pain seems to be caused by synergistic action, complex synergistic action between the PLA2s and the cytotoxins, where basically the PLA2s seem to potentiate the nociceptor activation caused by the cytotoxins. And you can see that's exactly the same mechanism in all three of these spitting lineages. Hemacartus, Asian spitters, African spitters, all do the same thing in terms of increasing the pain caused by their venoms. So we have three independent origins of the same complex toxin synergy that's associated with the evolution of spitting. So why did this happen? Why did spitting evolve? And why did we have this evolution of pain-causing venom associated with spitting? Well, to try to understand that, we need to start thinking, okay, when did spitting evolve and what else was happening in the world at the time? So when did spitting evolve? Well, the origin of the African spitting cobras goes back to somewhere between 5 and 8 million years ago, most likely 6.5, 7 million years ago. The origin of spitting in Asia goes back about 2.5 million years ago. In the case of Hemacartus, unfortunately, we can't really tell because it's a single species, but watch this space. So what else was happening at the time? Well, one other thing that was happening was actually us. Okay, so here's the phylogeny of primates. And what you can see is that the evolution of the African spitters corresponds almost embarrassingly well to the lineage split between the chimpanzee and all the hominins, including ourselves, as well as Neanderthals and Australopithecines and all the rest of it. So basically, the evolution of spitting may have happened at pretty much exactly the same time that our ancestors started to come out of the forests and walk on two legs while still using tools. The Asian spitters, in turn, 2 million years ago, or just over 2 million years ago, is where we have the first instance of hominins, Homo erectus, in Asia. So that corresponds very well as well to the origin, to the first hominin presence in Asia. So could they be related? Well, Harry Green, who many of you will have heard of, has been arguing that case for quite a while, and he calls the existence of spitting cobras Lucy's legacy. Lucy refers to that very well-known fossil Australopithecus afarensis from about 4 million years ago, which is one of the earliest human fossils that really documented how we evolved being bipedal first and having big brains and stuff later on. So what's the evidence there? Well, we know that primates are really nasty animals, and primates are well known for killing and injuring snakes, even if they're not planning to eat them. They sometimes eat them, sometimes they don't. They just go out of their way to kill and injure snakes. They use tools. A lot of primates will use sticks to bash snakes or will throw stones at them. And as the first hominins came out of the African forests, all of a sudden, not only did we have these animals that came out of the forest were more terrestrial than any of the others, more likely to meet cobras, but now they were walking on two legs. So they had both their hands free to get up to no good and start bashing snakes. Now, what does that do to the snakes? Well, to a lot of snakes, that just results in increased selection for long distance defense. If you have a predator that's using long sticks or throwing stones or things like that, you don't want to be having to bite it. You want to be able to fend that predator off from a distance before it can hurt you with its tools. So the idea that hominins may have been the selective factor behind the evolution of spitting is actually quite powerful here. It's the common factor, certainly between two of the two events of convergent evolution, the Asian and the African Naya. For Hemacartus, we need to see. But certainly for two out of these three events, it's the origin of hominins that seems to be the common factor behind these events. So I think it's a remarkable example of how we should think slightly differently about human evolution. When you talk to anthropologists, they often talk about human evolution in isolation from their surroundings. But really, when we think back to it, when our ancestors first walked out of the African rainforests, we were just another link in the food web. We were being eaten by other species. We were eating other species. Other species exercised selection pressures on us to avoid predation, and we exercised selection pressure on other species. We were just part of that food web. We were part of the system. And other species impacted our evolution, and we impacted the evolution of others. And in the case of cobras, Bryan showed his very nice results on how the presence of cobras may have impacted our resistance to cobra venoms, but quite possibly it may be that the fact that our ancestors were horrible to snakes and we're using tools to do that, that's the reason why we have spitting cobras to this day today. We impacted their evolution and they impacted ours. And I think that's a fascinating way to think about just, human evolution isn't some separate thing from the rest of the natural world. We were part of that environment. We were just another species in there, and we were impacted by and impacted that system millions of years into the past. So to sum it up, take-home messages. So first of all, venoms are part of a snake's interface with their environment, and therefore they're subject to selection, and venom evolution is driven by a whole bunch of interactions with other species in the environment. So that's predator and prey. Snake venom, again, isn't something that we should look at in isolation. And in order to understand that better, we really need to know more about what snakes do with their venom, how it works on natural prey as opposed to any other prey, and just how the ecology of venom works, how snakes use venom in the natural world. Lab experiments are great, but we need to understand better how snakes actually use their venom. And we really need more studies. And for those of you in Greece, you've got some great model species. Always think of this species here, Viperamidites, as a fantastic model. It lives in various parts in Greece and on the mainland and on islands where I think in some of these, at least, there's no native mammals. They're very different in size in some populations, probably different in venom composition and diet. So that would be a fantastic model to look in more depth at the ecology of venom and how that impacts venom evolution. So with that, thanks very much to a huge number of people that I won't all read out. And thank you very much to all of you for listening and for your interest. Thank you very much.
Thank you, Wolfgang. Very much appreciated. There's a couple of questions, see if we can just go through them very quickly.
Yep.
Would that be okay?
Yep, absolutely.
Would you want me to read them out loud, or you can?
Yeah, if you can read them out, that's, yeah.
So Argyrios Kurtidis essentially is asking, is there any research on the amount of venom released upon a snake bite for any species on offense and defense?
That's a really interesting question, and we don't have a huge amount of data on that. There was one study that looked at that, looking at a few pit vipers in the US by Bill Hayes and a couple of his students. And what they found basically is that, well, in any case, we already know that a lot of venomous snakes actually metavenom. So when they're foraging, they decide how much venom they're going to inject into their prey item, depending on, for instance, the size of the prey item. So that's actually fairly precisely targeted, at least in pit vipers. Again, very little research. In defensive bites, on the other hand, what they found is that there is huge variance in the amount of venom injected. And on average, it was actually more than in most predatory bites. There's this myth circulating that snakes want to save their venom and don't inject it in defensive bites. And that's simply not true. And if you think about it, if venom is used in defense, if you're defending yourself, you're about to die. Okay, that's strong selection pressure. That's much stronger selection pressure than, say, if I don't inject enough venom into that mouse, I'm going to miss my dinner. Okay, death is bigger selection than missing your dinner. And also from a simple behavioral point of view, you can also explain it because there was huge variance. And that's what we see in snakebite patients. Some people get bitten by a really dangerous snake and nothing happens. They have a dry bite. Other people get bitten by a snake that's normally not too bad, and they get a massive amount of venom and have a really horrendous experience. And if you think about that from a human point of view, if you go hunting sparrows, you're going to take a different gun than if you go hunting elephants. Okay, so when you're foraging, you adjust what you apply to your prey. If you're in a defensive situation, if you're camping at night in the forest and this really giant thing comes and suddenly attacks you, you're going to fire whatever you have. One day it might be a slingshot, another day it might be a grenade launcher, you know, and that's the variance we're seeing in snakebite patients. So that's the story. So that's the story there.
Well, thank you very much for this. The other one, super quickly, is did the research on the pain threshold study take into account the overall size, the mass, I would assume, of the people involved and their pain response? Would the results be different if, considering the snake's natural predators have less of a body mass?
The answer to that is no to all of that. I mean, in order to access that kind of data, we just had a questionnaire. We did ask about the sex of respondents and things like that, but we didn't have data on size and so on. So really, it was just the thing that we're really interested in is the trajectory of the pain. So the actual pain level, of course, is going to be really different between different people. Some people will smile if you hit them on a hammer. Another one will cry like a baby if they bump their head on a wall. But what we're interested in is the trajectory. Now, I don't know how people would score being stung by a bee out of 10. Some people might score it as 2 out of 10. Others might score it as 8 out of 10, but we're all going to agree it hurts straight away. And that's what we were trying to get at, how the pain at the start compares to the pain later on, whether the bite was really painful right from the start, like a bee sting, or whether the pain just came on slowly as a byproduct of other functions. So, I mean, it would be really interesting to answer those questions. It's quite a difficult one to actually do. I mean, the pain threshold is, because people are just so variable. I think unfortunately, that's pretty unanswerable. And again, even doing it with live animals, you'd have a major ethical challenge trying to do studies on pain perception in a large number of vertebrates in the lab. So unfortunately, questions like that are very difficult to answer.
Understood. Okay, well, Wolfgang, thank you very much for the presentation. Very much appreciated.
Thank you very much for the invitation, and I hope the rest of the symposium goes well as well.
Fantastic. Thank you very much. So we will now continue with Ilias Strachinis. I know we're, again, backtrack on time, but everything is all well considering the topic we have here. So Ilias, you can take the floor, one of our own from the Aristotle University of Thessaloniki, and some great work on all venomous snakes in Greece. Ilias, you have the floor.
Thank you very much. I'd like to thank you for the invitation and thank both panelists and attendees. Let me open my presentation. Sorry about that. Tell me if everything is okay.
Yes.
Can you see it? Great. So my presentation is going to be much simpler science-wise. In this presentation, I'm going to present the venomous snakes of Greece, focusing on the dangerous ones, and we will see how to identify them amongst all Greek snakes, how they are distributed throughout the country, and we're going to see some statistics about snake bites and deaths caused by snake bites in Greece. So let's get started. My name is Ilias Strachinis. I'm a Greek biologist and a PhD candidate in Aristotle University of Thessaloniki. I'm a member of Greek Herpetological Society and the owner of the website herpetofauna.gr, which is a website that contains photos and basic info of all reptile and amphibian species in Greece. Let's start by saying that all reptiles and therefore all snakes in Greece are protected by at least one of the following laws, conventions, or directives. And that means that in Greece, it is prohibited to kill a snake or to capture it or destroy its natural habitat. Everyone that is familiar with Greece, they should know that we have hundreds of myths about snakes in Greece. And I always start my presentations by showing this photo of a father and his child observing a viper crossing a dirt road somewhere in Peloponnese. And I always show this photo to show that vipers are not aggressive. They do not launch towards us, jump towards us to bite us whenever they see us. They're not so agile reptiles, and they will try to flee or stay still, to stay camouflaged as soon as they see us. So they're not aggressive. They will bite only if caught or stepped on, despite all the rumors. So let's see some very popular common myths about snakes in Greece. First of all, snakes chase people. No, they don't. They are afraid of us. They will try to flee as soon as they see us. Snakes are attracted by milk and drink it. No, they don't. Again, they don't even have the necessary enzymes to digest milk. Snakes whip with their tail, which is anatomically impossible. Their tail is very small, and the rest of it is the body that contains the vital organs. Vipers mate with moray eels when the moon is full. That's a good one. There are people that actually believe that a fish could breed with a reptile under certain circumstances. And last but not least, that the ecologists release snakes in the wild because they want to increase their numbers, and sometimes they do it by throwing snakes from a helicopter. That's a good one too. Sometimes people ask if snakes are useful to us, which is a very anthropocentric question. And but even as such, the answer is yes, because despite apart from being a very important part of the food chain and contribute to a well-balanced ecosystem from which our quality of life depends, they also protect our crops by controlling the population of rodents and insects. They protect the public health from spread of disease because they consume rodents and insects that sometimes are bearers of contagious diseases. And in the concept of this webinar, they contribute to the production of important drugs. Today, by taking advantage of specific toxins taken from snakes' venom, we can produce very important and effective medicines. So let's see how many snakes and which of them are venomous in Greece. We have in total 23 species of snake in Greece, and only 7 of them are venomous, but only 5 of them are potentially dangerous, and they're all species of vipers. The rest, 2 of the venomous species of snake, they belong to a different family, and they are Opistoglyphus, which means that the teeth that inject the venom are far back in the oral cavity, plus their venom is too weak to cause any serious damage to a human being. So 5 potential dangerous species of snakes in Greece, all vipers. How to tell a viper from a harmless snake? Well, at this point, let me underline that everything I'm about to say from now on, every rule or tip on the identification is only valid when we're referring to the snakes in Greece and Europe, and it should not be taken into account when we speak about snakes from other continents. It does not apply for snakes outside of Europe, so please pay attention on this. So all vipers in Greece share some common characters which we can use to distinguish them from the harmless snakes of Greece. First of all, they have a stout body. You can see a heavy built body, thick and short. Second, they have a wide head. Their head is broad. It's like protruding cheeks, like a pit bull or something. 3, they have a short tail that narrows sharply. As you can see, we have a thick body, and suddenly the tail narrows very fast. And 4, with an asterisk, they have a zigzag pattern on the along of their back, and this is with an asterisk because only the 3 species of viper bear this pattern. The other 2 have a different pattern, which we will see later on in this presentation. Here are some individual of the 2 most common viper species of Greece, the nosed horn viper and the Ottoman viper. These are nosed horn vipers, and these 2 are Ottoman vipers. You can see the robust, stout body, the short tail, and the broad head in all of these individuals. You can see the horn-like formation at the tip of the nose of the nosed horn viper. And you can see also the zigzag pattern along their back, where in some areas could be broken like here, but this should not confuse us because we can identify vipers from the combination of the 3 other common characters of the vipers: the stout body, the short tail, and the broad head. So, on the contrary, the harmless snakes of Greece have, as a rule, narrow heads. You can see the head is a lot narrower than that of the vipers. They have elongated, slim body. You can see how thin the body is. And they have elongated tail that narrows very gradually. Also, the tail is thin. So the harmless snakes of Greece can have a uniform coloration like this one. They can have spots along their back like this one. They can have blotches like this one here, or stripes, parallel stripes along their back. or a combination of blotches and stripes, but they will never have a continuous connected zigzag along their whole back, as vipers usually do, the three of the 5 species of vipers of Greece. There are some exceptions to these rules. The sun boa has a stout body, as you can see, but it has a very narrow head, a blunt tail, and they don't have continuous zigzag along their back, but it's like military-like camouflage coloration and not a zigzag. And also the European cat snake, which has a broad head and a coloration that is similar to the vipers. But as you can see, its body is very elongated and the tail too. And the pattern is disconnected with blotches along its back and not a continuous zigzag. Here's a viper to the right to compare with. So at this point, one should be able to identify a viper just from the proportions of the body. We have here 2 individuals, 2 snakes that have not the typical coloration. They're melanistic, so you cannot see the typical coloration of the species. One of them is a viper and the other one is a harmless snake. And just by looking at the proportions of the body, you can tell that this is a viper because of the stout body and the short tail, and this is a harmless snake because of the elongated body and tail, despite that both of them have a relatively broad head. So let me introduce you now to all 5 species of vipers of Greece, which are the only dangerous snakes for us in Greece. Let's get started with the nose-horned viper, which is the most common species of viper in Greece. You can literally find it almost everywhere. You can see the stout body, the broad head, the short tail in every individual, and of course the zigzag pattern along their back. It's a snake that usually reaches up to 85 centimeters at maximum. Usually they're smaller. Second, here are some more individuals of the nose-horned viper. Again, all the characters that I already mentioned. Second species of viper in Greece is the Ottoman viper. It's very similar to the nose-horned viper, a more robust snake and a lot bigger than, it gets a lot bigger than the nose-horned viper. It can reach lengths up to 1 meter, and sometimes it can exceed that length. So you can see that it also has all the characters and the zigzag pattern along the back. Third species of viper in Greece is the Greek meadow viper, which is an endemic alpine species of viper. It is actually a dwarf species of viper. We can find it only on the alpine and subalpine meadows of Pindos mountain range and nowhere else in the world. It reaches maximum 40 centimeters in length. So it's a dwarf species of snake, and it also has the weakest venom, and this makes sense because it mostly feeds on grasshoppers. Third species of viper in Greece, the adder. Yes, we have adders here in Greece too. The populations from Greece are at the southernmost edge of the species global range. We have a different subspecies in Greece. It's the Viperaberus bosniensis, and it has a different pattern than the northern populations. It does not bear a zigzag pattern on its back, but it has perpendicular bars along their back. The males have brighter colors than the females. And for Greek standards, this snake is also an alpine viper. We can find it from altitudes starting from 1,600 meters above sea level and higher. It reaches up to 65 centimeters long. And last but not least, the Milos viper, which is endemic to Milos archipelagos. In the typical coloration, they have a grayish beige color, and they do not have a zigzag pattern, but they have bars or squares or blotches along their back, which are brownish or sometimes orange. And of course, sometimes they can be in a uniform coloration like this one, orange color. This is a stunning individual that Thomas found on Milos Island recently, and I was there too. So here's a map of how these species are distributed throughout the country, throughout Greece. You can see with blue color the distribution, the range of the nose-horned viper. You can see it's almost everywhere. This map is in it shows the approximate range of the species. So you can see that the nose-horned viper, it's almost everywhere in mainland Greece and in many islands of the Cyclades, Sporades, or the Ionian Islands. With green color, you can see the distribution of the Ottoman viper. As you can see, it is distributed on many of the East Aegean islands, and in a small part of the mainland here in Thrace, where it lives in symmetry with the nose-horned viper. With yellow color, the Greek meadow viper along the Pindus mountain range. With purple color, 5 spots where the other lives. These are mountains in the north borders of our country, and their habitat is subalpine and alpine meadows. And the Milos viper with red color only on Milos, Kymolos, Polygos, and Sifnos. Islands colored in white are viper-free islands. So now let's see the 2 Opistoglyphus snakes in Greece. Just for the record, the eastern Montpellier snake males have a uniform greenish or grayish coloration, while juveniles and females have a spotted coloration. And the second Opistoglyphus snakes in Greece is the European cat snake, which we have already seen in previous slides. And these are the 2 venomous snakes of Greece that are Opistoglyphus, and they have too weak venom to do us any serious damage. All venomous snakes' venom is actually a cocktail of different toxins. Vipers in Greece have mostly cytotoxic venom and far more or less neurotoxic, which means that their main effect is to break the lipid membrane of the cells, causing severe damage to the tissue. On the other hand, neurotoxic venom targets the nervous system by stopping the nerve signals, which control the muscles actually, and lead to vital organs failure, such as the heart and the lungs, very fast after the snake bite. Fortunately, in Greece, we don't have snakes with mainly neurotoxic venom. And compared to the toxicity of the venom of vipers from other continents, Greek vipers have relatively weak venom. So that means should one get bitten by in Greece by a viper, they have a lot of time to safely visit a hospital for the first aid. So let us now see some statistics on some samples of causes of death in Greece during the period from 2000 to 2014, 15 years in total. Some statistics I obtained from the Hellenic Statistical Authority back in 2017. So during this period, we had 6 people in total that died from a snake bite. It's the same amount of deaths we had by dog attacks. 15 people in total died from attacks from farm animals, such as cows, horses, and pigs. 33 people died in total by mosquito bites through deadly infections. 44 people died in total by thunderstrike, which gives an average about 3 people per year. 73 people died in total during this period by bees, wasps, and hornets, probably by severe allergic reactions. And just to compare, more than 2,000 of people died by homicide, and more than 24,000 of people died by car accidents, which give about 1,600 people per year. To make those numbers more understandable, let's see some more numbers. Up to 200 people in Greece are bitten by venomous snakes every year in Greece. The deadly incidents are about 1 every 500, which means a mortality about 0.2%. And in all cases, these numbers concern elderly people and people with serious health problems, and the very rare case of severe allergic reaction and anaphylactic shock. So as we can see, vipers in Greece are indeed dangerous, but not as much as people usually believe. And that is because, on the one hand, vipers are not aggressive, and they will bite only if caught or stepped on. And on the other hand, Greek viper's venom is not capable of killing a human just within hours. If you wish to know the first aid after a snake bite in Greece, please visit herpetofauna.gr, where you can read them in Greek, only in Greek at the moment. That's it for me. Thank you very much. We can proceed now to the Q&A procedure if any questions.
So there is a question. Thank you very much for the presentation. There's a question from Manolis. He's asking, the Milos viper looks very much like the Lebetina viper from Cyprus. How can we tell the difference between them?
Well, some researchers right now treat the Milos viper as a subspecies of the Cyprus viper, the Macrovipera lebetinus. I think it's too soon to have any conclusions on this, because they only surveyed a few genetic loci, comparing those two populations. So it's too soon to tell if this is actually a subspecies of Macrovipera lebetinus. So I recommend we treat it as a full species, a separate species now, the endemic Milos viper. We can distinguish between the two of them. First of all, because they have a different distribution, okay? And there's a rule that if you count the number of the row, the number of the scale rows in the middle of their body, Milos viper has 23 rows, and Cyprus viper has 25 rows of scales in the middle of the body.
Understood.
Cyprus, the Cypriot viper gets a lot bigger than Milos viper.
Mm-hmm. So his response was, as I understand, there has not been enough research done on the Greek viper species. Is that the case in your opinion?
That's true. Yes, that's true. More need to be done.
Understood. Well, certainly a lot of people do it, right?
Yeah. Yeah.
So if no one else has any other questions and you have wrapped up.
Yes, yes, of course. Thank you very much for the invitation.
Fantastic. Thank you very much, Ilias. Again, one of our own. Very happy to have you with us. And we can now proceed with Juan José Calvete. We are very glad to have you here. Sorry to have kept you waiting. The floor is yours. Please, by all means, begin the presentation.
Okay, I wish just to start thanking you and your collaborators for organizing this meeting. So you don't need to worry for having me here waiting. I prefer to learn from you than to tell my things. So this is the most important part of any meeting, to learn from all the participants. Thank you. Let me see if I can. So this would be my slide there. And so this is the title of my presentation. You see it? Yeah.
Yes,
okay.
Absolutely.
Okay.
Perfect.
So let me start just with a small introduction that probably is not needed here, but just in any case, venom is an ecological trait that evolved some more than 100 million ago in a clade of reptiles, the Xenophidia, and the venom as venomous snakes are mainly within the vipers and the elapids families, although there are also some colubrids that contain snakes, but I will not be talking about this family. From what we know, or we think we know, is that the venom system evolved from gene duplications from ordinary genes that had their expression mainly concentrated in a tissue, the venom gland, and through the creation of multi-gene protein families by duplications and by natural selection, which means accelerated evolution, neofunctionalization, and pharmacological innovation, and through arms race, as we have heard in previous slides, the venom evolved to the state we find it today. So venom, from a compositional point of view, are not very complicated systems in terms of protein families they contain. They contain just a handful of protein families. The complexity of the venoms is in the molecular diversification that they have within and between the proteins that conform the venom. You see here just the variety of different PLA2 molecules that have been found in one or another venom, and also this like this tree of diversification of the three-finger toxin, but not all of them, of course, are within the same venom. They are in the whole family across the different species. So this innovation, this molecular diversification, is what is the challenge to understand venoms in molecular terms. Of course, there is also genomic and postgenomic genomic and postgenomic processing of the products, and they have been they involve gain and loss of different gene families. It has been also suggested that some microRNAs can also have a role in the transduction of the RNAs to protein, and of course is in more and more occasions we see that the crossing between similar but not identical species can have also a role in producing diversifications in venom across different populations. This slide, which looks complicated but is very simple, is just to tell you and to illustrate that venom, as any other field in science, has evolved in parallel with the evolution of technology. And the technologies that has been applied to different biological systems, sooner or later, it has also become available in the venomics field. So here is more or less in the late 80s, mass spectrometry was available to biological research, and about the turn of the 21st century, it was also applied to venom research, and so is true for any other approach, biological approach that was available at the time. And here you see a bit later the transcriptomic transcriptomic field started, and now we are immersed in the snake genome sequencing era, where every year several genomes and at an increasing pace are being released to the field. So this has given rise to this type of research, that from a very narrow hypothesis-driven research that is concentrating on a problem and putting all your effort to understand it. We have now many labs just dedicated to omics-driven research, which is fish whatever there is and then try to get any meaning out of it. This is the challenge of the omic technologies, of course. So I prefer, in my lab, to work with the tools of the omic-driven researchers, but with hypothesis-driven approach. This is how I understand venomics in the lab. So you see here the cumulative number of studies that have been published between the first stages of the field and the last year, 2020, and you see that this is steadily increasing number of research. As a consequence of this, we have a lot of venoms characterized, but still not so many in percentual terms. Only 22% of all the elapids have been in one way or another studied, and 78% is still without any entry in PubMed. Here in red are those that have been studied at protein level, underlined with top tower analysis, and the triangles are those that have transcriptomics, and the asterisk those that have the genome also sequenced. And the same is true for the viperids. Here the number of species is a little bit higher. The percentage of studies that has been released, 41%, and is increasing at a good pace probably in the next couple of years. If we look the whole all this analysis, of course it depends. You analyze venom depending on what you want to get from the venom, and we can divide the whole venomics field into ecological venomics and clinical venomics, clinical toxinology. The ones here the ecological venomic is driven by but I can see this here. Yes. So let me take this down somewhere so that I can put it here. So here is where natural selection plays a role, and where the outcome of the diversification of the venom is towards subjugation of prey. Need not to be killing the prey, but at least subjugation. We have also heard from Bryan Fry in the first talk. So everything how we test venoms and how we understand the functionality of venoms should be under the frame of natural selection and evolutionary terms. Whereas here in the clinical toxinology, everything is anthropocentric biased, because here the goal is not only to understanding, you don't need to understand the natural history of the animal producing the venom to understand the clinical implication of the venom. Here you need to understand the structure-function correlation, particularly to humans, and from ethical issues, at least from a mammal, which in our case is mainly the mice. It's not exactly the same physiology, but it's similar, at least has the same type of metabolism. So this should be the guide for anyone entering the field of what is the purpose of your investigation, to be on one side or the other side, or in both, but with different thinking in every case. So here is an example. And another concept that is also gaining power is the adding resolution in dimensionality to comparative venomics by analyzing whole clades of venom, not single venoms. Because if you analyze and compare the composition of the venoms in the frame of the diet, in the frame of the habitat of the snakes, then you can rationalize much better the nodes of diversification and try the hints why these changes occur in this particular clade. The same is here. Is here true. These are the Micrurus venom. Here is a PLA2, three-finger toxin, other proteins, and you see that this is a pattern, a dichotomic pattern towards venom, three-finger toxin-rich venoms or PLA2-rich venoms across the geographical radiation of the genus. So there is a tendency to get increased concentration of three-finger toxin as you go from north to south in the radiation axis of the species. There are, of course, some exceptions, which are always the most interesting thing that you can have, like these three species that are PLA2-rich venom species in a three-finger toxin domain dominated region. So this means that here some ecological situation has forced the venoms to maintain the PLA2 instead of changing towards the more three-finger toxin venom-rich secretions. So, and this has a certain explanation in the way the Amazon River and the Amazon basin was modulated during the last 20 million years ago. So you see here, there is almost a clear cut-off between the PLA2 rich venoms and the three-finger toxin, with a barrier being the Amazon River, but only after it came to its actual geographic bed. So this is also apporting hints to understand the venoms in the ecological terms. But venoms also cause a lot of accidents, and here is a map with some statistics. And so the venom, the challenge in the clinical ecology, in the clinical toxinology field, is to understand what is causing this suffering and how to fight it in rational terms. Also here, the distribution of the toxins and the clade toxinology gives some hints. This is the clade of African Naya, and you see that the three major components have also a logical path from the starting of the genus to the different particular branches in the phylogenetic tree, and how here the PLA2 was lost, and here the neurotoxin took more place, and the PLA2 were dominating this part of the tree. So this is important to know, and this provides you more hints than simple venomics. To get the picture, you need resolution, and to need resolution, you need to analyze, and you need to have good technology to analyze the venoms. Important also is that different labs work with the same technology to be able then to compare the different outcomes from different labs. And this is, I would say, everyone is working with different technologies, and sometimes it's difficult to compare data. So I will just present what we do and why we do. So we have, we believe that the key to quantify a venom is to separate is to leverage on the complexation of the venom to quantify the different components, because reverse phase chromatography is a quantitative technique, can be a quantitative technique, but you can quantify the amount of protein that is under each peak. But mass spectrometry, the technique that is used for identifying the components, is inherently non-quantitative technique. So trying to quantify through mass spectrometry instead of separating the quantification from the identification process can bring you to difficulties in getting a reliable quantification. So I would say it's flawed. And it is flawed simply because all the technologies that are being used, the so-called label-free, where you just take the whole venom, digest it with an enzyme, put it in the mass spectrometer, identify and quantify in the same experiment, either by the intensity of the ions that match to that protein, or by the number of, simply by the number of spectra that match to a given protein. But all these approaches, they are linked to this to a ratio between the observed and the observable ions. So you rely on what should be seen to quantify what you are currently seeing. And this means that if you have issues with the database is not complete, you are under or over or over quantifying different components because simply they are not present in the database that you are using for identifying the proteins that you put in one part or the other of the ratio. This is an example. This is the quantification using the method that we use by separating the quantification from the identification, and this is shotgun analysis with ion intensity. You see that this component this protein is biased towards the CTL component simply because it gives more matches in an incomplete database. And the same happened here with the normalized spectral abundance factor, where because of this term, this ratio over here is also biased against the same component. So this means that you are reporting something that is biased because of the non-appropriate technology that you are using. So we have recently in this paper that is just published in the last issue of Journal of Protein Research, make a proof of concept by quantifying the components of the venom with elemental mass spectrometry, which is inherently quantitative technology that can give you absolute quantitation, but only of atoms, only of isotopes of atoms. In this case, we use sulfur because all the proteins contain sulfur. And if you know the sulfur content of a protein, you can translate the absolute sulfur quantification into absolute protein quantification by knowing the number of atoms that you have per molecule of protein. So this was successful, the proof of concept, and we can say that as we have expected, our method is giving a relative quantification in percentage of grams per gram, percentage of weight of the venom component. So here you can translate this easily to absolute protein quantification in moles or whatever you like. So this looks here very complicated because here we use 4 different reverse phase runs and correlating one thing with the other between the elemental and the molecular mass spectrometry systems to correlate which peaks corresponds to which peaks in which chromatogram. But this can be simplified much in the future, and we are now working in a more simplified way where you have the same front end fractionation like in our original method, but then you can send the same ions that are being identified to absolute quantification by the other method, by the elemental mass spectrometry method. So in this way, if this technical issue is solved to hyphenate the high resolution molecular Fourier transform mass spectrometry to the elemental ICP-MS system, then you can correlate here just by the mass of the proteins that you identify here with the peaks of the sulfur and absolute quantitate everything. But there is another way that we are also trying to develop a little bit more. That is, well, why not use the extinction coefficient of a protein to quantify the whole chromatogram? After all, here the extinction coefficient is the absolute concentration in terms of milligrams per ml. If you know the extinction coefficient of the protein at 280, this is what I did in the 80s during my PhD to correlate to quantify proteins in this way. So this means that there are formulas like this here, where you get an estimation, and they're very reliable, very nice estimation from the absorbance of a protein in terms of the three major components that contribute to the absorbance of this protein, which is the number of tryptophans, the number of tyrosines, and the number of cysteine residues in the molecule. And with this formula, which is just very simple, this 5,500 times the number of tryptophan plus 1490 plus the multiplied by the number of tyrosine, and 125 times the number of cysteine residues divided by the mass of the protein, you get the absorbance from the absorbance, the milligrams per ml that you have at any point of the trace. So this is feasible. This can be done. We are work on this method, and I will just show you here as a proof of concept. This example, this is a chromatogram. You have here by top-down analysis the sequences of the individual proteins that make these peaks. We just concentrated here in the major ones, and here down you see the experimental coefficient done by weighting very precisely quantities of the proteins under each of these peaks, dissolving in a given amount of water, determining the absorbance, from there the milligrams per ml, and from this the experimental coefficient for each of these components. And this is the calculated ones according to this formula. And you see here down that from the experimental to the calculated is very accurate. So it's not exactly the same, because we didn't take into account all some here peaks. But at the end, if you have complete analysis, spectrophotometric analysis of the trace, you will be able to quantify. So this would be then much easier just to apply a venom into an HPLC, quantify directly from the trace, and identify the proteins that are coming on the other side with mass spectrometry. This could be automated and would be simplify and give a high throughput capability to the system, and then you can concentrate on doing many proteomes in less time, and well identified and quantified for all of them. So this is the way to change from individual to clade venomics in a rational way. So let me see. Another thing that normally one doesn't know, at least I had no idea of this a couple of years ago, is the thing that and this now relates to how to translate data from the lab to the let's say to the clinics or preclinical situation, data that you gain on the analysis of the venom, how to apply them either to ecological venomics or to clinical toxinology. You need to in some way normalize the data for the system that you are studying. And this is the allometric scaling. This means, and these are data, and this is under discussion since almost 100 years, from the last century. The people found a linear relationship between the metabolic rate of an animal or a system and the relation between the body mass and the surface of the individual. You see here, and this is true for any unicellular organism, to ectoderms and endoderms, independently of the size and metabolism of the individual. This means that if you have data that you obtain in a mouse, how to translate this to humans? Well, there are tables where from the ratio between the weight and the surface in square meters, you get a km factor, which is specific for each species. So if you have data from mouse to translate to the human situation, you just need a very simple formula, which is the dose for human, the human effective dose of a drug, and this is used in the pharmaceutical industry for many drugs. This dose, this effective dose, is equal to the animal dose multiplied by the animal km divided by the human one. So in this case, if this is an LD50 that you have done in mouse, the LD50 for human would be the LD50 for mice times 3 divided by 37. That's all. And with this, you can then think better in clinical terms. So let's see this example. This is the LD50 derived from mouse for Echinocelatus, and is 1.4 micrograms of venom per gram of mouse body weight. This is the way that usually people publish the data. This means that for a body weight of 20 grams, 28 micrograms of venoms would kill 50% of the population. This is the LD50. This means that if this snake bites you and you apply this same data, the LD50 of 1.4 micrograms per venom per gram of mouse should be multiplied by 60,000, because it's the ratio between the mass of a human to the mass of the mouse. And this means that you would need about 84 milligrams of venom per human of 60 kilo to kill half of the injected humans. So the LD50 would be 84 milligrams of venom per individual. And this is simply impossible, simply because this snake causes accidents, but the average yield is much less than this. However, if you convert the data, the LD50 into a human LD50, then this LD50 would be 1.4 times 3 divided by 37 is 0.11 milligrams of venom per kilogram of human, which translates into 6.8 milligrams of venom per human of 60 kilogram. And this is in concordance with the clinical data. So allometric translation of the data is very important if you want to move from the ecological to the clinical domain in a venomic analysis. Let me see. So someone asked if the venom yield of the snakes is known, and actually this is a very important issue in venomics. So it's not the same if you want to treat a victim of a snake is bitten by, is injected with 2 milligrams or with 200. So it doesn't matter what is the yield of the snake if you force the muscles here in the jaws to get the maximal capacity of biting in a forced situation. But as we have heard also from Wolfgang talk, snakes usually do not bite for hunting you. They will bite as a defensive. All accidents are defensive bites. So and in a defensive bite, nobody is forcing the snake to bite more or less venom. Actually, if the snake could think, it would inject the less amount of venom that convinced you not to bother it more. So this means that if you don't force the snake and let it just bite in here in the jar, you will have a more accurate view of the amount of venom that it can give in a defensive bite. And this is what you see here, the venom coming down. So you see, it just wanted to bite once and has no desire to give a second bite. And here we have the amount of venom that could have injected in a victim if this were the case. So we have done this, but this is mainly work from David Williams in collaboration with Snake Farm. And here are a table with several data for several species, the average yield that was obtained for this species, and this very different. It is from 31 in X romani, the previous X ocellatus, but from Cameroon, or only from here 11 milligrams to more than 900 milligrams in some species. So this should be taken into consideration when you when the clinician is thinking in how many vials of antivenom will need to treat a bite. It's not the same to have been bitten by one snake and the other, but actually most of the antivenom productions, they formulate their antivenoms for a same number of LD50 for mice, not even for human, for mice, which means that they are giving a therapeutic dose that has nothing to do with the real situation. It's something that the clinician will find out when is treating the victim, but has not been foreseen in the formulation of the product. This is something that should change also drastically. Well, another, how is my time?
I think 8 more minutes if we can conclude this.
8 more
questions.
Then I will hurry up a little bit, or just give you a couple of slides more and then allow for the question. So important also is to have to know which toxins in a venom are those that should be neutralized in a bite, because not all the proteins in the venom are life-threatening toxins. Many of them are injected in such small amounts that for the LD50 for human doesn't make any trouble to the body, and others are the ones that are leading the mortality of the venom. And here we have an example. You see, this is the LD50 for mouse. This is the LD50 calculated for human. This is the relative abundance of the venom. So in this toxicovenomic analysis, we have these three parameters, and we can calculate also from the average venom yield of this particular snake, which was 314 milligrams, the number of LD50 for human that can be injected in the bite, which is about 35 LD50s for the victim. So we found also a population of the same species that lack this neurotoxin. Is only made by these cytotoxins. So this, if you combine this data, this means, and this contributes with 18.6 human LD50 instead of the 30-something for the neurotoxic variant of the venom. But if you combine this data, you can say, well, the difference between the potential of one venom versus the non-neurotoxic venom is 16.4 human LD50s, which should be accounted for the neurotoxin that was not present in the second venom. So this 9% toxin accounts for 46.8% of the mortality of the venom. And all this huge amount of cytotoxins, they contribute together with 53.2. So now you see that the difference between thinking in abundance or thinking in normalized parameters is much different, and better understand the outcome of a neurotoxic versus a cytotoxin from a variant from the same species. I will just this is the last major technology that we are using also in the lab, which we call antivenomics. And here is just to resolve the capability of binding of toxins of an immobilized antivenom in resolving in molecular terms. So here we can say how much of this component is being recognized by the venom, by the antivenom, etc. And from this data, we can calculate the maximal capacity of an antivenom in terms of how many milligrams of total venom protein it can be bind per gram of the active product. Since we know also the amount of antivenom in the vial, we can, and the maximal binding capacity of the antivenom from this data, which is 14 milligrams for this total amount of venom proteins of Fab proteins per vial. This means that we can calculate it here the percentage of active compound in a vial. In this case was 24.7%. And if we use and we combine with the in vivo data, this is the potency, the neutralization potency of the antivenom in the mouse system, then we can calculate how this amount of toxin binding antivenoms are divided between those that do not neutralize anything and those that are clinically useful. And in this case, if it is the potency of the antivenom is neutralizing 6.5 milligrams of venom per vial, and here you can bind 14 milligrams per vial. Just divide 6.5 into 14, and you have 46% of all the toxin binding immunoglobulins are neutralizing immunoglobulins. And this translates into 11.5% of all the antibodies in the vial are clinically useful. This is also why many people in the literature say that this is the antivenom should be changed. They are very low effective. This is an old-fashioned technology to produce antivenom, and we will hear for sure in the next talk much more about this. But the actual thing is that you are comparing something that is designed for a toxin with a mixture that contains only 11.5%. However, if you purify these clinically useful antibodies from the old-fashioned antivenom, you increase substantially the neutralizing capacity, and we have been able to show in not published yet research that you can get from an antibody that neutralize only about 16 to 20% of venom per gram of total antibody by purifying the antibodies that are recognizing the particular toxin that carries most of the mortality of the venom, to increase the neutralization potential to more than 800 milligrams of total venom per gram of the purified antivenom. So the difference is huge. And on the other side, I haven't seen yet any synthetic or recombinant or whatever antibody that neutralize more than 16 to 20 milligrams per gram yet, probably, and I will be happy that this evolve in a more positive clinical way. But for the moment, I would say that criticizing what we have is easy, but actually are the only ones that are saving lives, and they can be improved for sure. But the solution is not just to throw everyone to everything that we have to the garbage and start from scratch with another approach. So this is something that should be spoken high in a conference, in a meeting like this. Oh, I think well, there are some other examples of things, but I think that enough concepts, enough thoughts for today. In another meeting, in another context, we can concentrate on more specific points of interest for the audience. So I thank you for the kind invitation to be here today, and am open for any question that the participants may deem.
Absolutely. Thank you very much, Juan Jose. I think there's a question from Vasiliki, if I'm not mistaken.
Yes, I have a question. First of all, thank you for this excellent presentation. You do a great job in this field. I have a question. In HPLC that you separate the proteins, what mixture of solvents do you use? This is the first question. And for how much time do you run the sample?
Well, the samples we have, let's say, a standard protocol, which is the one that we use to see how the complexity of the venom is. And depending on this and the separation, then we modify the gradient. The gradient is 0.1% TFA in water or in acetonitrile. And we go usually from 15 to 70% with a flat section at about 25 to 45, which is where more of the proteins elute. But this need to be optimized for any venom. So the total length of the gradient, depending if it is a semi-preparative column, 25% times 0.4 centimeters, we use about a 90% gradient to isolate the peaks for electrophoresis and the downstream mass spectrometry. But if it is just to compare venoms, for instance, once you have the venom well characterized and you want to compare 50 venom from 50 individuals or from 50 population to see how well are they related, then we use a small 15 centimeters column, 0.2 in inner diameter, and then you can go down to 40-45 minutes gradient with the same kind of solvent. So if you have any specific question, you can email me and we can clear it in more details and for your necessity.
Thank you very much, because I'm very interested in this field. And I have one more question, if this is possible. Do you think that QTOF MS is suitable for identification?
Sure. Sure.
Okay.
Sure.
Thank you very much.
You're very welcome.
Fantastic. So at this point, I would like to thank you again, Juan José. Thank you very much for being with us and honoring us with your presence and presentation. And if everything is wrapped up, we can move on to our next guest speaker, Timothy Jenkins from Danish Technical University, Tropical Pharmacology Lab. Quite exciting stuff here, human monoclonal antibodies, if I'm not mistaken. Tim, you have the floor.
Hello, Thomas. Thank you very much for having me. Let me just share my screen. See if we can get this going here. Can you see everything okay?
Absolutely. Good to go.
Brilliant. Well, it's quite an honor to be here. Quite a prestigious crowd, a really nice mix of people that you got together for today. Really nice expertise, and I feel quite honored to be part of that. I'll be presenting on behalf of a tropical pharmacology lab and talking a bit about our approach that Calvete already alluded to a bit in terms of looking at next generation snakebite therapeutics. First of all, I will answer one of the most commonly asked questions, which is, why are we doing snakebite research in Denmark? That's a very good question. It's a beautiful place. It's rather cold and dark in this time of year, but as you can see here in the background, we've got one of the most famous sites in Copenhagen. It can be rather beautiful when the sun is out. And in the bottom left, we've got a group of a crowd of international people from pretty much anywhere around the globe. And we work on a whole array of different things. We've got antibody discovery, which is our bread and butter. We do a lot of protein biophysics, venoms and toxins, a lot less. I mean, especially following Calvete's talk here, a lot less, but it's something that we're also taking into account. I'm personally responsible for the bioinformatics arm of our discovery processes. And we're also very interested in science dissemination, entrepreneurship, and obviously very interested in getting the new generation of students keen and excited about venom and anti-venom research, which is also why I'm here today. Now, why Denmark? Well, when you think about Denmark, you usually think about Vikings and some pretty nice pastry, some halfway average beer, but you don't really think about snakes. And the reason why we are doing snakebite and antivenom research in Denmark is really the tech. We're trying to apply the latest technology that has been developed in companies around the globe in different fields like cancer, and try to apply it to this neglected field that is snakebite, and use these next generation approaches to see if we can drive our treatment of patients to the next level. Now, as has been already introduced quite thoroughly, snakebite is a major issue. A lot of people die. Quite importantly, also a lot of people that don't die are still permanently affected, either physically by having lost a limb, or at least financially, because antivenoms, when available, are rather expensive and can mean quite a heavy hit on your economy. Also, just the loss of income when you can't work on the fields anymore is quite substantial. So it is a very important disease and something that we really should be looking at, and luckily is now also receiving more and more attention. Now, I think it's always important. I don't like showing pictures that are gruesome, but it's important to keep in mind that we're not doing cool science here. We're doing something to solve a real world problem. And that's true for everybody working within this field. We care about the humans that are being affected. And here we've got a small girl in Nigeria that was bitten by a spitting cobra. This is not even a very severe envenoming. It's a medium envenoming. And luckily she survived this bite, but she will be permanently scarred by that experience. And this is just to keep in mind that we really need to do something. Now, there is traditional antivenom, and I want to highlight here that our approach of next generation therapeutics is in no means trying to take away anything from the current products that are being used, improved, and are saving lives, because as Juan Calvete noted, they are the things that are actually saving people's lives right now. And if we didn't have them, the world would be way worse off, and the numbers would look significantly worse. What we are trying to do is, while people are doing this work and improving these antivenoms based on animal blood products, we're trying to see if we can give us another edge in the treatment while we're using these antivenoms, to see if we can drive it further. And our idea is to identify key medically relevant toxins within the snake venoms and make specific monoclonal and recombinantly expressible antibodies against these toxins, which will then open up a lot of opportunities in terms of manufacturing. We won't need to use animals to produce these antivenoms anymore. We can make these antibodies depending on what format we use, either in big fermentation tanks or in larger mammalian cell cultures, which makes the whole thing a lot more scalable and can also be more economical in the long run. Now, the main technology that we're using at the Tropical Pharmacology Lab is based on a Nobel Prize winning tech developed by, John McCafferty and Greg Winter, and involves the use of phages, bacteriophages. And the idea here is that these bacteriophages have your antibody library encoded within them. So whatever you have, it can be antibody sequences from human donors, it can come from llamas, it can come from horses, anything, it can be synthetic, is encoded in these phages. And you've got millions and millions of these phages with unique genes, so they can then express the protein, which in this case is the antibody, on their surface. So you've got this link between genetic information and the expressed protein that you can use to actually discover your antibodies in vitro. So what happens is that we take this library of antibodies, we have our phages, they're expressing the antibodies, and we have our targets. So let's say we use one of the neurotoxins from a cobra. We immobilize that, and then we throw our whole library against this toxin and just see what sticks. So this is a binding step that you can see here. And after that, we basically wash everything that doesn't stick away. We cut off our phages so they're free to use again, and use these phages that we know are binding to the target to infect bacteria, grow them up, and do this iteratively to get better and better binding phages to our target. And in the end, we can then characterize these antibodies that we've got out of these phage display campaigns. And this is basically the main technology that we're using to discover the antibodies in our group. Now, one of the main breakthroughs we had for this technology was actually a paper published a few years back, where we were able to show that some of these antibodies that we discovered in this fashion against black mamba toxins were able to neutralize the venom of that snake in vivo, of the specific toxins in this case, in vivo. So we knew that this tech obviously still has a long way to go, but it worked. And there's a significant promise to using this. And it also was one of the things that opened up the gateway for us actually receiving some significant funding. And this is just to highlight that people are starting to care about this problem. People with the means to actually solve snakebite are caring. And that's not big pharma. This is the EU, so the ERC in this case. There's a Danish fund in there, Venom fund, and also quite importantly, the Wellcome, which is a UK-based trust, which is putting a lot of money towards solving this problem. And this is also to encourage all of you students now. This is not obviously in the same funding levels as cancers, but there is money to do very interesting science and actually try and figure out how we can solve this problem, which is and should be solvable. Now, one thing that also came out very nicely in the last talk was that one of the key issues we're facing with making any kind of treatment against snakebite is that these snake venoms are incredibly complex. So what I have here is just an overview over all snake venom toxins that are currently registered in UniProt and clustered based on how sequence similar they are. So each of these circles or nodes represents a single toxin. And we can overlay some annotation here where we can figure out what are the different types of toxins, what are the different families, and so on. And this just gives you an idea on how big the diversity of targets is that we need to be hitting to make good antivenoms. And to give you a specific example, one of the main projects we have in our group now is actually trying to develop a next generation antivenom based on these recombinant monoclonal antibodies for sub-Saharan Africa. Now, there are around 24 different venomous snakes in sub-Saharan Africa. And if we want to make a good product, we need to be able to neutralize all of the medically important toxins in all of their venoms. So to give you a rough overview, this would mean around 100 or so different toxins that we need to be able to neutralize. And as you can see here, they fall in all of the different categories of snake toxin families, which makes this an incredibly complex task. this is feasible. We could discover 100 different antibodies, no problem. But if we're thinking about the end solution, it's not really a viable product. Whatever we'll be making with 100 different antibodies will be so incredibly expensive that nobody could afford it, not in Denmark, and especially not in sub-Saharan Africa. So we need to be smart about what we're doing here. One of the key things here, again, is that snakebite is a disease of poverty, and a lot of the technical decisions that we are taking are guided by this idea of making our product as simple, as effective, and obviously as cheap as possible. Now, there are quite a few variables that play into the factor of how we can make this happen. How can we make an affordable next generation antivenom? I've got one of the tables here from a paper that published together with Andreas Lausen last year, where we looked a bit into the cost dynamics of what factors really affect the final cost of such next generation antivenoms. And one of the important ones also highlighted in red here is how effective our antibodies are. How many antibodies do we need per toxin in a certain envenoming case? So making very, very efficient antibodies is quite important. And one of our PhD students, Lina, decided that would be her focus of her PhD and dedicated the last 2.5 years of her life to this. And so what she did is actually used some antibodies against alpha cobra toxin that Andreas had discovered already. They were binding quite well. They were showing really good signals in vitro, but when we put them in mice, they weren't neutralizing very well. So the idea was that we could use something known as light chain shuffling, where you take the constant region, so the CDR3 heavy region of that antibody that is primarily related to the binding interaction, and then just try and shift stuff around and see if by chance you can actually improve your antibody. And so what she did is that she took all of these antibodies and she did a lot of chain shuffling. She did phage display again on this new library of chain shuffled antibodies, and was able by doing that to make new antibodies based on these parents that were actually able to neutralize envenoming in vivo, which was a really nice sign, again, for us to see, okay, if we've got decent antibodies, we still have methods to improve them to being good enough to actually, in mice, neutralize toxicity. And this just shows one of the roots of research that we're doing is how can we make high affinity, very effective antibodies. However, even if we make super effective antibodies, this doesn't solve the problem of having 100 or so different targets. So another very important thing that we are exploring is how can we actually make our antibodies cross-reactive? Highlighted in red here shows how the different levels of cross-reactivity of antibodies affects cost dynamics. I should explain here quickly, the orange bar is the cost in US dollars, so $620 of Favipiravir, which was for a very long time the best available antivenom in sub-Saharan Africa. No longer is available, Sanofi stopped producing it. And the numbers next to it are the different costs for making an equivalent product using recombinant antibodies. So the different colors of the bars represent the levels of cross-reactivity. And as you can see, they also play a big role in terms of driving the cost of the final product down. So how do we approach this issue of making cross-reactive antibodies? Well, importantly, if you look at a single snake, the diversity of toxins is quite daunting. But adding more and more species, especially when we belong to the same family, so if we're talking about elapids, having a lot of different elapids or having a lot of different vipers, the diversity between these different species is not as great as within a single one. So actually the complexity doesn't increase so much because they have a lot of similar but different toxins. So the idea here is, can we make some antibodies that can actually bind and neutralize a cluster of similar but different toxins? So in this case, antibodies that could potentially neutralize these different subgroups of toxins. And that is a clear way of decreasing the complexity of our final product and making a viable solution to the problem. So this is all nice and good. The theory stands, but how can we actually make our antibodies cross-reactive? And we are using two main techniques to try and achieve this. One is the idea of cross-panning. And as I mentioned earlier, we're using phage display where we have our antigen, our toxin that is immobilized. And what we can do, instead of using the same toxin every round we do this, which is usually 3 rounds, We can also switch the toxins and select for phages that bind toxin A, but then also toxin B, and maybe toxin C, or we go back to toxin A. There's a lot of different combinations. So another PhD student in our group is trying to figure out what the smartest way is to combine our antigens to get the broadest cross-reactivity whilst retaining specificity. So making good antibodies that can cover a lot of similar toxins. And this is one of the main strategies, which we've also shown to work, published last year. And another technique that we're using that was already demonstrated to work really nicely when you immunize horses with them to make more cross-reactive blood product-based antibodies is the idea of consensus toxins. So again, you can take this cluster that I showed just now of the different toxins that are quite similar, and you can make something like an average toxin out of these different toxins, also known as a consensus toxin. Obviously, there are some certain rules that you should take into account when making your average. And if you make a too large list of different toxins, what you get out is totally irrelevant. So you need to be smart about this as well. Then you can express this toxin recombinantly. You obviously need to test that it's functionally active in the same way as the parent toxins are. You can then use that during phage display. And the hypothesis is that if you get binders against that consensus toxin, you will also get binders to at least most, maybe all of the toxin used to make this consensus toxin. And this technique can also be combined with cross-panning. And this is another area of research that we are pursuing here at the Tropical Pharmacology Lab. But that kind of brings us to the next question of how can we actually identify which different toxins might be neutralizable by a single broadly neutralizing antibody? So how do we identify these clusters? How do we decide on the list of toxins we include? And that's something that I did during Corona, basically. We were forced to work a lot from home, so I put a lot of efforts into some more computational projects. And I thought a bit about this issue. And traditionally, the way you would do it is you would do sequence alignment. So you look at all of your different toxins, look at how similar the sequences are of them. And then based on the score you get, you can say, okay, everything that's 90% similar should be close enough. Everything is 80%. And that's all nice and good and has value. But we are working in a 3D space. When we're talking about antibody toxin interactions, we are looking at 3D interactions. So it would be a lot more elegant to look at this, at least also in a structural space. However, we were faced with an issue here, and that was that the majority of toxins from snake venoms have not been crystallized. We don't have a lot of structures for snake venom toxins. And what we did then is use some of the latest technology available to model all snake venom toxin proteins together with a Greek PhD student here at DTU. So this might be an inspiration for you guys. He did his master's at DTU, got a master thesis project with our group. And then we helped him get a PhD with a neighboring group, and he still collaborates with us. So really great success story, both for him, but also for us, because we've done some really cool work together, and we managed to model all of these snake venom toxins. And then just as we were done, I don't know if you heard about this, but Google DeepMind brought out the next generation of protein structure prediction. So we decided that obviously not everything we'd done was useless, but we wanted to compare it to this next generation solution. So we also modeled all of these snake venom toxins with AI using this new transformer-based machine learning approach that Google DeepMind developed. And this is known as AlphaFold 2. We then also developed a tool called VTox that actually helped us make this whole thing useful and actually look at what's going on in our data set. So how can we visualize the data that we have now? How can we compare the similarities? And basically what we managed to do is make these huge dendrograms. All of these toxins here are clustered in proximity based on how similar they are. So we can identify all of the toxins very quickly and see how closely they are related. This sounds maybe a bit abstract, so let me give you some real life examples of how we might use this. Here's just an example of what the tool looks like. Let's say we're interested in myotoxin 2, phospholipase A2 from a viper, Bothrops asper, that you find in Central America. And if we looked at this here, what we have is a list of different snake genera. Then we have also the type of venom toxin. So in this case, these are Viper PLA2s. We then also have the similarity scores. So obviously it's 100% similar to itself, but also to all of the others, annotated with UniProt IDs, and basically get a quick overview of what else might be interesting to look at if we have an antibody against mytoxin 2, for instance. And that's exactly one of the things we're doing with this now is we've got an antibody we know binds toxin A and B. What other toxins might it also bind? So for our already discovered antibodies. Another thing we can also do is use it to understand what we should cross-pen against. So if we want to get a certain set of antigens, we say this is a cluster of different toxins. These are the toxins that we think based on their similarity make most sense. They are different enough to be different and interesting, but similar enough for an antibody to be able to bind. And then select our cross-penning targets based on that. Then we can also use that to make our consensus toxins. So we get a list of these toxins and we make our consensus toxins. Great. And then we combine with 2 and have both the consensus toxins and cross-pen. And this is just to give you an idea on how we actually think about making cross-reactive antibodies, what different considerations come into that, and the different skill sets. We're talking about hardcore lab skills where you're working on phage display discovery, cloning, molecular biology, to being able to do Python coding and massive data analysis. So it's a broad spectrum of different skills that we are needing in our research avenues. Now, a couple more points I would like to highlight is that we are primarily interested in human antibodies, IgGs, but there's also a lot of other antibody formats that might be worth looking at. They have a lot of advantages over IgGs, obviously also some disadvantages to them, but they could work quite well in unison as a final product. So some of the ones we're most interested in are nanobodies that come from llamas, and DARPins, which are a synthetic antibody scaffold. And they have a lot of advantages in terms of stability, being small, very, very easy and cheap to express, and having quite a nice overall versatility and engineerability. And these different types of antibodies can be used for different applications. So some things might be better for systemically acting toxins, some might be better for locally acting toxins. And that's all I want to show here. And our final vision for end product would be actually having a mix, a cocktail. Some things will be best neutralized by IgGs, some maybe by nanobodies, but also some of the small molecule inhibitors like the Resplidip are very, very promising. And in reality, what will happen is probably that we'll have a mix of these different compounds as our next generation therapeutic. And quite importantly, what I want to highlight is that another cool thing we can do is actually use existing antivenoms and just help them, fortify them. So some of these antivenoms are very, very good at being so cross-reactive just by being generated by the animal immune system, but they might not be the best at neutralizing the most toxic toxin, especially if it's not very immunogenic. So we can put a specific antibody in these polyclonal antivenoms to make them better and still use a combination of old and new to just get better patient outcome. Now, some of you might not be super interested in snakebite itself, even though that would be a crazy thing to think, but it can happen. So there's also a lot of these rules that you can apply to other fields. Some of these understanding on cross-reactive antibodies, how to make cross-reactive products, how to make the whole target space less complex. So things like neoepitopes in cancer are relevant to think about. HIV, viruses, we're talking about COVID and all of the rapidly mutating variants. All of these areas require smart antibody technology, smart vaccine technology, and a lot of these rules and thoughts apply to these fields as well. And just as an outro, a bit of an outlook on what we'll be working on more and more. Sorry, just had a bit of an internet glitch, is actually looking at some of the next gen fields. These are buzzwords, but these are real buzzwords, big data and AI. I'm now looking at different ways of taking a lot of these new lessons that are learned in other fields and applying them to snakebite on a computational level. And one of the things that we're doing now is we're sequencing all of our antibody outputs. So when we're doing all of these rounds of phage displays, sequencing all of the outputs, looking at how the sequences evolve, like what are we missing? Are we losing anything? Looking at the whole evolution of sequences, which ones are doing well, which ones are doing poorly, also in cross-panning. and adding a lot of other data sets. So looking at epitope data, looking at paratope data. And once you start combining all of this data, you can start unraveling the underlying rules of cross-reactivity and specificity. And if you understand the rules, you can use AI to also start predicting some of these patterns and use this big data to actually start understanding when you have 1,000 antibodies, you could have this tool that can actually tell you, okay, this is where they might bind. These ones might be the best to actually look and take forward and characterize further in vitro. So kind of a pre-selection step, computational pre-selection step that just makes the antibody discovery more efficient. And obviously the long-term goal would be to remove a lot of this antibody discovery in vitro and make it all in silico, computational, start predicting antibody sequence against specific targets. And that's honestly a bit sci-fi. And when I proposed this to our collaborator who works a lot with AI, he also said that if we manage to do this, it would be the Nobel Prize. So I think that's a bit further down the road, but still you need to have grand visions and ideas so you have a goal to aim for. And this was just, again, to show you that if you're maybe not interested in antibody discovery, but you love coding, you love doing Python work, you love doing AI, you can still work in a field that is as cool as Snakebite, because we all know it's hard to sell coding to be very cool. I've tried a lot during my PhD. Wasn't super easy, but if you combine it with Snakebite, that is where really the holy grail of coolness sits. So don't be discouraged by that, be encouraged, and please get in touch if you are interested in doing projects or collaborating or learning more about what we do. And this year is just to thank everybody who's involved in our research, all of our collaborators, our funders, everybody who's making all of this work possible and allowing us to explore these novel concepts. And finally, thank all of you as well for spending your Saturday to actually learn a bit more about Snakebite. So thank you very much.
Thank you very much, team. Your assistance here has been much appreciated. There's quite a few questions. Unfortunately, there's not enough time to go through them, but one that has popped up more than once is future outlook. Like, what are we looking when it comes to how far down the line when it comes to human monoclonal antibodies? Everybody seems to be interested in that. Estimations or just, how the outlook is would be much appreciated.
No, that's a very good question. And if I had a definitive answer, I'd be very happy. I have some suggestions, some guidelines. For instance, for the Welcome Trust, we promised that we would have a set of recombinant monoclonal antibodies that neutralize all key medically relevant toxins from sub-Saharan Africa in vivo in 4 years' time. So that would mean we have, it's an ambitious goal, but it's a feasible goal, but that would mean that we have something to take forward into preclinical and clinical trials. Now, the biggest question mark for me is not really the development. I think that it's going to work and it's not going to take a ridiculous amount of time. What's really difficult to answer is the regulatory pathway afterwards, because we're moving from a blood product, which is currently being used and evaluated, to therapeutics. And there is no comparison that we can draw. We don't know if we're going to follow the same clinical path as current products, or if they're going to start changing a lot of the rules and a lot of the necessities that we need to hit before it can be clinically approved. And that is the biggest question mark. And that could be, if we're going the normal clinical path, I'd say probably 2 to 3 years. On top of that, it's actually quite fast within Snakebite. But if they change a lot of things, then yeah, the number goes up to a level that I don't know and I can't answer, unfortunately.
Understood. So Timothy Jenkins, thank you very much. To mention here, Konstantinos Kalogeropoulos, who is the Greek guy, the Greek person that has been participating and is actively engaging the DTU. He wanted to be with us. He couldn't, unfortunately, due to time constraints as well. So he's accessible for students out there. And I hope it's okay for people to outreach to you if that's
yeah,
fantastic.
Definitely. That's also one of the reasons I want to highlight him. I think he's not just a good role model for anybody here from Greece. He's an awesome guy, great scientist. And I'd be happy to connect people with him. Or as I mentioned, please do get in touch if you just want to know more, if you're interested in doing a thesis or a project, an internship also. Just contact me and we'll see what we can make happen.
Fantastic. I will make sure to share your email address if that's okay.
Yes, please do.
Absolutely. Great. So thank you very much, Tim, for this. Appreciate it. And we can now move on to the next speaker, who is, again, one of our own here in Greece, Vasiliki Moulasioti. She's actually doing her PhD right now. Snake antivenom, Greek species. Nothing has been done so far. So yeah, Vasiliki, you have the floor.
Thank you. Let me share my screen. Okay, do you see the right screen without?
Yes, absolutely. You're good to go.
Okay, just give me a minute. So my name is Vasiliki Moulasioti. I'm a chemist and I'm a PhD student at the University of Ioannina, Department of Chemistry. I'm very honored to be here in this webinar and have the chance to meet the experts in this field. So Thomas, thank you very much for this invitation. The presentation is about snake antivenom and a few things about my PhD project in this field. So as previous speakers have said, globally, millions of envenomings occur each year, which correspond to some thousands of deaths. Major incidence of snake bite estimated is in South and Southeast Asia, followed by Africa and South America, whereas envenomation is lower in Europe and North America. Some data about Europe. Snake envenoming is an uncommon but potentially severe injury in Europe. 3 snake species of Vipera genus are responsible for most significant envenoming in Europe: Vipera berus, Vipera aspis, and Vipera moditis, as you can see in the figure, and the distribution for each species. So the venom composition of such snakes is presented here. The 3 dominant protein families are PLAs, serine proteases, and metalloproteinases. Phospholipases A2 present neurotoxic and myotoxic effects. They promote inflammation, pain, paralysis, and intensify hemorrhage by inhibiting coagulation. Serine proteases rupture capillary vessels, execute their primary toxicity by altering the hemostatic system of their victims and by inducing edema and hyperalgesia. Hemostatic effects include perturbation of blood coagulation, fibrinolysis, platelet aggregation, with potential deadly consequences for snakebite victims. Metalloproteinases are zinc-dependent. They exert hemorrhagic activity, and they have the ability to influence multiple steps of the blood clotting cascade, resulting in a lethal combination of systemic hemorrhage and incoagulable blood in prey and victims. Here is a chronology of envenomic therapy, how it all started from 1890 when serum therapy paved the way for antivenom therapies. In 1895, Calvete introduced the polyclonal antivenom, and 2 years later, Paul Ehrlich was the scientist responsible for the first large-scale production of antiserum. In 1940, Pauling introduced the first antibody model, and through the years, many discoveries have helped improve antivenom manufacture, like PCR, DNA sequencing, hybridoma technology, phage display, leading to the latest research about human IgGs using phage display. Despite the advancements in science, since Calvete introduced antiserum therapy, antiserum still remains the only effective treatment. Many factors are likely to have contributed to this, including the neglected character of the disease, the complexity of finding an alternative treatment, and the low economic incentive for companies to develop treatments against envenomings. So the current technology is about venom extraction, mainly horse or sheep immunization, blood drawing from the animals, isolation of antibodies from the serum, concentration and formulation, and of course bottling of the antivenom. An antivenom is characterized as polyvalent or monovalent. If the hyperimmunizing venom is obtained from a single species, then it is considered a monovalent antivenom. if the antivenom contains neutralizing antibodies raised against 2 or more species of snakes, then the composition is considered polyvalent. The current antivenom in market are polyclonal antibodies, or their Fab2 fragments for hyperimmunized horses or sheep, as I said, with a whole venom. Some data about antivenom in Europe. The last 20 years, the cost dramatically increased. In 2017, 8 antivenoms were available. And I have to mention that there is no standardized protocol for antivenom administration across Europe, and no antivenom licensed by the European medicine agencies. Most of the European industries has halted their production. Now across Europe, I think the only available antivenom is Viperfav and Viperatab from Inoserp. Viperfav is a polyvalent antivenom against Viperaberus, Viperaspis and Viperamodhitis species, and it's a Fab2 fragment from equine origin. And Viperatab is a monovalent antivenom raised against Viperaberus, and it's a Fab fragment from ovine origin. Grace is applied to the Viperfav antivenom, and its dose costs about 3,000 euros. These expenses, of course, are covered by the state, but still the cost is very high. Many problems are associating with antivenom. Adverse reactions are high in list. They are classified by WHO as early, which refers to anaphylactic reactions, and pyrogenic reactions, which occur within 24 hours of antivenom administration, and late reactions, which refer to serum sickness, which develop between 5 and 24 days of treatment. In addition to these adverse reactions, antivenoms do not always be effective against various complications related to snakebite. And furthermore, there are situations where all the venom components are not targeted by the antivenom. For instance, many studies have shown that there are antivenoms which do not recognize the low molecular weight proteins. And of course, the stability in liquid form is also a problem which must be faced. So new approaches are being developed in order to surpass all these problems, and they include monoclonal human IgGs, single domain antibodies, aptamers, small molecule inhibitors like varisplatib, which is a potent inhibitor of secretory PLAs and was used firstly to treat acute coronary syndrome, endogenous inhibitors from snakes, and IgY antibodies. I will focus on antibody production in chickens because this is what we do in our lab. We synthesize peptides and we immunize chickens in order to produce antibodies for various proteins and for both treatment and diagnosis. So why chickens? Because they require lesser antigen dose for IgY production. We collect the antibodies from the egg, so this method is not invasive for the animal. And of course, chickens are phylogenetically distant from the humans. And this method is cost effective because large amounts of antibodies are produced in the eggs. And before closing this presentation, I want to say a few things about my PhD project in this field. We selected only clinical important toxins. We selected PLAs from Viperaberus and Viperamodhitis species. We did studies about antigenicity, hydrophilicity, and hydrophobicity of these toxins in order to select specific peptide epitopes. We synthesized the peptides in solid phase according to FMOC-TERT-Wutil strategy. And then we conjugate the peptides to a sequential oligopeptide carrier, which has been developed in our lab. We don't use the KLH, which usually is used. We immunize the chickens. And as I said, we isolate the antibodies from the egg yolk. We purify them through affinity columns. And we do in vitro assays for antivenom specificity, immunoassays like ELISA, and in vitro tests for hemorrhage estimation, some cell cultures. We try to work according to the 3 R's in antivenom testing: refinement, reduction, and replacement. And I want to say that I faced many difficulties when I started this project. The two main difficulties were the extremely high cost of venom and toxins that our lab couldn't buy. And I'm very grateful to Ilias and Thomas that provided me with the venom. And also in Greece, I couldn't find someone to guide me in this field because, as I said at the beginning, I'm a chemist, and this research wasn't not my field of expertise. But I do want to get involved and do something in this. And I have to say that the first concept was to work with Milos viper, Microviperra zweiteri, but I couldn't find any data in UniProt or PDP, and I couldn't work with this. So this is a future project, maybe. I hope I will find the chance to work with this snake and do something about the antivenom for this species. And so this is the research group of our lab. In the head, our professor Vasilis Tsikaris, and my colleagues Vasilios Mousis and Evgenia Fotou. Thank you for your attention.
Hi, Vasiliki. Thank you very much for the presentation. Essentially, there's a fellow chemist that asked a question while you were referring to the supply of venom for the PhD and so on, and she asked, like, what beginning your thesis and so on, choosing such a very niche, let's say, subject, what were the challenges, what were the difficulties that you faced, and what would be your quote unquote advice as to someone who wants to get into that in Greece at this time?
I think a lot of research has to be done in species in Greece. I'm a chemist. I didn't have no idea about snakes or what. I started studying and finding someone who knows about this stuff. Eventually, I found you and Ilias for providing me the venom. Because as I said, in Greece, it's very difficult for most of the laboratories, to the best of my knowledge, to buy this venoms or toxins, because I need the venom and the toxins to for my ELISA assays. So it's very difficult to start a PhD under this situation. I don't know what else has to be done. I think more interest in this field.
Mm-hmm. Well, thank you very much for this. At this time, we will take a short break. There has been an issue with the talk of Sir David Warrell, which is going to be later down the line. So we will take this, let's say, opportunity, because we have been nonstop since the morning, and we will pause for about 40 minutes, and we will carry on with the speaker from Costa Rica. I hope that's okay with everyone from the panelists and the attendees. Great. So it will be open. The room will remain open, and I will reactivate as soon as I have, as soon as the next speaker is in line. Okay.
Okay.
I think we are ready to resume if everyone is at the panel at this time. This actually gives us more time. Senor Gutiérrez, welcome. Thank you very much for being here with us.
Thank you for the invitation. Greetings to everybody.
Hi from Greece. How is the weather over there?
Oh, just great. We are starting our dry season. Juan Calvete can testify that.
Okay, so this gives us more time, actually. So because I would assume it is 7:51 Costa Rica time. Am I correct? Or 8?
Yeah, 7:52.
7:52. Fantastic.
In the morning.
In the morning, yes.
Oh.
Yeah, we had to go a long way to get all the different time zones in this webinar. Luckily, we were right in the middle of it all, Australia and Latin America and so on.
Yeah.
So thank you for being with us. You can absolutely begin with the presentation. The next speaker is going to be Carl Barden from MedToxin in the United States. And that is going to be at 8:45.
Yeah.
8:45 Costa Rica time. So yeah, let's take time, make the most of it. Please go ahead, you have the floor.
Thank you very much. Well, first of all, it is my great pleasure to be here. I thank Thomas for having contacted me and inviting me to this activity, and congratulations for the organization of this activity. It is a pleasure to share this seminar, this webinar with colleagues, students, and friends. And really thank you for the invitation on my behalf and on behalf of Instituto Clodomiro Picado. So I will share my screen. Okay, can you see it?
Yes, absolutely.
Okay, so I will let me put the time. I will present today an overview of the problem or the issue of snakebite envenoming in Latin America and the Caribbean. And again, thank you very much for the invitation. First, I will go over our continent, describing the magnitude of the problem in different countries in terms of cases of envenoming and deaths and so on. So we will start in the north with Mexico. Mexico reports, the public health authorities of Mexico, about 8,000 cases per year of snake bites. And the most important, Mexico has a wide variety of snakes, particularly rattlesnakes. Mexico is the center of origin and dispersion of rattlesnakes. And two of the most important species are Crotalus scutulatus and Crotalus atrox, the western diamondback rattlesnake. But there is also the moccasin, Achistrodon bilineatus in Mexico. In southern Mexico, Chiapas, Bothrops asper, which over there is called Nauyaca, is very important. And then 1 to 2% of the cases are inflicted by coral snakes of the family Elapidae and the genus Micrurus, such as Micrurus nigrocintus. So there is a wide variety of snakes in Mexico. And the study of the venoms is quite interesting because there is a mosaic of variation in venom composition. And this has been studied by several groups, the group of Juan Calvete, the groups in the Autonomous University of Mexico, and so on. Then we move to Central America, our region, where snake bites snakes inflict between 5,000 and 6,000 bites per year. The country with the highest incidence is Panama. In fact, in terms of incidence, this means number of cases per 100,000 population, is the highest incidence in the whole continent. Then our country, Costa Rica, Nicaragua, Honduras, and Guatemala, we have about 600 cases per year. And then El Salvador and Belize report about 150 cases respectively. So it's an important issue in our region in Central America. And in our region, the World Health Organization classifies in category 1 species of snakes, that is the species that inflict the highest number of cases and the most severe ones. The two species that you see in the top, Bothrops asper, which we call in Costa Rica terciopelo, is called Equis in Panama, and is called Barba Amarilla, yellow bird in the rest of the region. Then the rattlesnake, the Central American rattlesnake, Crotalus simus, which is distributed in the Pacific dry corridor of our region. And again, coral snakes inflict between 1 and 2% of the cases, such as Micrurus nigrocintus, the most important species. By far, Bothrops asper is responsible for for the largest number of cases in our region and the most severe ones. So in Costa Rica, for instance, about 70% of the cases are inflicted by Bothrops asper. Then we move to the Caribbean, and in the large islands of the Caribbean, Cuba, Puerto Rico, Jamaica, Haiti, Dominican Republic, there are no snakes in the families Viperidae and Elapidae. But there are 4 islands in the Caribbean, Aruba, Martinique, St. Lucia, and Trinidad, where venomous snakes of the family Viperidae are distributed, and they inflict between 50 and 70 cases with very few deaths. One species which is very interesting and important in Martinique, it's an endemic species of Martinique, is Bothrops lanceolatus, the original fer de lance. And this snake is interesting because it's very similar in many aspects to the other species of Bothrops, but clinically, it inflicts a unique pathophysiological effect, which is a very severe thrombosis, which causes infarctions in the heart, in the lungs, and in the brain. So clinically, these envenomings are very complicated, and they need to use, of course, antivenom. There is another endemic species in the island of St. Lucia, which is Bothrops caribeus, which is very similar to Bothrops lanceolatus, and then Bothrops atrox in other islands. Then we go to South America, where snakebite and venom is a huge public health problem. The country having the majority of the cases is, of course, Brazil, which is a continent by itself, with almost 30,000 cases per year, and a wide variety of snake species. Then we have countries with high number of cases, such as Venezuela, 7,000, Colombia, 3,000, and then the other countries with about 1,000 or more cases per year, except Uruguay, which has less than 100 cases per year. So snakebite and venom is a really big problem in South America. And there is a long tradition in the study and the handling of this problem in South America. The first antivenoms in the Americas were produced in Brazil in 1901 at the Instituto Butantan. There is a wide variety of venomous snakes of the families Viperidae and Elapidae in South America. In the northern part of South America, Bothrops asper is important. Then in the Amazon region, in the wide range of Amazon region, Bothrops atrox is the most important one. Although also Bothrops bilineatus, an arboreal snake, is also present in the Amazon region. Then in the southern region of Brazil, Argentina, and so on, Bothrops jararaca and Bothrops alternatus are important, and Bothrops mugeni is present in the states of Mato Grosso do Norte, Mato Grosso do Sul in Brazil. So the genus Bothrops has a wide diversity of species and expansion in South America. Also in South America, we have the rattlesnakes, particularly the species Crotalus durissus, the South American rattlesnake, with several subspecies. The most important one is Crotalus durissus terrificus. And also there is a wide variety of coral snakes, such as Micrurus frontalis. By the way, recently a very nice, beautiful, and extensive book on the biology and toxinology of coral snakes has been published, and I recommend it to all of you because it's a very comprehensive text on coral snakes, which is a fascinating group of snakes with a fascinating natural history and toxinology as well. So in terms of the numbers I have given to you are similar to the ones described by Jean-Philippe Chipot in this publication in PLOS Neglected Tropical Diseases. His data are based on data from the Ministries of Health. And all in all, it reports 57,000 cases of envenomings in Latin America with 370 deaths. Again, I say based on the statistics of the Ministries of Health. But we know that many cases of snake bites in Latin America and elsewhere do not attend hospitals or health facilities, and therefore they are not included in the official number. So it is very likely, and this has been recently demonstrated in the region of around Manaus by the research group in Manaus and Instituto Butantan, where they found localities, riverine localities in the Amazon region where a lot of people do not attend health facilities and therefore are outside the official statistics. So these numbers are probably higher in the region. Okay, so in addition to the number of envenomings and deaths, we have to look for other consequences of snakebite envenoming, which usually have been overlooked in our field. And there is a need, therefore, to document the extent of physical and psychological sequelae. I mean, people that survive an envenoming and yet develop some permanent physical and psychological sequelae. And I give you an example here, a case shared by Professor David Warrell from the University of Oxford, is a snakebite case by Bothrops atrox in the Amazon region of Peru, bitten in the upper extremity. This person had to travel to several health facilities which did not have antivenom, eventually reached a facility where he received antivenom, but by then the local necrosis, the local tissue damage was quite extensive, and therefore he had to be amputated. So we don't know exactly the extent of this sequela in Latin America. This is an area that requires further investigation. But in addition to the physical sequela, muscle loss of muscle, chronic kidney failure, neurological sequela, amputations, and so on, there is growing evidence in the world that the psychological sequelae are quite important in snakebite envenoming. And in several studies in Sri Lanka and sub-Saharan Africa, Nigeria, they have described what is called in psychology post-traumatic stress syndrome, which is a very serious psychological consequence, together with anxiety, depression, and other psychological manifestations. So we have to look not only for physical sequelae, but also for psychological sequelae. To analyze health issues from this perspective, there is a concept called DALY, which means disability-adjusted life years that have been lost. So for instance, if a person is bitten when he or she is 20 years old and his life expectancy is 70 years old, this person has 50 years of disability-adjusted life years lost. So when the impact of snakebite envenoming is estimated by this parameter, it becomes quite evident the seriousness of this pathology. So another issue is how to document beyond the biomedical aspects of envenoming, how to document the social suffering in vulnerable communities. By social suffering, I mean, for instance, if this person, if this peasant is bitten by a snake and suffers some sort of consequences, this affects not only this person individually, it affects his family, it affects his community. And this is particularly important in indigenous communities in our countries, which, by the way, are affected to a larger extent than other types of communities. So how to document this social suffering, how to follow up these cases, this is a challenge for not only for biomedical research, but for social research as well. Anthropology, psychology, sociology, economy, and cultural studies, and so on. These two graphs to the left are from a study by the group of Robert Harrison in Liverpool and show the close relationship between poverty and mortality in snakebite envenoming. Each dot here is a country, and you have here the snakebite-induced mortality in the vertical axis, and in the horizontal axis, you have the per capita government expenditure in health and the GDP per capita. And you can see very clearly that the lower the investment in health and the economy of the country, the higher the mortality. So snake bites are clearly a disease of poverty, and this is why they are considered a neglected tropical disease. But there are other issues that have not been investigated in detail and are quite relevant and interesting. For example, what is the effect of migrations on the incidence of snakebite? In Central America, as you well know, especially in the northern part of Central America, from Honduras, El Salvador, and Guatemala, there is a massive migration of people who live in impoverished conditions, a massive migration to the north through Mexico, aimed to get to the United States. And along this migration, these people get into regions with abundant snakes, and therefore snakebite and venomins are likely to be a problem in these migrant populations. We don't have information about that. Also, snakebite increase, the incidence of snakebite increase in natural disasters, and this has been demonstrated in flooding in India, for instance, Bangladesh, and also in Latin America, in Ecuador, and other regions. And of course, our region is affected in some countries by armed conflicts, which impact on the populations because they have to migrate to other regions, and this also may have an impact on snakebite and venom. So this is a very complex problem that requires and deserves further investigation to better understand the effect of these aspects on the incidence of snakebite. Also, an interesting question, which is being asked by several research groups, is how will climate change affect the incidence of snake bites in Latin America? It is clearly that climate change is affecting the distribution of snakes, the distribution of human populations. So how will this impact in the incidence of snake bites in the future? And for this, there have been several algorithms and models that approach this question. One example are the studies by Carlos Yáñez Arenas from the Autonomous University of Mexico. And in this case, he and his group have predicted regions with higher incidence of snakebite problem. So this is the present time situation, and the red regions are the regions with highest risk of snakebite, using a variety of parameters. And here to the right is the projection that they have done for the year 2050. And you can see that there are regions like, for instance, this region in the border between Mexico and the United States, and particularly this region in the Amazon region, which will see an increase in the incidence of snake bites because either of changes in the distribution of snakes, changes in the use of land by human populations, demographic aspects, and so on. So this is another area that is very interesting to predict what is going to happen and to take interventions in public health. Now I will move to the clinical presentation of snake bites, which I gather you have already heard from some of the panelists about this. But in Latin America, we have three main syndromes, I would say. The most common syndrome is the one inflicted by envenomings by species of the family Viperidae, in particular by species of Bothrops. And these envenomings are characterized initially by local tissue damage, local effects, characterized by necrosis, edema, hemorrhage, blistering, pain, and these effects can be very severe, depending on the volume of venom injected. And then these venoms also induce systemic effects, particularly coagulopathies. Blood becomes incoagulable, as you see here in this tube, collected from a patient bitten by a snake. The blood is incoagulable, whereas in contrast, a blood sample from a normal person clots normally. And also the other clinical manifestation, which is quite relevant, is bleeding, systemic hemorrhage, which may be manifested by bleeding in the kidneys, in the lungs, pulmonary hemorrhage, and the most serious consequence is a cerebrovascular accident, as observed here in this tomography shared by Dr. Rafael Otero from Medellin, showing a massive hemorrhage in the right cerebral hemisphere. And this may have, of course, serious neurological consequences. So this extensive bleeding usually brings cardiovascular shock due to hemodynamic alterations and may bring what is called multiple organ failure, which is the cause of death in these cases. So this is syndrome, and number one is the most frequent syndrome inflicted by viperine snakes. Then the second syndrome that we see in our region, particularly in South America and in some parts of North America as well, is inflicted by some rattlesnake species, not all of them. In particular, South American rattlesnakes of the species Crotalus durissus. And this is completely different or quite different from envenomings by Bothrops because it is associated with neurotoxic manifestations. So there is a picture of what is called descending neurotoxic neuromuscular paralysis, which starts with the palpebral ptosis, the closing of the eyes, oropharyngeal paralysis, difficult in swallowing, and then neuromuscular paralysis, which may bring death due to respiratory paralysis. As you see, there is no much local tissue damage. And another clinical manifestation in these cases is systemic muscle damage, what is called rhabdomyolysis, which results in the release of large amounts of myoglobin, the muscle heme protein, which is filtered in the kidney and is responsible for the appearance of colored urine, which is called myoglobinuria. And this abundant presence of myoglobin in the kidneys may cause acute kidney injury. So the clinical manifestation in these cases are rhabdomyolysis, muscle damage, neuromuscular paralysis, and acute kidney injury. And the reason why this clinical picture is so different from the Bothrops one is because these South American rattlesnakes contain high amounts of the toxin named crotoxin, which is a neurotoxic and myotoxic phospholipase, quite active, and very low amounts of hemorrhagic metalloproteinases. And then the third syndrome is characteristics of coral snakes of the family Elapidae, genus Micrurus, and is basically a descending neuromuscular paralysis characterized by ptosis, oropharyngeal paralysis, respiratory paralysis. And coral snakes do not inflict major muscle damage, nor other clinical manifestation. Is basically a neuromuscular paralysis due to the action of either presynaptic neurotoxic phospholipases or postsynaptic three-finger toxins, neurotoxins. So these are the three main syndromes, and this is why for the clinicians is very easy to know which group of snake caused the accident and therefore to select the antivenom. Locally, unfortunately, in our country, we have an extensive knowledge on the biochemistry and toxicology of snake venoms, and this knowledge has been increased significantly in the last decades by the introduction of the proteomics methodology with the leadership of Juan José Calvete, with whom we have had a very productive and fruitful collaboration over more than a decade. And other groups like our group in Costa Rica, groups in Brazil and Mexico have also contributed to the proteomic analysis. So we know the proteomes of many snake venoms from Latin America. And I give you here an example of a study by Juan Calvete and colleagues on the venoms of the rattlesnakes, demonstrating the difference or explaining the difference in the clinical manifestation of rattlesnake envenomings in our region. For example, here to the left is the proteome of the Central American rattlesnake, Crotalus simus. And you see there is an abundance and predominance of metalloproteinases, which are responsible for hemorrhage, necrosis, and so on. Whereas in the case of the venom of the South American rattlesnake, there is a predominance of the neurotoxin crotoxin with very little amounts of metalloproteinases. This explains why this venom is neurotoxic and myotoxic, not hemorrhagic, whereas this venom is hemorrhagic. Interestingly, these venoms show important differences according to the age of the snake. So in Central America, the newborn and juvenile rattlesnakes are more similar to the South American pattern than to the adult Central American pattern. And you see here how the newborn rattlesnakes in Central America have a high amount of crotoxin. So this issue of ontogenetic variation in venom composition is very interesting. We also know the biochemical features and structure of several toxins, such as this hemorrhagic metalloproteinase, and also the mechanism of action of the toxins. Our group in Costa Rica and other groups have been exploring the mechanism of action of hemorrhagic toxins, like illustrated in this diagram, myotoxins, coagulant toxins, and so on. So we have a good basis of knowledge of the venoms and their mechanism of action. And what we need to do is to translate this basic scientific knowledge to the clinical aspects and to the antivenom manufacturing. Now let's move to talk about the antivenoms in Latin America. As you know, antivenoms, animal-derived antivenoms, are the only scientifically validated and approved by the World Health Organization treatment for snakebite envenomings. And antivenoms production started, as I mentioned, in the year 1901 in Instituto Butantan in Sao Paulo, Brazil, by the famous Brazilian scientist Vital Brasil Mineiro da Campanha. And then other countries, as we will see, also took on the production of antivenoms. We have many manufacturers in the region. Our region is in a privileged situation in this particular aspects. Most of them are in public institutions, but also there are some private manufacturers in the region. And from the standpoint of the type of neutralizing molecule, the type of pharmaceutical active principle, we have two main types of antivenom in our region. In Costa Rica, Colombia, Peru, Bolivia, we produce a whole IgG type of antivenom. So we purify the immunoglobulins from the blood of horses immunized with venoms. In other countries such as Argentina, Brazil, and Mexico, they manufacture FAB2 products. And these are generated by the use of pepsin. Pepsin, a proteolytic enzyme, cleaves the Fc fragment in immunoglobulin and generates an FAB2 fragment. This is the landscape of public antivenom manufacturers in the region. As you see, there are manufacturers in Mexico, Costa Rica, Venezuela, Colombia, Peru, Bolivia, Brazil, of course, 4 manufacturers, and Argentina. So we produce different types of antivenom, and I am very pleased to tell that we have established a network of regional cooperation and partnership between these institutions in which we collaborate, we exchange, we do seminars, workshops, training programs, we do troubleshooting within a frame of solidarity and cooperation. I would like to briefly summarize how are antivenoms manufactured. Initially, what you need is the venom of the snakes, because this is the basis for the whole process. And in our institute in Costa Rica, we have a serpentarium with a large number of snakes from our country. So we collect these venoms. Venoms are immediately frozen and freeze-dried to be used in research or production. Then venoms in very low amounts, such that they do not inflict major damage to horses, are injected in this way subcutaneously to horses, using an immunization schedule that has been developed over the years. As I said, the amounts of venom that need to be injected to raise an immune response are relatively low. And every 10 days, we inject an additional dose of venom. And depending on the type of antivenom, we use different venoms. So we produced 2 antivenoms, the polyvalent one, which works against the pit vipers. And for this, we mix the venoms of Bothrops asper, Crotalus and Lachesis stenophris, the fer-de-lance, the rattlesnake, and the bushmaster. And this combination of venom generates an immune response that protects against most of the venoms of Viperidae in Central America. And then the other antivenom we produce for Latin America is the anticoral antivenom, for which we use the venom of Micrurus nigrocintus. Once the immune system of the horses has synthesized a good amount of antivenom, an accepted amount of antivenom, then we bleed the horses from the jugular vein. The volume of blood collected is very well. I mean, this volume is selected on the basis of obtaining a good volume of blood and antibodies, while not affecting the cardiovascular status of the horses, and this is closely followed by veterinarians in our institute. Then the blood is collected in plastic bags containing anticoagulant, and horse blood have the peculiarity that the erythrocytes sediment spontaneously So you don't need to use a centrifuge to sediment the erythrocytes in the blood of horses. And therefore, we leave these bags for 1 day. They separate the red blood cells and the plasma. We collect the plasma, which we use for antibody purification, and we resuspend the red blood cells in saline solution and reinfuse them to the horses to avoid a state of anemia. And this is called a manual plasmapheresis. And then with the plasma, we go to a laboratory where this plasma is fractionated. In our case, we use caprylic acid fractionation, which purifies the immunoglobulin fraction. And finally, this product, after a series of quality controls and formulation, is bottled in 10 milliliter glass vials and distributed to health facilities. So this is the basic protocol for antivenom production in our institute. And as I said, we produce 2 types of antivenoms, the polyvalent one against pit vipers and the anticoral one against coral snakes. Antivenoms need to be, because of the great variability in venom composition and the fact that antibodies are specific against the antigens for which they are induced, then it's very important to make sure that the antivenoms are effective in the neutralization of different venoms. And this is called the preclinical evaluation of antivenom efficacy. And for this, we have a set of techniques that we use for the clinical evaluation of antivenom efficacy. We check for neutralization of lethality, hemorrhage, edema, myotoxicity, coagulant effect, defibrinogenation, and so on. And also antivenoms need to be evaluated at the clinical level for efficacy and safety. And there have been several studies in Latin America where a protocol of controlled, randomized, double-blinded trials have been done, such as this one carried out in Colombia with our antivenoms by the group of Dr. Rafael Otero Patiño. So clinical evaluation of antivenoms after preclinical evaluation is very important. Then once you have an antivenom, let's suppose that it's a good antivenom preclinically and clinically, you have not solved the problem yet, because you need to ensure that these antivenoms reach the rural health posts, the rural communities where snake bites occur. So the question here is a question of public health. Do antivenoms really reach the regions in highest need? And there are a series of problems regarding this question. First of all, the epidemiological records in some countries are poor. So we don't know exactly where is the highest incidence of snake bites, and therefore where antivenoms should be deployed. Another problem is that in some countries, and particularly in the current fiscal crisis, there is a low budget for antivenom acquisition. So there is a risk of purchasing lower amounts of antivenom than needed. And then there is problems with the bureaucratic processes of acquisition. Also, sometimes antivenoms are kept in city hospitals and not deployed to rural settings where they are mostly needed. Then liquid antivenoms can be produced in either the liquid form or the freeze-dried form. The freeze-dried can be stored at room temperature, but the liquid antivenoms need to be stored in the fridge. And for this, you need what is called the cold chain, ensuring that the temperature is between 3 and 8 degrees centigrade all along the shell life on the antivenom. And this is not always available. And then, of course, a more general problem is that in some regions, there is a deficient provision of health services in remote rural settings. So this is an issue of public policies, health policies, a strengthening of health systems. And the final goal is, of course, to bring the antivenom to the rural health posts where they are needed. I'd like to show, for instance, the type of analysis that can be done in this aspect. And this is a study by Hanson and colleagues in Costa Rica. To the left, you see the incidence of snake bites according to the districts. And you see a highest incidence here in the southern part of the country, in the Talamanca region, in some districts in the northern part. And this is the distribution of hospitals and clinics. And you see, this is quite interesting, that in some regions where access to health facilities is more than 3 hours delayed, are regions with high incidence of snake bites. So after this information, we discussed with health authorities And now antivenoms are being deployed to primary health facilities where people can reach rapidly after the bite, and then they can be transferred to the hospitals. So this is the type of analysis that needs to be done in our region to ensure the availability of antivenoms to everybody in need. Then there is, of course, the need. I mean, you may have enough antivenoms for a country. You may distribute these antivenoms in an effective way to rural settings, but then you need to have specialized, trained health staff in the health facilities able to administer the antivenom and to treat the complications of envenomings. And for this, you need a good knowledge on the subject. And this is another strong point that is being worked in our region. So for this, it is necessary to prepare and validate national and regional protocols of diagnosis and treatment. This is, for an example, the one we have in Costa Rica, which we distribute massively to health facilities and health students. And also we have prepared this algorithm, which is placed in emergency services in our hospitals and clinics. And of course, this is part of a permanent education activities in health centers and teaching in university courses. So this is an activity that needs to be done permanently in our region because the health staff is permanently rotating in our facilities. And then last but not least, there is the issue of prevention. The best way to reduce the impact of snakebite is to prevent them. And there are ways to prevent them with good community education, such as using boots, avoiding the use of hands, the touching with the hands, the soil during agricultural work, and so on. So for this, it is necessary to do prevention community programs, which, this is very important, should be applied to the local contexts and should involve the communities. It shouldn't be a hierarchical, vertical approach that comes from the expert to the communities. It should be worked together with the communities. And therefore, this relationship with the communities should be dialogical, horizontal. And this is particularly important when working with indigenous communities, as we have done in Costa Rica. We need to take care and to develop ways to take care of people suffering from sequela from snake bites. And here I show a qualitative study that we did with a colleague, a sociologist from our institute, a qualitative study in southeastern Costa Rica, which showed the impact of this sequela in terms of the life and the quality of life of these people. And this needs to be taken into consideration because right now, these people are not being properly attended in Latin America and other parts of the world as well. I would like to round up my presentation saying that this issue of snakebite envenoming, which for many years lacked without attention by health authorities and other stakeholders in the world, fortunately, due to the advocacy efforts of many people and groups in the world, we have managed to impact on the World Health Organization, who took this problem, incorporated snakebite as a neglected tropical disease, created a resolution for attending this problem, and in the year 2019, developed this global strategy for prevention and control of snakebite envenoming, which is being unfolded right now and is based on 4 pillars. So the goal is to reduce snakebite deaths and disability by 50% before the year 2030. So there is space for optimism that this problem is going to be reduced on a global basis. And in Latin America, the Pan American Health Organization, which we closely collaborate, and the Department PANAFTOSA from Pan American Health Organization is taking the lead in the development of this strategy. And this has created this network of antivenom manufacturing laboratories. And we work very actively with these laboratories and with national health authorities in this respect. So I'd like to mention that the work that has been done in Costa Rica over more than 50 years has been a collective effort of this group of people and other people that no longer work with us, together with colleagues from many different countries with whom we have established very productive partnerships and collaborations. And it is through this collective effort of many people that we have managed to provide solutions and understand this problem. So I'd like, again, to thank you very much for the opportunity to present this overview to you. I'd like to thank Thomas and his colleagues for the invitation. And this is my email address in case somebody would like to receive more information about my presentation and any other aspect of our work in Costa Rica. So thank you very much. I'll be glad to take comments and questions from the audience. Thank you.
Thank you. Thank you very much for this. Actually, one question that popped up when speaking in class with a professor of mine was, what is the nature of the relationship between the Clodomiro Picado Institute and the University of Costa Rica? Because apparently students found out videos on YouTube of people extracting venom and so on within the institute, if that is not, if that is correct.
Yeah, well, our institute belongs to the University of Costa Rica. Our university has a research system based on institutes and centers for research, and we are one of these institutes. So we belong to the University of Costa Rica. We are adscribed to the School of Microbiology of the University of Costa Rica. So all of us are employees of the University of Costa Rica. Mm-hmm.
So essentially it's one and the same.
Yes, exactly.
Right. The second question Nikos essentially sent was, when it comes to the clinical image of allergic reactions to poor antivenom, what is the case there and what is being done to address that? Better antivenoms?
Mm-hmm. Yes, the adverse reactions to antivenoms largely depends on the physicochemical quality of the products. And there is a large variation in the world of the physicochemical quality of the products. But when an antivenom is produced by good manufacturing practices, when the active principle has a good level of purity, the incidence of adverse reactions for example, in Brazil and Costa Rica is between 10 and 15% of the cases. Usually these adverse reactions are mostly mild. They are cutaneous reactions characterized by rash and itching mostly. And in very few cases, we have systemic reactions such as bronchospasm or hypotension or colics and so on. So these are easily managed. These 10 to 15% of adverse reactions, which, like I said, are mostly mild, are easily managed by the administration of antihistamines and steroids in the clinics. And this is controlled in terms of 15 minutes. So the antivenom infusion can be continued after that. So when we visit hospitals in Costa Rica, this is not a big issue because of the fact that reactions are relatively scarce and they are mild. In case of reactions which are more severe, like for instance, bronchospasm or hypotension, then the indication is to administer adrenaline by the intramuscular route, but this is seldomly the case. A different situation occurs in other regions of the world where the incidence of adverse reaction may be as high as 50 or 60% of the cases, and in these cases, there are many more severe reactions. But it has to do with the physicochemical quality of the products. And this is why it is so important, the action that the World Health Organization is taking right now, because the World Health Organization issued the guidelines for the production, control, and regulation of immunoglobulins, which provides very specific indications on how to present antivenoms. And also the WHO is developing a program of what is called pre-qualification or risk assessment program for antivenoms, where experts evaluate the antivenoms and the facilities. In fact, Dr. Calvete is involved in these evaluations and other colleagues. So they also visit the facilities, and therefore they through these pre-qualification programs, they are stimulating the improvement of antivenom production in the world. But in general, to conclude my response, if an antivenom is manufactured with good manufacturing practices, even though it's an animal-derived product, it can be a very safe and effective product, and is the central therapy for snakebite and venom.
And at the same time, you're actually keeping animals within the Instituto, Clodomiro Picado Institute, am I correct?
Yes, we have a farm which is located nearby our facilities, and we keep about 120 horses.
So you have the entire, let's say, supply chain for the raw material.
Exactly. We have the entire supply chain. And I would like to mention that we distribute the antivenoms for all Central American countries, for Ecuador, eventually for in some cases of emergency to South America. But I like to mention also that we manufacture an antivenom for Sub-Saharan Africa. And this antivenom is being distributed currently to Nigeria, Burkina Faso, Mali, Benin, Central African Republic. This is a different antivenom manufactured with African venoms. I didn't mention this because I was referring to Latin American situation, but we also have this other type of antivenom.
So it has expanded.
It has expanded,
from Latin America.
Yes, exactly.
Understood. So that would also make for a good, let's say, stock in the back in case of research, right? So 2 for 1 kind of thing for the venom supply.
Well, the reason for this was to contribute, because there was in the decade of the 2000, there was a call, an emergency call for the lack of antivenoms or the poor availability of antivenoms in Africa. So our institute and other institutes around the world decided that we would like to dedicate part of our facilities and our capacity to contribute. I mean, this is a partial contribution because the need of antivenom for Africa is huge, but there's a contribution for solving this problem in Sub-Saharan Africa.
Understood. So if anyone else has a question at this time.
I have a question. I want to ask, do you believe that it's better to immunize animals with a mixture of venoms, or it's better to immunize different animals with one single venom and then somewhat mix them for antivenom? I mean, is there a problem with immunosuppression for the animal?
This is a very interesting question. You can do both ways. And in fact, the WHO guidelines accept both ways. You can prepare monospecific antivenoms in different animals and then mix them. Or you can do as we do, we mix the combination of venoms in the single animals. For practical purposes, it's much easier to do the way we do, because then if you have different animals with different venoms, you have to combine the serum with different potencies and adjust the potency and so on. So for practical reasons, we haven't done that, and most of the manufacturers don't do that. But this is perfectly feasible. And what you mentioned of immunological relationships or interactions, this is another important issue that is being investigated, because we know from our experience that some venoms exert a negative immunomodulatory role over other venoms. So we have studied this, and the way we solve it is to inject the venoms separately in the same animal, separately in the time. So the animal receives all the venoms, but not the 3 of them at the same time, to avoid this negative immune interaction. For example, in the old time, when the Sanofi Pasteur used to produce an antivenom for Africa, it was an antivenom, a polyvalent antivenom against both viperids and elapids. What they did was to immunize a group of horses with viperid antivenoms, a group of horses with elapid antivenoms, and then they mix the plasmas. But most of the manufacturers, as far as I know, produce the antivenom, injecting all the set of venoms into the animal. Of course, the big question here is which venoms to use that ensure a cross protection, and there is a lot of research going on for this. But this is a very interesting. In general, the immune response of animals to venoms is a challenging topic because of the complexity of the venoms and the complexity of the immune response.
Yes, no, thank you very much.
You're welcome.
Okay, so thank you very much for your time. Thank you, José María. It's very much appreciated you being here and on such short notice. It was great talking to you. We look forward to making more of these. Apparently, there's quite the demand. And yeah, if that's all, we can move on to the next speaker, if that's okay with you.
Thank you. Yeah, absolutely. And again, thank you very much, Thomas, and thank you very much, everybody. It has been a pleasure to be here, and look forward to future interactions.
Fantastic. Thank you very much. Thank you, Mr. Gutiérrez. So at this time, I would like to welcome two fantastic people, Carl Barden and Mara Roberts, essentially from the land Florida, United States of America. Welcome.
Thanks for coming us. Thank you. Hi.
Hi, guys. Good morning or
Hi.
afternoon, whatever time it is where you are.
Yes, it's like 20 minutes to 5 p.m. in Greece at this time, but there's people from all over. So Senor Gutiérrez, who's in Costa Rica, so essentially you must have pretty much the same time zone in this.
Well,
so how are we going to proceed with this? Can you hear us well?
I can hear you. He can't hear much, but I'll translate. Go ahead.
Okay. So essentially what I was going to say is we're going to do this a bit differently than the rest of the presentations. I'm going to include a video with Carl and yourself, the facility, the work that you're doing over there at MedToxin Laboratories, essentially. And yes, right after that is done, we will continue with the Q&A, if that is okay with you.
Great.
Fantastic. Give me just a little bit of time.
Do a video, and then we'll do Q&A.
Okay, great. And I talk right to the screen.
Okay, I will carry on with the video.
What's happening, guys? Ken and Harkin here with the Reptile Discovery Center, and today we're going to be watching Carl Barden milk a few different species of venomous snakes so he can get the venom, and we're going to find out what they do with that venom here. It's going to be a great episode, so stick around.
Went to college, and I was trying to figure out a way to build this serpentarium, and I couldn't figure out how to do that, at least at that time, on a zookeeper's salary. So two of my older brothers were flying at the time, and my oldest brother said, look, this flying thing might provide you blocks of time, and it might be a good way to seed your serpentarium plan. So I got all my ratings and I started teaching, I started flight instructing, started flying for a living, and at the same time, of course, I was accruing snakes, and I started producing venom in my kitchen. I had snakes all over the house I was in, and Bill Haas started buying my venom. And so then we were kind of able to keep going, and we found the current property. So we built the lab 18 years ago, 1999. And then I flew for another, almost 15 years. And the whole time doing both things, producing venom and running business and flying for my income. And so we opened it up, built the rest of it, and then opened it to the public in 2009. So it's kind of been my whole life to get it to this point, but so far, anyway, it's worked.
That's fantastic.
And it's in decline in many areas where it was.
Here we go. Yeah, that's a big, heavy-bodied snake. That's the worst snake bite you'll get in the United States just because of the amount of venom that animal has. Not necessarily the toxicity, right?
What's that?
The bite of an eastern diamondback is pretty bad because of the amount of venom they put in you, not necessarily because of the toxicity.
Toxicity too. Toxicity also, eastern diamondback venom is no slouch, but quantity is the big part of that.
Gotcha. Here we go.
You're good, no, you're perfect. All being normal parts of an adult diet.
It's a little gnarlier. Ooh, look at that. It's a little gnarlier when you got a big bodied snake like that that has a lot of strength in it.
A serious bite by this snake in an adult or
My
a
God.
child can rapidly be fatal if not treated promptly and properly with antivenom. Discovery Center.
Dude, that was a lot of venom, man. I mean, it exploded out of the animal's mouth. I mean, look at the yield that guy gave you there. So that is more than enough venom to knock you out.
The bad snake emblem on
Here we go.
a cobra's hood. Its real purpose is probably to confuse predators. This snake his name website, and he's named website because he's on our website as nature, and he's getting old now, and he's slowing down. But as a young snake, he was terrifically nervous and very, very spirited.
The Asiatic and his close relative, the Indian cobra,
Yeah, I can tell.
are still
Pretty good for an old
responsible
man.
for many snakebite deaths
Don't
each
you
year,
bite? No.
ranging over a large part of Southeast Asia. The monocle cobra is still quite common in many areas.
And he's still a good producer for his old age.
Yeah, that looks amazing. How old is he?
Cobras possess
Think
a
about
small,
20.
thick,
Oh my God, he wasn't
front
born here, but we
fang.
got him young.
And as the
And,
snakes are induced to bite, you can often see these fangs.
Okay, so I've stopped sharing at this time. This is just to give a little bit of an idea to everyone watching what it is exactly that you're doing. We're trying to showcase the entire ecosystem from the production of essentially venom extracting all the way to the anti-venom and all the steps that are included in this. So the first question would go along the ways of, you have a lot of animals, if we're not mistaken, you have more than 1,000 animals currently housed. What are the challenges of keeping and breeding and producing essentially the venom from a healthy stock?
So Thomas wants to know, we have over 1,000 animals, and what are the challenges of breeding, keeping, and producing venom from a collection of that size?
Yeah. So the challenges are several-fold. Like any large animal collection, there's a tremendous amount of time dedicated to just basic husbandry. So keeping the animals clean, fed, in reproductive weight, that kind of thing. All of that stuff plays a part in it. And then obviously you have this added layer of risk when 90% of the collection is venomous. Training help, finding help, making sure that your help is safe, adequately prepared, trained for the day ahead, that kind of thing. All of that plays a role. And there's, of course, an element of liability involved. It's different if you were working with, let's say, a less dangerous animal, that kind of thing. And of course, snakes, like all animals, require veterinary care from time to time. So reptiles, a lot can be done through preventative maintenance and maintaining a healthy collection, proper quarantine procedures, this kind of thing. But like anything, there are times where you wind up with a viral issue or a bacterial issue or this kind of thing in a snake or a group of snakes or that. So all of that plays a role. And then, of course, the next level is the production aspect of things. I hope that answers that.
Absolutely. And so I have the questions already. So the second question would be, you guys are working together. How tough is it for someone to be assisting the person that is extracting the venom and the cooperation that you have between you two?
Oh, it's not hard at all to assist. Carl and I have implicit trust in each other. So that is really, really helpful. And because both of us are in such a focused flow state, if you will, at the collecting table, it's really easy to do this as a team. We work well together. We understand each other
as a team, because this is not a one-man job, as I understand. And
no,
keeping,
most of the time it is both of us.
like the big cobras.
So Thomas was wondering, how easy is it to work as a team in this thing and not as a one-person operation?
Yeah, you know, of course, that Tom, Maura, and I have been doing this together now for 10 years. So kind of an unspoken language there. And as you might imagine, if you bring a new person into that picture, once again, there's an entire period of training and getting the person familiar with what you do and how you work. And of course, the animals and the distances and this kind of thing. But we've been at it so long together, you begin to, you know what the other person is doing probably before they're doing it. So that's a big help.
True. And you are producing venom essentially. For people that don't know, that there's sides in this, pharmaceutical companies buy that, but at the same time, a lot of it goes on research. Am I correct?
So he was wondering, a lot of our venom goes to pharmaceutical companies, but also a fair amount of it goes to research. Is that correct?
Yeah, still we think research for the last 25 years is typically about 20% of our production a year. And if this fluctuates, there are occasional projects or time to time where that figure goes way up. Most of the time it doesn't fluctuate too much below that. Sometimes those projects are ongoing for years, and sometimes those projects, of course, come and go rather quickly. And the venom quantities vary all the way from 10 milligrams up to hundreds of grams of material based on what the application might be. But research, if I were to just say how much of it over the past 25 years, typically about 20% of our work a year is research driven.
Understood.
Lots of researchers, as some of your audience knows, do their own production, particularly if it's a small quantity of material.
Understood. And there's an inherent risk involved in producing venom. People have asked, have you had snake bites? Have you had experience with different snake bites from different species? What was the reaction to that? And so on.
So how many snake bites? What was your reaction, and across what variants of species?
Yeah, so over the past 30 years, it's been 11 snake bites. I haven't had a snake bite in 16 years. So those first 15 years, as you might imagine, were a little bit bumpy. Across a variety of species, black mamba, green mamba, West African green mamba. So I've been bitten by 3 of the 4 species. That's very embarrassing. Several rattlesnake bites, cobra bite, green tree viper, copperhead, cottonmouth. So it's kind of been a mixed bag. And as some of you in the laboratory community may also experience, I am hypersensitive to venom. I'm allergic to venom now and have been since the early 1990s. So we've had 7 anaphylactic events across those bites. 2 of the bites were dry bites, a Mojave rattlesnake, no venom, copperhead venom bite, almost no venom. So the allergy problem is significant. And I'm also allergic to equine serums now. I'm still doing okay with the sheep. The ovine stuff is still fine at this point, but the equine stuff I'm allergic to as well. So managing those allergy problems and anaphylaxis has been a real challenge. Snakebite induced anaphylaxis is traumatic. I'm unconscious in usually 4 or 5 minutes despite my EPIs and an airway kit. So it's something we really don't take lightly. We've changed lots of our safety processes over the years, and we've dropped the number of snakes we handle in a day. And so things have gotten markedly safer over this past decade or so.
Understood. So another question would be, at the other side of this, like, what would be your opinion, and especially Carl's, when it comes to treating a snake bite, even as a patient? There's a saying that goes along the lines of keep giving antivenom, don't stop until all the symptoms subside. Is this the case? Would he agree with that, or would he not?
So is the process for you as a patient, do you believe in just keep getting antivenom until the symptoms subside, or as a patient, how would you prefer your treatment to go as far as the administration of serum?
Yeah, probably some of your audience on here is far more qualified to answer that question than I would be, but sure, we do have some say in the direction of our care, and historically our outcomes have been really good, and we attribute that to lots of serum early and continue serum as needed. So I feel very strongly about giving plenty of antivenom, and our outcomes have seemed to indicate, and granted this is across a range of snake bites. In other words, some of them are more serious than others, but we've had very good outcome. I have all my fingers, this kind of thing, and we attribute that to early and adequate antivenom.
Mm-hmm. Now, there's another question a professor asks. Essentially, he says, so handling snakes, do you think it's a question essentially of when, or a question of if getting
Do you
bit?
find it's inevitable to be bitten in doing what you are doing?
That's a great question, Thomas. You know, as I talk to my colleagues around the world, most of us feel pretty strongly that this can be done without accidents. I'm now approaching about 180,000 handlings without an incident, but we're probably about 450,000 total handlings. And we feel like when the numbers get up into those hundreds of thousands of interactions, that doesn't include feeding and cleaning and packing and all the other things you do with snakes. It is certainly the possibility of a mistake is there. And anybody I know in the United States, but really even around the world with really large numbers of venom extractions, and I don't mean 10,000 or 50,000 or 80,000, I mean hundreds of thousands of handlings. Those persons have accidents, that they have bites. You know, none of us are keeping score, so to speak, but you do have a sense of how many accidents you've had and what you could have done to prevent them, and could this have been done better. And Bill Haas, the early model here, really kind of the father of large-scale venom production. Certainly there were many, many accidents over the course of his career, but you know, the last figures were in the neighborhood of 2 million venom extractions. So yeah, I don't know where that works out for him. Right. But anyway, but we think it can be done. As I said, I'm 15 years now without an incident. And I'm getting old and slow, and I'm hoping to not have any more. So we'll answer that again in 10 years.
Understood. So would you think that it's the human factor essentially that we're going up against instead of the snake actually going for the kill here?
You think that it's the human factor that you're fighting up against rather than snake behavior?
Sure. You know, it's always the human factor, right? You know, the animals, the poor guys, they're under duress. We have this agreement, they have to pay the rent. I feed and house them, that kind of thing. And so we fill this unusual link in a chain. Would it be better if there was never any interaction? Sure. And maybe that day will come. We're not there yet. Somebody's picking up snakes somewhere if there's antivenom around. So, but yeah, the human factors is a big one. It's certainly the biggest player.
Okay, so just two more questions. So a student asks, do you face any issues when it comes to preserving and shipping the venom? And what would be the process that you're following after extracting?
So do you face any issues with preserving and then shipping the venom? And what is the process from when we collect it to when we do that, when we do ship it out?
Yeah. So most of the large laboratories are similar or the same. Not everybody is identical, but pretty much. So venoms are weighed and sold and shipped in lyophilized form and freeze-dried form. They are pretty stable biochemicals, and they're really stable once they're freeze-dried. So the venom is collected. All of the labware is kind of laboratory grade cleanliness. I don't say venoms are collected sterilely because it's not a sterile environment, nor is the snake's mouth. But everything is managed as you would any other sterile product in a laboratory environment. Then they're spun, filtered, if you will. The venoms are centrifuged under fairly high speeds to separate out any insolubles, this kind of thing. They're brought down to -70°C, and then they're freeze dried for often a significant period of time to really get that moisture content way, 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 way down, almost to nothing. And once you've got that freeze-dried stable powder, if you will, crystal powder, the venoms can be stored for long periods of time with little or no degradation. There's some research out there regarding the stability of venom components over kind of a course of different environmental challenges. And some of the molecules are really stable, and some of them a little bit less so. And as I said, that's why sometimes a researcher will extract and process his own material, because he wants to know exactly the procedure from the animal's venom gland to his microscope, so to speak. But most of the time across the spectrum, the production and processing is pretty uniform and results in a really good quality product, which can then be shipped literally around the world with little or no degradation or impact. And the researcher winds up, or the antivenom manufacturer, whomever it might be, winds up with a really good, stable product.
Fantastic.
I hope that answers that.
Of course, yes, it does. So another student asks, have you got a venom production line? Do you distinguish until what age you can extract venom from a specific animal, and from what age do you begin doing that?
So age range, from what age do you start collecting venom? And then what age do you retire the animal?
Yeah, that's a great question, Thomas. There's quite a bit of variability there based on the application. So as many of your listeners are aware, some venoms have ontogenic shifts, protein shifts, or toxin shifts over the course of the snake's life, or from a juvenile snake, which might be a prey shift involved, to an adult snake, this kind of thing. So sometimes it will depend on the application. If we're trying to make an antibody for an antivenom purpose, the hope is for a very broad spectrum, a broad sweep of snake toxins, if you will. So in those instances, we start pretty young. Sometimes we start the snakes on the line at 6 months and run them all the way up into late adulthood, 20 years on the venom line, that kind of thing. So you get a very broad spectrum antibody protection. That way, if your victim is bitten by a juvenile, all of those things are kind of represented, if you will. Otherwise, we typically start the snakes on the venom line at about 18 months of age, and they will often spend the better part of their lives. Sometimes we'll retire a snake. We've actually had rattlesnakes that stopped growing fangs. So they were genuinely geriatric at that point. They still produce venom, but they have no delivery system. So he can bite the collecting receptacle, and then we can pipette off the venom, but we realize the animal is now getting really old. So we'll often retire a snake in that instance. But it depends somewhat on the application. And then otherwise, it's from about adolescence in a snake's world, young adulthood, all the way up through the course of their adult lives. Many of our snakes now on the line 15 years, some of them longer than that.
Fantastic. So the last question would be, if I'm not mistaken, you would always be, my apologies, you would also be sort of a rescue center for animals like rattlesnakes or copperheads, snake removals, and so on. Would that be a correct statement?
So do we also offer, are we a rescue center for nuisance removals and unwanted pets and all of that?
Yeah, so the public part of the lab, which is the Reptile Discovery Center, is just that. It's a small reptile zoo. We are a rescue. We are a drop-off site for venomous animals that have been removed from yards or schoolyards or this kind of thing. Our entire coral snake line of about 70 snakes is built up of nuisance removals and snakes that have been found in yards and in developments, this kind of thing. So we do operate in that capacity as a rescue. Also for a whole host of other reptiles, lizards and tortoises and people's pet snakes. Their son went off to college and the mom didn't want the ball python anymore, this kind of thing. All of those wind up here, or many of them.
Mm-hmm. So a colleague asks, are there some basic steps someone should follow in case they get bitten from a specific snake? Would that be different? I guess what he's trying to ask is, would that be different, the reaction, if you get bit by a coral snake and you see no symptoms, with what would happen down the line, whereas you would get bit by a rattlesnake?
So Thomas was wondering, are your steps after you've been bitten by a coral snake or an elapid different than the steps that you would take if you'd been bitten by a pit viper?
So are we talking just in-house, our own setup?
No,
In general.
Generally.
Okay. Yeah, I was going to say, right, question for somebody else. In general,
Yeah.
we're talking about. Again, there are probably people on our webinar that are more qualified to answer that than we are. And, you kind of have to get into where are we talking about? Are we talking about Australian elapids? Are we talking about African snakes? Are we talking? So in North America, not really much first aid for snakebite. We don't use pressure immobilization here or anything of that nature. There's nothing really out there that the victim can do beyond we suggest people wash the snake bite. And then, of course, the old remain calm is a big help. But otherwise, in this part of the world, in North America, really, it's an antivenom issue, and it has to be done in a hospital. So our patrons, our visitors, we always recommend call 911 and or go to the hospital. We take ourselves to the hospital. We don't wait for an ambulance. But lots of people prefer that. They prefer to be transported by an evac or an ambulance situation. So again, I know in other parts of the world, Australia, Africa, this kind of thing, they're doing different things. But in North America, it's get to the hospital as quickly as possible.
Understood. Another question would be, would you also rescue?
1 second, Thomas.
Yes,
I'm just going to wait for María to come back. She's got, hold on for 1 moment.
Okay, sorry, Thomas, go ahead.
Okay, so the question was, would you also take rescues that would be species that are not endemic in North America? And do you think that is important? And the reason why we're asking this is because we have such an issue here in Greece, and it's growing all the time with people just bringing in venomous species. So yeah, essentially that's the question.
So do we take in animals that are not endemic to North America as rescues? And the reason that they're asking is because sometimes this can be a problem with people just bringing on all this non-native stuff, and then they don't have anywhere to put it.
Yeah.
Or when they're done with it, or it gets out.
So yes, we do take non-native animals. In fact, about half of what we get is non-native animals. And your observation in terms of what to do with that kind of thing is very valid. We run an adoption program here where many of the animals that get brought in are rehomed to suitable homes.
Non-venomous animals that are.
Of course, and all of that, I'm talking about non-venomous only, right? But that's an ongoing challenge, especially here in Florida, which is really the land of introduced species. So something we're always kind of looking at and trying to fine tune.
I understand. So essentially, these are the questions. We would like to very kindly thank you for taking the time and being with us, sharing your knowledge and insights.
So appreciate it.
It is very important to talk to people that are hands-on and are, right at the beginning of the supply chain. It's just of utmost importance.
Well, thank you, Thomas. We so appreciate you having us.
Thank you, and thank all of you guys. Fun to see all those guys out there.
Thanks,
thank you.
Guys.
Absolutely. Have a great day. Thank you, Carl.
You too. Talk
Bye-bye,
soon,
María.
Thomas. Bye.
Okay, so at this time, I would like to I would like to welcome Sir David Warrell, who's with us.
Not
Welcome,
sir,
David.
Thomas, not sir, please.
Okay, David Warrell, world-renowned professor, and so on. Welcome. Thank you for being here with us and for making the time and changing your schedule. It is very much appreciated.
Good, thank you. Thank you very much, Thomas, and thank you for asking me to join you for this meeting. I'm sorry I haven't been able to be present for some of the other sessions.
As we understand, yes.
Now, I hope you can see my screen, can you?
Yes, yes. That
is it full screen?
Absolutely, yes, it is now.
Good. Well, as again, thank you very much. It's very nice to speak to an audience of Greek academics of various stages, I gather. I'm not quite certain about your backgrounds, and I do apologize in advance if my language, as well as being in English, which is bad enough, I use too much professional jargon, because I'm a medical doctor, but also a very enthusiastic amateur herpetologist. And I'm sorry I haven't been present for some of the earlier talks in which perhaps the taxonomy of the Greek venomous snakes was discussed, and undoubtedly the proteomics of their venoms, if my friend Juan Calvete has already spoken. The thing I want to emphasize first of all is that snakebite is an enormous burden, particularly in low and middle income countries. And that's why my title slide is of a typical busy day in a hospital in West Bengal in India, very crowded. And as we now know, many of those patients lying in this crowded ward will have suffered from snakebite. So it's in tropical countries, a long way from Europe, that snakebite has its greatest impact. Well, for many years, people suspected that snakebite was the most neglected of all the so-called neglected tropical diseases. And the former Secretary General of the United Nations, Kofi Annan, who was born in Ghana in West Africa and had some personal experience with snakebite, thought that this was the biggest public health crisis you've likely never heard of. And the trouble was that before 2011, the only data available were hospital records. And these grossly underestimate the problem of snakebite, because as we all knew, in rural areas of developing countries, most snakebite victims don't come to hospital for one reason or another. They die at home, or they're treated by local traditional practitioners, and their deaths are unrecorded. And it's clear that community-based surveys were needed to give us a true impression of the impact of snakebite in these countries. And the first of these studies to really tackle this problem was the so-called Million Death Study, orchestrated by the Registrar General of India and run by a group of expatriate Indians working in the University of Toronto, led by my friend Dr. Prabhat Jha. They determined the cause of death of people dying in the community using the technique of verbal autopsy. Now, this is quite controversial for some diseases because it relies on questioning the bereaved relatives or the neighbors of the deceased person about the circumstances of their death. And as you can imagine, if someone dies with a high fever, it could be a whole lot of different things, malaria, dengue, all the rest of it. But snakebite is sufficiently dramatic and memorable to make this the subject of a reliable determination by verbal autopsy. So this study, which was extremely well planned, used 6,671 randomly chosen sample areas in all parts of India. Each one of these areas had a population of about 1,000 people. And this covered the whole country. It was independent of hospital underreporting because it was based on information gathered in the communities from the deceased people's homes and villages. And here you can see some of the questioners taking details from a relative. The results of this study were quite staggering because they found that in 2005, deaths attributable to snakebite in India were 46,000, and these are the 99% confidence intervals for that figure. This amounted to 1 snakebite death for every maternal death in India and 1 snakebite death for every 2 HIV/AIDS deaths. It was appalling that nearly a third of these victims were children, and this figure was more than 20 times higher than the official figure that the government of India was working on, and partly explains why the government, the Ministry of Health in India, had completely underestimated the size of this problem. Because it was a regional study, all parts of India, it was possible to see which states of India suffered worse. For example, here, Uttar Pradesh had 9,000 snakebite deaths a year, and so on. And this enabled the authorities to deploy scarce resources to the states most in need of help. And this paper was published in 2011. And the results of this study, more than anything else, influenced the WHO, the World Health Organization, and persuaded them to include snakebite in their list of neglected tropical diseases. And also they categorized snakebite as a grade A, a top priority. This encouraged international funding agencies such as the Wellcome Trust to put generous funding into research on a topic which up till then had been very greatly neglected. There was a follow-up to this study published last year by the same group. So during the period 2000 to 2019, no fewer than 1.2 million Indians died of snakebite. This amounts to an average of about 58,000 a year, which seems to be increasing, probably as a result of increasing population. So these are the latest guesstimates, best guess figures for the number of people dying of snakebite in each continent. You'll see that in Europe, there are very few, probably far fewer than 50 deaths a year now. In Latin America, about 4,000 deaths. Africa is the large unknown. There are enormous countries, enormous populous countries, like Democratic Republic of Congo, for example, where there are really no data and very difficult to get reliable information because of the state of war that's existed there for so long. And in Oceania, it's particularly New Guinea has a large number of deaths, but Asia, above all, not only has the largest numbers of deaths, but the best documented snakebite mortality, as I've already said, amounting in India alone to about 58,000 a year. And I want to emphasize too that for every person who dies of snakebite, several survivors are left with permanent handicap of some sort or another, a physical handicap, for example, having lost a digit or limb that's become gangrenous after snakebite, or a more subtle form of morbidity, persistent psychological disability triggered by the very stressful circumstances of a snakebite. Now I want to go over the basic principles of snakebite envenoming from the clinical point of view. And I do realize that most of you are not clinical people. And so again, apologies. And if any words I use are unfamiliar, do please ask about it. First of all, there are effects of envenoming, that is injection of venom at the time of the bite. And incidentally, even if you're bitten by a venomous snake, there's only about a 50% chance that venom will be injected. The rest of these bites are so-called dry bites for reasons that are not fully understood. Venomous snakes can bite you and actually produce fang puncture marks in the skin without injecting venom. Anyway, if there is envenoming, the effects may be seen locally at the site of the bite. And the common ones grouped under the term cytotoxicity. I think that earlier speakers may have attributed particular venom toxins to these particular activities, physiological effects. In the case of cytotoxicity, these would be digestive hydrolases like phospholipases A2, zinc metalloproteinases, hyaluronidase to some extent, other hydrolases that are snake digestive enzymes. So there's local swelling, bruising, blistering, and in worst cases, necrosis of tissue involving skin, subcutaneous tissue, and muscle. Then there are the generalized effects, which are obviously more life-threatening. There are a group of effects under this term hematotoxicity, comprising coagulopathy, that's disturbance in the blood's normal system of achieving hemostasis after an injury, spontaneous bleeding, I'll be showing you examples of that in a moment, and also leakage of fluid and blood from the blood vessels caused by impact of these of toxins on the permeability of vascular endothelium. This leakage will result in hypovolemia. That's the technical term for really having too small a volume of blood to circulate. And this is going to lead to a fall in blood pressure. Then there are neurotoxic effects. These are all seen at the peripheral neuromuscular junction, and they present as a descending paralysis. Again, I'll be showing you examples starting with paralysis of the upper eyelids and then progressing to life-threatening paralysis of the respiratory muscles. Various forms of cardiotoxicity, cardiac rhythm may be affected. The heart may fail because of myocardial heart muscle failure, again resulting in shock or low blood pressure. Impact on muscle, particularly the phospholipases A2, the myotoxic phospholipases A2 can damage muscle not only locally at the site of the bite, but also throughout the whole body, causing what's called rhabdomyolysis. And one of the complications of rhabdomyolysis is kidney damage. But this is just one of many mechanisms by which the kidney may fail as a result of snakebite envenoming. And the technical term for this now is acute kidney injury, or AKI. So look, I apologize in advance. Those of you who are squeamish should certainly keep your eyes closed for the next 20 minutes or so. But I'm always told that, don't pull your punches, David. People want to see what snake venoms can do to humans. It will perhaps stimulate them to work harder in the laboratory to find out mechanisms and cures for these devastating effects. So to illustrate cytotoxicity, which is seen particularly after envenoming by Viperidae, but some Elapidae, particularly the spitting cobras of Africa and Asia, and also the burrowing asps, a small group, the subgroup of the Lamprophiidae. And my example here is from the African black-necked spitting cobra. This is a little girl in Nigeria when I was working there in the 1970s. Unlike the classical expected effects of an elapid venom, which is to cause neurotoxicity, these venoms are predominantly or exclusively cytotoxic. So this little girl arrives with a massively swollen arm and chest only 18 hours after the bite, and then there's progressive evidence of destruction of the superficial tissues, the skin, the subcutaneous tissue, and so on, as a result of the action of the venom. Then there are the hematotoxic effects, particularly caused by vipers, but by many of the elapids living east of Weber's line or Wallace's line in Southeast Asia. So Australia and Papua and New Guinea and some of the islands of the Western Pacific, and some colubrid venoms also cause these effects. So here you see spontaneous bleeding. This man is coughing up blood. This is bleeding from the gums, a very important clinical sign of impaired hemostasis. A fatal cerebral hemorrhage this little girl is bleeding per rectum. And I should be mentioning this again in a moment. A simple test of failure of hemostasis is the 20-minute whole blood clotting test. It has the merit of requiring only a new, clean, dry glass tube or vessel, not expensive apparatus. So it's an appropriate bedside test in many of the countries where snakebite is most common. So in this particular case, you'll see that the victim is bleeding freely from razor cuts made at the site of the bite. That's one of these bad and not recommended methods of first aid. And the blood that's been left in this glass vessel for 20 minutes has failed to clot, showing that the clotting factors have been consumed as a result of the procoagulant venom enzymes, and the liver was unable to keep pace with this destruction of clotting factors. And at this stage, the blood will no longer clot in response to surface activation, in this case by the glass wall of the vessel. In real life, it would have been activated by damaged vascular endothelium, a site of injury. Now, neurotoxicity, and I'm going to illustrate this from a case I saw early on in my time in living in Thailand for 7 years in the 1980s. So this child was bitten while asleep at night by a Malayan crate, which at that time was not recognized as a medically important species in Thailand. These are the marks made by its upper and lower jaw teeth. These are the venom fangs. And when we first saw this child, 3 hours after the bite, he was already showing early signs of neuromuscular failure, failure of neuromuscular transmission. So the technical term, and many of these technical terms, by the way, are pure Greek, so they should be very familiar to this audience. Well, I pronounce this ptosis, but you might call it ptosis or something like that, I don't know. It's paralysis of the levator palpebrae superioris muscles, that's Latin, which normally raise the upper eyelids. And it's also affecting the muscles that control his eyeballs. So he's got what's called external ophthalmoplegia. He would have double vision because the optic axes are no longer in parallel. He's also got early paralysis of the facial muscles. And you may notice that he's a bit blue in color. And this means that his gas transfer is beginning to fail due to early respiratory muscle paralysis. So he's becoming cyanosed. So a bit later, he has more pronounced ptosis and paralysis. 7.5 hours after the bite, he's no longer able to protrude his tongue or open his mouth. And at 9 hours, he has to be intubated and mechanically ventilated to keep him alive, because his breathing muscles are no longer able to produce adequate gas exchange in his lungs. So here he is being ventilated. In this case, it was actually manual ventilation using an anesthetic bag. So this makes the important point that because you haven't got a special machine, you shouldn't give up. You can keep patients alive just using manpower or woman power. In this case, a relay of medical students, doctors, nurses, and relatives, all helping to keep this boy alive. And I'm glad to say that this was successful. So here he is 6 hours after the bite, ready to go home, although he still has a slight degree of ptosis. I think his expression is reassuring that this has been a successful case. Now, cardiotoxicity, 2 examples. This is one of the tropical rattlesnakes, Crotalus terrificus, in Brazil. This patient went into acute left ventricular failure. This chest X-ray shows fluid in the lungs and an enlarged heart. And unfortunately, this was a fatal case. And the heart muscle at autopsy showed extensive damage caused by phospholipase A2 toxins in this tropical rattlesnake venom. Here's another example, and this happened in my own country, in England. We have only one venomous species in England. You have another subspecies in Greece of Viperoberus. But this woman nearly died. She was admitted with a very horrible looking electrocardiogram, suggesting to the cardiologist that she had what's called global ischemia, or shortage of blood supply throughout the heart. It was far more likely actually to have been a direct effect of the Viperoberus toxin on her cardiac muscle. Anyway, I'm glad to say that with antivenom treatment, she made a rapid and complete recovery. And here's a more dramatic example of a so-called Takotsubo cardiomyopathy. This is where a venom, this is just one example. In this case, it's the Japanese mamushi, or Gloidius blumhoffii, causes a curious dysfunction of the cardiac muscle, so that the apex of the heart fails to contract, whereas the base of the heart does contract. Now, generalized muscle breakdown. I mentioned this before, the technical term rhabdomyolysis. This would be an early sign. The patient's plasma becomes rather brownish. This is myoglobin, derived from skeletal muscle, that's been released through the damaging effect again of phospholipase A2 myotoxins. This is the urine here, also brown, and this is the urinary sediment. These are plugs, casts of the tubules, the renal tubules, formed of concretions of myoglobin. And this is myoglobin stained in a cross-section of the kidney, as it were, blocking the tubules in the kidney. Here's one example of this happening, again from a tropical rattlesnake bite. This young girl has signs of neurotoxicity, so she's got bilateral ptosis and facial paralysis, and her urine is very dark brown due to this myoglobin staining. And probably the most severe example of rhabdomyolysis is caused by some sea snake venoms. This is from my late colleague, greatly revered colleague, Alistair Reid from England, who worked in northwestern Malaya and Penang Island. And this is one of his patients, a Malayan fisherman, again with signs of paralysis, trismus, spasm of the masseter muscles caused by active breakdown through the effect of phospholipase A2 and brown urine. And another patient, it's agonizing for this patient to have his muscles passively stretched because of the effect of the active breakdown by myotoxins. And the kidney, here are some effects of the kidney. Sea snake bites can also cause kidney damage, but probably the worst venom in the world for damaging the kidney is that of the two species of Russell's viper in South Asia, the Western Deboia russellii and Eastern Deboia siamensis. So here's a patient in Sri Lanka showing the myotoxic effects with, again, breakdown of muscle causing myoglobin. This patient from Burma has kidney damage caused by shock, by persistent low blood pressure. That's another effect of Russell's viper venom. And this is an example of what's called microangiopathic hemolysis, where the endothelium is damaged and fibrin is deposited on the lining of blood vessels so that the red blood corpuscles, as they flow past, are mechanically damaged. These are so-called schistocytes or helmet cells, red blood corpuscles damaged through rubbing against the rough wall of a blood vessel. And in a way, a beautiful example of deposition. The red staining here is fibrin. That's the end product of the blood clotting cascade. Fibrin plugs deposited in this is the glomerulus, in the glomerular capillaries and in the peritubular vessels. So various different mechanisms by which the venom of Russell's vipers can cause kidney damage, which may be fatal, particularly in countries that don't have a well-developed system for supporting patients with chronic kidney damage. Now on to treatment. Again, I'm just going to give you the basic principles. So before the patient gets to hospital, it's very important to reassure. A very frightening experience, I can assure you. I've been bitten twice by venomous snakes, and I was extremely worried. Of course, I probably knew rather too much about the likely effects. So anyway, it's important to try and immobilize the victim, because any movement, any muscle contraction will squeeze the blood vessels, the lymphatics and the veins, and promote the spread of venom through these channels from the site of the bite to become generalized. Remove any tight bindings around the bitten limb, because it's like to swell. And really the only first aid method that has any evidence going for it at the moment, and I'll be showing you next to what this amounts to, is the so-called pressure pad immobilization method. Transport the victim as safely as possible, but as passively as possible, to medical care, ideally in the recovery position. That's lying prone on the left side, where the patient is least likely to inhale vomit if they were to vomit. Prevent early death on the way to medical care. Early deaths can result from a very low blood pressure. That's the reason for having the patient lying flat. Respiratory obstruction, this could be just simple as the tongue falling back, or obstruction through vomit or paralysis. And avoid harmful treatments. And many of these treatments are those that certainly I, as a Boy Scout, was taught, such as making incisions at the site of the bite, suction, and so on, tight bands of various sorts. This is the pressure pad immobilization method, which is very simple and undemanding. All you need is a pad of some sort of material, doesn't matter what it is, to apply local pressure over the site of the bite. The aim being to compress the veins and lymphatics draining the site of the bite, and so delaying the spread of venom taken up into those vessels. So this sort of pad, roughly 6 by 6 by 3 centimeters, secure this very tightly with a non-elastic bandage, and then splint to ensure immobilization of the bitten limb. Now on to the medical treatment. Again, basic principles. I think that all patients who are known or claim to have been bitten by snakes should be admitted for at least 24 hours of observation. The reason is that you can really never be sure whether the patient has been bitten by a venomous or non-venomous snake, unless, of course, they bring the snake and it is definitely non-venomous. There's always uncertainty there. And the effects of envenoming may develop rather slowly. For example, I've looked after patients with coral snake bites. That's a Western Hemisphere elapid snake, which causes paralysis, where no signs of paralysis appeared for 14 hours after the bite. And if this patient had been sent home, it could have been disastrous. So 24 hours observation, reassurance, and don't forget analgesia. Most snake bites, and there are some exceptions, are exceptionally painful, as I know to my cost personally. A very key question, has this patient got such an important systemic effect of envenoming that is destroying the ability of the blood to clot? So is the patient suffering from anti-hemostasis? And this can be checked simply with the 20-minute whole blood clotting test that I've already demonstrated. And the key question for the doctor is whether antivenom is indicated. And this is quite a controversial question. Certainly in many European countries, there's been a reluctance to use antivenom. Certainly in the United Kingdom, there was a period when it was believed that antivenom was more dangerous than envenoming. And it was my late colleague, Alistair Reid, who finally reversed this opinion. Our French neighbours were much more slow to come round to the use of antivenom, even though it was a Frenchman who really developed antivenom for the first time, Albert Calvete, at the end of the 19th century. So this is a key question. Does the patient need a specific antidote to the snake venom? The clinical history, or the story, the evolving story from the patient is very important. And if the patient's unconscious, of course, you will have to rely on the help of a neighbor, a relative or colleague who can give you the story. You need to know the sequence of events, the chronological history, as much information as possible about the snake. Of course, if the snake has been brought, that's very helpful if you have someone competent to identify it. If not, these days, you can easily send a photograph to an expert and get very quick advice. The patient needs rapid assessment because they may be absolutely on the brink of death. They may be moribund and they may require urgent resuscitation. And then eventually a full physical examination, which should be repeated at intervals to follow the evolution of envenoming. Examination of the bitten limb is an obvious priority, looking for fang marks, evidence of interference before the patient reached hospital. There are a lot, particularly in the developing world. Traditional treatments are very popular, as I said already. So incisions, application of snake stones, use of herbal remedies, either topically or ingested, and very often the use of tourniquets. Some of our patients in Bangladesh, for example, would have multiple tourniquets, not just applied to the bitten limb, but to the other 3 limbs as well. By the way, a tourniquet is a very tight band, the aim of which is to cut off the arterial blood supply to that limb. And clearly this is a very dangerous form of first aid because left on too long, it can cause irreversible damage to the limb, quite apart from the snake venom. One's going to be looking for evidence of bleeding, bruising, and lines across the lymphatic vessels, tenderness and swelling, and the extent and rate of spread, and the local lymph nodes. For example, on the lower limb, this would be behind the knee and in the groin. In the case of the upper limb, it would be behind the elbow and in the armpit. The lymph nodes will become enlarged, painful and tender as the venom components reach them. Shock is very important to determine. This is an important effect of snake venoms, particularly actually the European vipera. I'll be talking about those a bit later. Shock is often one of the most important effects and is an important cause of the, fortunately now, rare deaths from vipers in Europe. So you'll be looking at the rate and rhythm of the pulse. The ECG I've shown you already. The echocardiogram is very useful as evidence of failure of the cardiac muscle, evidence of depleted circulating volume, and evidence of bleeding, and secondary effects of shock, which may render the patient unconscious or their urine output may fall. Here's just one example of the importance of assessing hypovolemia or impaired circulating volume. This is a puff adder, easily recognized from these arrow-shaped or U or V-shaped markings down its back. This is one of the most important and common causes of venomous snake bites throughout the vast savanna areas of Africa and into the Middle East. And this patient I saw in Kenya shows massive swelling with formation of blisters. And I think you can well imagine, in this case, the venom toxin, of course, leakage of plasma and blood into the bitten limb, this leakage might well have depleted the circulating blood volume. However, in this case, when we measured his blood pressure lying flat, it was normal, absolutely normal for a man of his age. However, when we sat him up, it fell from 135/70, this is millimeters of mercury, to 95/60 millimeters of mercury. And this is good evidence of hypovolemia. So this is a way of assessing what is an extremely common and important effect of envenoming, particularly with snakes like the puff adder, but also more generally. I talked a bit about neurotoxicity, and I mentioned that the earliest sign was drooping of the eyelids, or ptosis, bilateral ptosis. And this Sri Lankan lady, envenomed by a Bangor ceruleus, a common crate, shows early ptosis. She's struggling to keep her eyes open by contracting the frontalis muscle in her forehead. And you'll notice also she's lost her normal facial expression through paralysis of the facial muscles. And a patient from Papua New Guinea, bitten by a Papuan taipan, Oxyuranus scutulatus. We're asking him to look upwards towards this pen. And as you can see, he's unable to raise his eyelids fully to completely open the pupils. And this is early ptosis. And in itself, it's not life-threatening, but it's extremely important because it shows that there is neurotoxicity in this. The effects of the venom have caused neurotoxicity, and this may well progress in a descending fashion, initially involving the muscles innervated by the cranial nerves, the nerves that leave the skull directly, and then progressing to the muscles that protect the throat and ultimately allow you to breathe, the respiratory muscles. As far as bleeding and coagulopathy is concerned, a very useful sign, first of all, is there continued bleeding from the fang marks. This patient, bitten by a Malayan pit viper, Calicilasma rotostoma, has persistent bleeding 40 minutes after the bite. This implies that the blood is not clotting. And the sign I've already told you about, bleeding from the gingival sulci or the gums, is a very useful sign of damage to blood vessel walls, not serious in itself, but if this is happening in vital organs such as the brain or the gut, clearly it could be life-threatening. You can detect coagulopathy if you have a well-equipped laboratory. As I've said already, for many developing countries, the best thing may be a simple bedside test. This test is certainly not as sensitive, as reliable as a modern coagulation laboratory, but it has the advantage of being quick and simple. And it has remarkably good sensitivity and specificity for detecting a fall in fibrinogen. This is one of the principal blood coagulation factors, to less than 0.5 grams per liter. So I think this is a useful test. Now, I've mentioned antivenom. Now, what on earth is antivenom? Well, first you need to catch some snakes, and these Irula tribesmen in India, in Mamallapuram, in Tamil Nadu, are expert at finding snakes. Within a few minutes of starting out with them, we found this saw-scale viper, Echis carinatus. You need to milk it of its venom, and then you need a cooperative large animal, a horse or a sheep, or perhaps in the future a llama, a camel-like animal, and immunizing these animals with initially tiny but increasing doses of venom stimulates production of neutralizing antibodies. And at some point, the level of antibodies is sufficiently high to bleed the horse the red blood cells are returned to the horse immediately. And from its plasma is refined antivenom, which is then administered to the patient intravenously, always intravenously. And in the developing world, it's very often nurses or health officers rather than medical doctors who will be treating snakebite. So antivenom is a polyclonal immunoglobulin with various degrees of purification, and certainly a very imperfect product, but one which I think has saved innumerable lives since being introduced at the very end of the 19th century, but very great room for improvement. You don't give antivenom to everyone who claims they've been bitten by a snake. You look for evidence that the risks of antivenom justify the benefits of antivenom justify the risks of antivenom. So shock, illustrated by these two patients, spontaneous bleeding, neurotoxicity, myotoxicity evidenced by this myoglobinuria, and very rapidly progressing local envenoming. This swelling is just sweeping up this man's arm. He's bitten by a Echis romani in Nigeria. You can practically see the swelling progressing. Antivenom alone may not save the patient's life. You may need other system support. This patient is having his circulatory system supported by fluid replacement. This man is having artificial ventilation. These two patients are receiving treatment for acute kidney injury, renal replacement therapy. In its simplest form, peritoneal dialysis in a township hospital in Burma, rather more sophisticated in a mission hospital in South India. This is hemodialysis. There are a lot of other drugs which have been used for snakebite, and it's shocking to see that they are still being used, even though their benefits have been totally rejected and their dangers have been widely publicized. So corticosteroids are widely used, I think mainly because doctors feel they've got to do something, antihistamines as well, and so on, blood products, including fresh frozen plasma, and so on. One good treatment, though, that you can safely give any patient who's had a snake bite, even a non-venomous bite, is a booster to their tetanus immunity, because bites, penetrating injuries do carry a risk of tetanus. And so it's quite reasonable to give a booster of tetanus toxoid. And it also may provide some reassurance to a patient that at least something is being done to help them. Now, finally, I want to say a few words about snake bites in Greece. And I must admit, first of all, that I've never treated snake bite in Greece. However, on many occasions, I've been asked to advise about snake bites that have occurred in Greece, both in usually in British tourists, but also sometimes from Greek doctors asking my advice. So I do apologize. I can't speak with great authority here. I have tried to read the literature as much as possible and to talk to colleagues who are more familiar firsthand with the snakes that causing problems in Greece. So this is a quick summary of the venomous species. So Vipera amidites meridionalis, widely distributed and generally recognized, I think, to be the commonest cause of potentially serious snakebite in Greece. Vipera berus bosniensis, just in the very northern part of Greece. Vipera either Yersinia graeca or Vipera graeca. I would have been interested to hear what Wolfgang Wüster thought about the taxonomy of your Greek meadow viper. That has quite a restricted distribution in northern Greece. A marvelous snake, certainly my favorite European snake, is Macrovipera schweizeri, the Milos viper, which is restricted to the Cyclades Islands. And then the Ottoman viper, Montivipera xanthina, which is quite widely distributed, not only on the Greek mainland, but also in these islands of the Eastern Mediterranean. And finally, a non-Viperidae snake, Malpelon insignatus, which is widely distributed in Greece, which is one of the few so-called colubrid, using it in a wide sense, that has been shown capable of causing envenoming. So let's start with this extraordinary painting. If you go to the Prado Museum in Madrid, the famous picture of Titius by the famous artist Titian, showing poor Titius was a giant, and he's having his liver torn out, should actually by, I think, by vultures, not by ravens. But if you look at the bottom right-hand corner, a very interesting detail added here, and sure enough, this is Vipera ammodytes, the horned or long-nosed horn or sand viper. Sand viper is inappropriate because it liked rocky terrain, not sand. And this is, I think, correctly credited as being Europe's potentially most dangerous snake. But I want to say here that I've been struck by reviewing the clinical literature in all the countries where Vipera ammodytes occurs in Europe. What a wide variety of severities and manifestations there are. And this is surely going to be an absolute field day for Juan Calvete and his Venomics crew to take apart this so-called species, not really at the level of taxonomy, but the level of venomic diversity, because it's quite clear to me that some of the devastatingly severe effects of Vipera ammodytes envenoming, for example, notably published from Croatia, are not seen elsewhere. So there clearly is enormous variation in venom composition throughout this so-called species and its geographical distribution. These are two Greek specimens of Vipera ammodytes meridionalis. And by the way, I'm grateful to my colleague Matt Wilson, who's made available a whole lot of very good images of Greek snakes. Now, the clinical effects. First of all, the local effects are many of those I've already illustrated, and they can be quite severe. Certainly, necrosis of bitten, that is, gangrene of bitten digits, is quite frequently mentioned in accounts of Vipera ammodytes bites. Neurotoxicity, I could find reports only from Croatia, from Split, not from Greece. But again, there may be publications in the Greek language literature that I haven't seen. So I'm very open to being taught on this subject. But it seems to me that clinical neurotoxicity has been reported from very few places, possibly only Croatia, even though the PLA2 presynaptic neurotoxins of ammodytes venom, I believe, are present in widely throughout its geographical distribution. So the effects of neurotoxicity, as we've already discussed, are ptosis, paralysis of the eyeballs, difficulty swallowing, difficulty speaking, and possibly more extensive weakness, although I need to be convinced that these neurotoxic effects are ever life-threatening in themselves, although this is suggested from the Croatian literature. Shock is an important effect of ammodytes, caused by hypovolemia, but also by myocardial damage. And anaphylaxis, which is a familiar syndrome from Vipera berus envenoming in the United Kingdom. Bleeding, abnormalities, as you'll see from these dramatic illustrations, extensive bruising or ecchymosis here, and blood-filled blisters, extensive bruising here from these patients. Intramuscular hemolysis has been described from Greece. That's breakdown of red blood corpuscles in the bloodstream at a massive scale, and generalized muscle damage, releasing a lot of potassium, which can be dangerous, as in sea snake bites, and causing metabolic acidosis and acute kidney injury. And fatalities have been described from Vipera ammodytes, particularly in Croatia, rarely children, both in children and adults. So this is certainly a formidable venomous snake and one that should be taken seriously. However, if you read the Greek literature, you will see an emphasis that most cases of bites by this species cause only local envenoming, modest local envenoming. And this is my most recent case of snake bite in Greece, treated at a distance, as it were. And this is from Ios Island in the Cyclades. And this is the Amidites, Vipera Amidites does occur in Ios Island. And this is from Matt Wilson's website, a roadkill specimen from Ios Island. And they're said to be smaller. The island specimens are much smaller, and I'm sure their venom may be different as well. In this case, the patient was a young male herpetologist. He was bitten on the wrist and felt immediate numbness. There was swelling and tenderness of the hand, which extended up his arm. After only 10 minutes, he was seen by a local doctor in Ios, and that's incredibly quick treatment. So I think we have to give this local doctor top marks for accessibility. You can't get much quicker than that in treating snake bite. However, the doctor's treatment was somewhat irregular. He applied a local injection at the site of the bite. I'm not sure what it was, but it caused some blanching of the skin, suggesting vasoconstriction. So it may have been adrenaline. He also gave the patient adrenaline injection into the wall of his stomach. And he was given a very strong corticosteroid called methylprednisolone and told to take a course of tablets. And as I said a few moments ago, this is now extremely irregular treatment. The patient contacted me at that stage because he was a bit worried. And I recommended, first of all, he should stop taking the methylprednisolone and that he should travel to the nearest hospital, because I felt that he needed blood tests and possibly antivenom. I was aware that most bites by this species in Greece tend to be mild, but knowing the full spectrum of possibilities of envenoming, I was concerned that this young man was being treated in isolation already by a doctor who'd shown a slightly irregular or atypical style of treatment. Anyway, he traveled to Santorini Island Hospital, and the blood test there showed he had a raised white cell count, but the only treatment he was given was intravenous antibiotics, and he was observed for only 12 hours in the hospital before being discharged. And I was rather surprised by this treatment, but of course, the local doctors undoubtedly have vastly more experience of the usual severity of these bites, so I'm certainly not in a position to criticize. Here is the site of the bite, 10 minutes after the bite on Ios Island, and 6 hours later, that's the site where he was given this local injection that caused local blanching of the skin, and that's the bite mark. So I don't know what this injection was, but it's certainly not at all typical treatment, recommended treatment of snake bite to give anything by local injection. I don't think it was antivenom, by the way, and in any case, local injection of antivenom is not recommended. Now to the Bosnian adder and the common effects of envenoming by the Bosnian adder are these affect local pain and so on. You're now very familiar with that. But I've already mentioned the anaphylactic syndrome. Now, anaphylaxis is a rapidly developing and life-threatening complex of symptoms that most of us will associate with allergy, with hypersensitivity. I'm sure you're familiar. If someone who's hypersensitive to peanuts or something like that eats something containing peanuts, they may within minutes be on the edge of death, with symptoms ranging from shock, sudden fall in blood pressure, asthma, contraction of the airways producing wheezing, swelling of the lips, tongue, and gums, and upper airway, so they can't breathe, and other symptoms, diarrhea and vomiting and so on. So a very dramatic complex of symptoms that we normally associate with allergy, but it's also a very common early symptom in patients envenomed by European vipers, particularly Vipera berus, but also Vipera aspis, and also from what I've just been saying, Vipera amidites. And these symptoms can come on within minutes of the bite. That's very, very rapid. Systemic effects within minutes, undoubtedly caused by venom toxins that activate the endogenous pathways, resulting in release of mediators, not just histamine, but other vasoactive compounds. The rare effects of Vipera berus bosniensis are systemic bleeding and coagulopathy, acute kidney injury, particularly in children, cerebral edema, that's very rare, adult respiratory distress syndrome, various cardiac problems like the woman's ECG I showed you just now in England with the horrible looking electrocardiogram, and various other very serious effects. Now, neurotoxicity has been documented for Vipera berus bosniensis by my colleague Tamas Malina in Hungary. So here is the patient showing divergent eyes and drooping of the eyelids after a bite by the Vipera berus bosniensis. Now your special Greek meadow viper, Vipera graeca. There was a very useful review in 2011 by Kreshak and colleagues from Hungary, again with Tamas Malina as a co-author. Herpetologists are almost exclusively the victims of bites by this species because it's very reticent and it lives in remote areas. Only local, very local, mild local and systemic envenoming has been described, although in the historical literature there were some fatalities claimed particularly from this subspecies in Hungary. So this is the distribution of the different subspecies of Orsini's viper, Vipera orsinii, and the distribution of Vipera graeca. Now, as I mentioned, my favorite European snake, these two specimens were in Jersey Zoo, actually, some time ago. I was able to photograph them. The distribution is very localized. It's an impressive looking snake, and some have classified it with Macrovipera lebetinus, which is interesting, which is quite a formidable snake from further east in distribution. This famous herpetologist, Dr. de Bedriaga, is reported to have, by Bullinger in the Snakes of Europe, this is early in the 20th century, on Milos, this much dreaded snake, the bite of which is probably as bad as that of its Indian ally, the daboya. Well, I've not been able to find any evidence to support that. In fact, I'd be very interested to hear any reports of bites or envenoming by this very impressive looking snake. I'm sure there have been bites, but unfortunately none has reached the literature. And now the Ottoman viper, as you know, quite widely distributed in northeastern and the eastern islands of Greece. Very few reported bites, none that I could find from Greece. There are reports of rare cases of massive local swelling with increased creatine kinase. That's a muscle enzyme suggesting muscle breakdown and even coagulopathy. But this really doesn't seem to be an important cause of envenoming. And in any part of its distribution outside Greece, there's very little information available. And finally, now the the only non-viperid snake and venomous snake of any real significance in Greece, the eastern Montpellier snake, Malpelon insignatus, quite a distinctive looking, highly active snake. This is the western species, photographed by my Spanish friend Javier José Carrasco Araujo, and showing the rear fang through which it inoculates its venom. This is my friend's hand after being bitten by the snake. So local swelling is quite commonly described after bites by Montpellier snakes. But the really exciting thing is that there are well-documented, authenticated cases now from Spain and France of classic neurotoxicity, that's ptosis and so on, external ophthalmoplegia, caused by the venom of this non-viperid snake. So extremely interesting. I don't think it's high risk, but I think it should encourage people to handle the snake with caution. So just a few summarizing remarks now before I finish. Bites by indigenous Greek species, what are the special considerations? People may question whether species diagnosis is important. I think it is. I think it's important to distinguish between different venomous snakes because their potential for causing harm is so variable, as I've shown you. The trouble is that since bites are rare, doctors may lack training and experience to deal with them. And I've recently been, again, going through the literature. There's very little in the way of publications, at least in English language, accessible from Greece. I think the potential severity of some of these bites may be underestimated. I still believe that any bite by Vipera amidites is potentially dangerous. There's a habit of inappropriate surgery. I've seen mentions of fasciotomy, which is now widely discouraged in snakebite, largely because you can't diagnose compartmental syndromes clinically in snakebite patients. You have to confirm by invasive measurements. And antivenom may not be available locally. This is what I've discovered through my distant management of snake bites in Greece. I'm not sure whether there is expert advice available from national poison centers in Greece. There certainly should be. And now in the case of bites by exotic species in Greece, and it was through this that I first came into contact with Thomas. We were put in contact not over a bite by a Greek snake, but by an exotic snake. And this is something which I've had a great experience in UK with some of my colleagues, Alistair Reed and Michael Edelston and David Lelou and others. You usually know the species of a bite by an exotic snake because the owner usually is quite certain which species it is, but you don't know its geographical origin. And now in view of the demonstrated variation in clinical effect geographically, this becomes increasingly important. This is a really horrible thing. People are hybridizing snakes. I've got cases of hybrids, not just between Bitis gabonica and Bitis arietans, between rattlesnakes, but even between Asian and Indian spitting cobras. And I think that an occasion is going to arise where it will be impossible to know which antivenom to recommend if you're dealing with envenoming by a hybrid snake. Some of the names used by amateur snake keepers are very misleading. For example, hognose vipers are actually pit vipers from Central America, but hognose vipers are usually just hognose snakes. They can cause quite unpleasant local envenoming. They're a colubrid snake, but certainly not a viper. The victims may be very reticent, very unwilling to come to hospital or to give details of what's bitten them, because at least in the United Kingdom, many of these venomous snakes are kept illegally, so that the owners don't want to be charged for this. This is a particular risk of bites by exotic species, that amateur snake keepers may well have been repeatedly exposed to venom, even by tiny bites, or perhaps not even bites. They may have become hypersensitized to the snake venom, so that when they are finally bitten, the clinical effect is not of envenoming, but is of hypersensitivity, of anaphylaxis. And this can be very confusing for the doctor to distinguish envenoming from anaphylaxis. There may be delays in obtaining specific antivenoms, and there's a great risk of bites by species that bite very rarely in the wild and about which very little is known. And these are just some from my own experience. Proathris superciliaris from Southern Africa, rare Trimeresurus species, for example, from the Philippines, Bitis parviocula, the Ethiopian mountain viper, and Protobothrops mansionensis. These snakes are extremely popular in captivity and are likely to bite reptile keepers, but there's very little known about the effects of their venoms. So thank you for your attention, and I'm sorry to speak for rather long. Thomas, you certainly won't invite me again. I've gone over my time, apologies, but thank you for giving me this opportunity.
Thank you very much. Thank you very much, David. I don't know if other people would have questions. I know it's really late. We've been here for about 6.5 hours, but I wanted to ask one question before moving on to other people, if there are any. How long is too long to wait when it comes, for example, I've had a person I know who was bit by a Mycurus, a coral snake in Florida, and she didn't get any symptoms up to 3 days later. She was hospitalized preventatively, didn't get any antivenom and so on because no symptoms did arise. And then she started coughing, essentially blood at some point when she was out 3 days later.
Well, that sounds a weird case, Thomas, because coughing blood is not a feature I associate with coral snake envenoming. I wonder whether something else was going on there. Certainly, development of neurotoxic features can be delayed. I mentioned the delayed cases, certainly from crates from Bangor, it can be 18 hours or longer, also Mycurus, and delayed development of bleeding and clotting problems from the South African bomb slang, Dyspholidus typus. There are examples after more than 24 hours, the patient suddenly starts to bleed. I think that 24 hours is a reasonable period, a compromise for observation. But I suppose the doctor should always warn the patient to make contact again if any symptoms develop. But I'm very unhappy with your description of coral snake symptoms. I wonder what was really going on there.
Yeah, that was actually very weird because we were in contact at the time, and yeah, that's what was it, coughing blood, which didn't make a lot of sense for such a bite. Would anyone else like to ask any questions, maybe?
Thomas, I'd love the opportunity in the future to correspond with Greek physicians, surgeons who specialize or are interested in snakebite. I speak from very great ignorance on this topic, but I'm very keen to learn. And considering you have such a fascinating herpetofauna in Greece and its islands, I think there's a lot to be learnt here. And very often observations that are not published, but they're well documented by the particular medical person who looks after the patient. So I would be very happy for you to pass on my email contact to any Greek clinicians, and also very happy to discuss particular cases as well.
Because we were working on a project with Ilias Strachinis, who is essentially the herpetologist of Greece, #1 here. And we were on Milos, right? So handling, essentially not free handling, but we were exposed to and had contact with the Milos viper and so on. And we were discussing how nothing has been documented. We haven't heard of anyone dying from the Milos viper. Then again, maybe there's variability when it comes to the red specimen as opposed to the gray specimen and so on. But no literature there at all, sadly. So we will certainly do our best to address that. And if we come across any information or physicians, like you said, I will make sure to pass your email on and maybe your phone number as well, because most of these cases are quite urgent.
Well, thank you again for making this contact and stimulating me to look at the literature for Greek herpetology and Greek toxinology as well. Thank you.
Absolutely. Thank you very much. Thank you very much for being with us. I would like to thank everyone who has participated in this and for making this happen today. This should conclude the entirety of the webinar. I don't know if anyone wants to say something, otherwise this should be the end of it. And people can go and eat and whatnot, because we've been here since the morning. Okay, that settles it. So it was great speaking to all of you. We feel lucky because we really don't have a lot of research going on. We don't have a lot of experience in this country. There's certainly a lot of students, a lot of people, different areas, from Calvete's perspective, from Mr. Gutierrez, or from your perspective as well. And essentially, yes, we hope that this changes in the future. We pick up projects, we cooperate, we collaborate, and yeah, why not work together and we can learn from people who have actually gone a very long way when it comes to
you know Juven, the organization, which is on European basis and everyone can join it.
No,
and it's for interchange of students and researchers from one country to the other, and there is financiation for every year from the from the union.
I was not aware of it personally. Maybe other people. We tend to have an issue with financing and funding when it comes to all these things, because no one thinks about the snake bites here or the potentiality essentially when it comes to venoms.
Okay, I will ask the manager of the network to send you information so that you can just make it publicly available in your country, and everyone that wants to join is welcome. There are no rules at all for joining.
Fantastic.
Except that the British are no longer allowed in.
No, the British are allowed, but with a high fee. Economical fee.
I'm feeling very bad about that as someone who voted against Brexit. It's very unfair.
Okay, so thank you to all speakers, all attendees, and yes, look forward to maybe organizing something like that down the line in the future with a different, very more compact segment and topic and so on. But as a first initiative, I think it was a success. So I think I speak from everyone from over here.
Thank you.
Thank you very much.
You're welcome. You have a good day.
Thomas, you did a great job.
Thank you.
Bye to everyone.
Bye.
Bye-bye.
Bye.